Assalamu alaikum wa rahmatullahi wa barakatuh. Esteemed guests, our global viewers who are watching us from around the world, welcome and thank you for joining us for the day 3 and the final day of Riyadh Global Medical Biotechnology Summit 2021. I'm Muhammad Dakhil, your Master of Ceremonies. We have a very productive day. 20 speakers joining us from around the world, from academics, experts, and thought leaders in the field of biotechnology gathered in this summit to push the boundaries of our existing thinking and practice and propose innovative models that will impact the country, the region, and the rest of the world, and will definitely have a direct impact on health and economy. To kick off the day, we welcome Dr. Paolo Martini, Senior Vice President of Moderna, to deliver his keynote presentation, moderated by Dr. Manal Al-Amri, Chairman of Innovation and Entrepreneurship Department, and the Head of Development and Medicine at KMARC, Ministry of National Guard Health Affairs. They will discuss in depth the application of biotechnology towards diagnosis and treatment and how the implications of biotechnology pervade all area of medicine. I welcome you both to the Riyadh Global Medical Biotechnology Summit. The mic to you, Dr. Manal. It's a great pleasure to have you and Dr. Paolo with us today. The stage is yours.
Thank you, Ria. Good morning, good afternoon, everyone. It is my great pleasure to introduce our keynote speaker for today, Dr. Paolo Martini. Dr. Paolo Martini is the Chief Scientific Officer for Rare Diseases, Hematology, Rare Alliances at Moderna. Dr. Martini has more than 20 years of experience in drug discovery, working on molecular mechanism underlying monogenic and multigenic metabolic and fibrotic disorders. In addition, his laboratory is focused on identifying novel therapies and applying translational approaches for drug development in rare diseases and hematology disorders. Dr. Martini currently is exploring the identification of several targets and key animal models for translating RNA in human patients. Previously at Shire Pharmaceutical in Lexington, Massachusetts, as a Senior Director of Discovery Biology and Translational Research in Massachusetts, he focused on fibrotic diseases of muscle, kidney, skin, lung, and bone marrow, and metabolic liver diseases, with particular emphasis on different therapies, modalities for pathways modulation. In addition, Dr. Martini has been supporting phase 1 and phase 2 clinical studies for lysosomal storages and chronic diseases. He has also worked at EMD Siriano prior to Shire Discovery Research. After graduating at University of Milan, Italy, he moved to Germany for postdoctoral work at Schring AG, now known as Baylor, in Berlin, Germany, and then to the University of Illinois in Urbana-Champaign, USA. Furthermore, Dr. Martini has been collaborating with organizations supporting research and clinical development of rare metabolic disorders and fibrotic diseases, and served as a member of the Scientific Advisory Board of the Keystone Symposia. Thank you. The floor is yours, Dr. Martini. Welcome, and thank you very much for being with us today.
Thank you so much for having me. It's an honor to be able to speak to all of you today. I hope to be able to focus on a few areas so you will understand why we think that the messenger RNA is a really flexible platform for different therapeutic areas. So if you go to the next slide, sorry, I cannot control the slides from here, but I will ask for the next slide again. Thank you. The next one, please. So, as you know, Moderna, in the past 7 years, 8 years, we actually started around November 2011. So, it's been a very intense ride, so to speak. We have been focusing exclusively on messenger RNA. The reason why we wanted to focus on messenger RNA because we think that being sort of the software of human life by copying the DNA and translocate it into the cytoplasm, it allows to basically translate many different type of proteins. So it has a flexibility in that sense that give us the opportunity really to express any type of proteins without the limitation of the technology. Like, for example, in enzyme replacement therapy, you can only express a majority of soluble protein or only peptides, etc. While the messenger RNA gives you the opportunity really to exploit any type of protein, talking about transmembrane, mitochondrial, secreted, intracellular, any really type of protein in the different organelle. If we go to the next slide. And that's why we have the opportunity really to focus on so many different targets that can apply to many different therapeutic areas. If you look in the turquoise color, for example, those are all potential vaccines that we have in development. We're also looking at secreted proteins, intracellular, intramitochondrial, and also many different applications. So we look at oncology by overexpressing, for example, checkpoint inhibitors and all these kind of things. And also we have the ability to produce any type of protein even locally for maybe local activity in some foci during, for example, surgery due to myocardial infarction. So you can, for example, inject VEGF and reconstitute the revascularization of the ischemic tissues. So there is an incredible use for messenger RNA. The only problem with the messenger RNA, as you know, that is very labile and not very stable. So we have been doing a lot of work at Moderna in order to understand how to make the messenger RNA suitable for the human body basically as a drug. We can go to the next slide, please. So we obviously have different requirements from different modalities. What does that mean? If we do prophylactic vaccine, cancer vaccine, intratumoral injection, localized regenerative medicine, we can do systemic secreted protein and systemic intracellular therapeutics. But all of these, next slide, please. All of these, next again, sorry. All of these is in a dose-dependent protein expression. The beauty of the messenger RNA is that if you give X amount of messenger RNA, you always get X amount of protein. So we can control the dosing of the messenger RNA in a very specific manner. Obviously, we have, back one, please. Yeah. Obviously, we have different ways of delivery system. It can be local. And when we're local, we are obviously looking at prophylactic vaccine, cancer vaccine, immuno-oncology, where we can actually inject directly into the tumor. Or we do systemic delivery. This is more suitable, for example, for rare genetic disorder or a systemic disorder. We also have the advantage, back one, sorry. We also have the advantage of the limited duration of effect in some instances. For example, for the vaccine, we do a couple of injections and then you have a reaction to it, which is great. It's building antibodies against, for example, the spike protein. For instead, rare genetic disorder, we use a chronic dosing, which is actually allow us to maintain the patients with a replacement gene using, for example, the wild type sequence, so we can reconstitute the original phenotype of the disease. Obviously, we also have a different type of reactivity to the messenger RNA. A messenger RNA is known to be very immune reactive because it's generally recognized as a virus, as we know. In the case, obviously, of vaccination or cancer vaccination, obviously, the immune activation is desirable because, in fact, we want the immune system to recognize the foreign protein to start to build antibody against it. Instead, for example, for localized regenerative therapeutics or systemic or rare diseases, we obviously want the immune activation to be completely dampened. And so we do have, and I'll show you later in some of the slides, we do have a way to make the messenger RNA immune silent. If you go to the next slide. In order to build, obviously, a strong platform, we have to look at every aspect. Obviously, messenger RNA is delivered via lipid nanoparticle. And as I mentioned to you, because we have different ways of treating patients, either if it's vaccination or rare genetic disorder or oncology, etc., we do actually have to work quite a bit on the messenger RNA itself. So we do actually have an incredible effort from Moderna in understanding how messenger RNA works, how we can deactivate the messenger RNA against the immune system, how we can increase, for example, the translation of the messenger RNA once it's transduced into the cells, and how we can optimize the amount of protein related to the messenger RNA that we give to the patients. Delivery obviously is the most important factors because it's the only way not only to protect the messenger RNA by encapsulating in a lipid nanoparticle, but it's also a way to transduce specific organs with the messenger RNA so that it can be translated into the targeted protein. And so we do a lot of work in having our own proprietary lipid nanoparticle that obviously they have the ability to transduce this different organ and different tissue, and I'll give you some example later. Manufacturing of the drug is paramount. We have been putting a lot of effort in understanding how we can actually create a drug and how to scale it so that it can be introduced into the clinic and eventually into the market. So we have built an incredible infrastructure at Moderna to be able to actually produce as many messenger RNA as we want. We generally produce about 1,000 RNA per week, and we can scale up to 20 to 40 messenger RNA even for clinical trials and eventually for the clinic. So I think having the control on the manufacturing process gives us the opportunity to really create a drug that is not only a suitable target, but is also very pure and superior compared to many other messenger RNAs. Can we go to the next slide, please? So today I will talk to you about how we engineer our messenger RNA delivery vehicles, how we make some of our vaccine. As you know, now we are very famous for our COVID-19 vaccine. And I'll talk to you a little bit about how we manufacture it in order to avoid immune recognitions and also to achieve cell specificity or organ specificity for the expression of the targeted protein. So if you go to the next, please. So obviously we target different tissue with a multiple route of administration. And this is important because depending on the therapeutic areas, we need to have different reactivities and different, obviously, ways of triggering a response from the body. For example, for the vaccine, we use intramuscular injection. As you know, probably many of you have already taken the Moderna vaccine, and you know that is a shot in the arm, followed by a booster shot within 28 to 35 days post the first. For intracardial injection, we actually use a naked RNA, and this is a collaboration with AstraZeneca, where they are currently running a clinical trial, phase 2, in myocardial infarction patients, and they are injecting naked RNA in the ischemic areas using VGF to try to reconstitute the revascularization of the ischemic areas and potentially restoring functionality of the tissue. For intratumoral injection, right now we are obviously focusing on those solid tumor that we can achieve, we can reach with the needle, so that we can perhaps reconstitute some checkpoint inhibitors so that immune cells can migrate towards the tumor, or many other different ways so that we can utilize the immune system to start to target the tumor itself. For a rare genetic disorder, which is actually my specialty, we do IV infusion. And the reason is that by giving a systemic by using a systemic delivery, we have the ability not only to reach the liver as a major organ for receiving the messenger RNA, but also we have the opportunity to target different organs, as we know that in many of the genetic disorder, many organs are implicated in that particular disease. So different route with a different application, if you will. Next slide, please. So now I'll talk to you a little bit about how we make our vaccine, which I think you're very interested in that. So we go to the next. The mechanism of making a vaccine is actually very simple. We analyze the sequence of the virus, and in many instances, for example, in many of our vaccine, and this is the flexibility of this particular technology for vaccine production, is we have the opportunity really to encapsulate different messenger RNA that will create the spike protein, and then by giving basically the messenger RNA to the human, the human body will produce the spike protein, which will trigger obviously an activation of B cells, T cells, etc., by producing obviously the vaccine. And how does that happen? It's once the messenger RNA has been injected and then gets translated, obviously antigen-presenting cells are attracted by the virus, and then they produce a response. Can you go a little slower, please, with the slides and wait for me? Thank you. Once the messenger RNA is given, as I said, is injected, the cells actually, the antigen-presenting cells have the ability really to translate the spike protein. Can you go back one, please? Have the ability to produce the spike protein, and then that activate the B cells that have the ability really to recognize the spike protein and start to produce antibody. Next slide. Once obviously B cells have the ability to transform into plasma cells and start to build the memory of the antibodies, so that once we have the next injury from the virus, we actually have a recall of the antibody. Next slide, please. Once also the antigen-presenting cell have the ability also to recall T cells or or whatever we call the memory cells, and these actually allow us to start. Can you go back, please? Can you wait for me to move the slides, please? Once the T cells have been recognizing the spike protein, they start to build a memory of the. They start to build the memory of the T cells. Next slide, please. So obviously lipid nanoparticles are a key to deliver messenger RNA. And in the case of the COVID, we use COVID-19, we use lipid nanoparticles that are very immune reactive, so that the moment that you inject into your into the body, into your muscles, there is a recall of all these inflammatory responses induced, and there is a recall of all these type of cells that they have ability not only to react to the lipid nanoparticle, but eventually to react to the messenger RNA, which is also in this case very immune reactive, and eventually to the protein. And the reason why we use lipid nanoparticles that they're immune reactive is that we want really to recall all the immune cells to the area of injection, so that all the cells that are recruited, B cells, T cells, plasma cells that they are recruited into the area, are actually now able to be present when the spike protein has been produced by the local cells. And then we have an incredible repertoire of antibodies that has been produced against that particular antigen. By combining different messenger RNA, we can create all the full structure of the virus, And that generally is the best way to go because that creates a series of different antibodies towards different areas of the spike proteins, allowing, for example, in the case of the COVID-19, to have a repertoire of antibodies that can target many different variants. And we have seen that with the spike of the Delta variants, for example, our vaccine was still very effective in producing an immune response against the Delta. And we were able really to cover many of the different variants because the way we designed the spike protein and the way that the spike protein is produced by the cells in a very incredible fashion with a perfect structure that obviously trigger an incredible amount of different antibodies. Obviously, the lipid nanoparticles are required in order to protect the messenger RNA. We know the messenger RNA is very unstable, particularly when it goes into the system. So if we use a different lipid nanoparticle for different modality or for different therapeutic areas. For rare diseases, the lipid nanoparticle that we use, and you can see the schematic in here, it's a very stable and a very non-immunogenic lipid nanoparticle. We use aminolipid that first of all, they are ionizable, so which means that once they are integrated within the cells, being ionizable and being in an aqueous environment will be dissolved very fast. And then we use the typical component of the lipid nanoparticle. They're typical like sterols, like cholesterol, phospholipids, and obviously we have some of the PEG lipids. Our PEG lipids, though, are unique. As you see that they are decorating in orange the structure of the lipid nanoparticle, they are also hydrolyzable themselves, which means that they maintain the stability of the lipid nanoparticle into a vial, but the moment that they go in circulation, they really shed from the lipid nanoparticle, leaving the lipid nanoparticle basically sort of naked, if you will, but avoiding, therefore, any potential blockage by IgG that we have generally in our body against the PEG, because we use on a daily basis toothpaste, cream that they contain a lot of PEGs, and therefore that could be an hindrance to the ability of the lipid nanoparticle to deliver the messenger RNA to the targeted organ. So by removing the PEGs in the system, in the blood, basically allow us to have a naked lipid nanoparticle that then has the ability to bind to receptors and then to be internalized into the cells. We go into the next. Next slide, please. So these are the complexity of the lipid nanoparticle is actually quite unique. As you see, they're generally running in a range of 70, 80 nanometer in size. And the components, the different components are obviously the messenger RNA that is encapsulated inside. And then we have phospholipid, ionizable lipid, as I mentioned to you, sterols, and the PEG. Once the lipid nanoparticle come together, they create these very nice spheroids that protect the messenger RNA inside. And once the PEG is shared, they have the ability, as I said, to bind to the receptor and eventually to deliver the messenger RNA intracellularly. And, they're a very small component, if you will, but they create a very nice circular structure protecting the messenger RNA, which is negatively charged, while the lipids are positively charged, so that once they come together, they really create these beautiful spheroids that they're very stable, particularly because of the PEGs. Next slide, please. One of the mechanisms that we have understood so far, and for some of the lipid nanoparticles that we are blood, basically shed from the lipid nanoparticle, and now the lipid nanoparticle has the ability to opsonize with ApoE, and eventually create a structure that is very similar to an LDL VLDL, which can be taken up by the LDL receptors within the cells and create the endosome for eventually the release of the messenger RNA intracellularly. Excuse me. Next slide, please. So once the lipid nanoparticle get to the desired cell type, there are many factors that obviously influence the ability of the messenger RNA to be translated into a functional protein. And so first of all, we have key factors, the internalization, and then the endosomal escape, and then the ribosome engagement, which obviously allow then for the translation of the desired protein. And the beauty of the messenger RNA that once the messenger RNA is released by the endosome, you can actually translate any type of proteins within the cells or secreted proteins so that you can even target distant organs from, for example, from the liver being the major organ receiving the lipid nanoparticle. Get to the next, please. And this is, I want to show you what is the efficiency of endosomal release from our lipid nanoparticle. On the left panel, that's the industry standard LNP. And on the right panel, we are looking at modern LNP releasing GFP messenger RNA. If you see this red dot that you see in the majority in the left panel with the industry standard, these are still LNP that are engaged with messenger RNA that haven't been able to escape the endosome. If you look instead on the right and you see a fully translated GFP and no red dots, it suggests basically that our LNP have the ability really to disrupt the endosome and to release nicely the messenger RNA into the cytosol and having a full translation of the targeted protein. And that's why we see all this beautiful fluorescence, GFP fluorescence. If we get to the next slide. So so we resolve endosomal escape and how efficient it is. Actually, it's pretty efficient. So if we go to the next slides, you see that we have about 25 times improvement in endosomal escape using our lipid nanoparticle. And you can see in the lower panel how much of the GFP has been translated versus, for example, the industry standard, where you still have a lot of lipid nanoparticle engaged with the RNA that they haven't been escaped from the endosome. And so we do actually have a superiority in our lipid nanoparticle in releasing the messenger RNA that eventually gets translated into a protein. If we get to the next. So now ribosome engagement is important. We do actually engineer our messenger RNA, particularly in the secondary structures, so that we can favor the messenger RNA to bind to a ribosome and eventually translate the protein. Generally, our messenger RNA is highly translatable because we create a secondary structure that create a higher affinity compared to the endogenous messenger RNA in order to translate then the protein. We'll go to the next. And here, as I was mentioning, we have 25 times better endosomal escape, and then actually we have at least 2 to 3 times better translational engagement, suggesting that by engineering the secondary structure of the RNA, we can actually have a better translation and a better utilization by the ribosome of our messenger RNA. Next slide, please. So now, next I want to talk to you a little bit about avoiding the immune system because it's important. As I mentioned, while in the infectious diseases and vaccine, we want to have a very much reactive messenger RNA. Can you go to the next? So obviously in rare diseases, we want to avoid very much any type of immune reactivity to the RNA and to the lipid nanoparticle, so we can concentrate on the translation of the targeted protein. Generally, RNA, as you know, the way it is, particularly if it's an RNA with a usual nucleosides, once it's given, has the ability to bind to toll-like receptor 7 and 8, and by binding to toll-like receptor, it does trigger the immune system in a response, in creating an interferon gamma and interferon alpha response with a cytokine cascade, and eventually a call for the immune system to start to recognize the cells that have been presenting the new messenger RNA and try to eliminate them. Also, there are other mechanisms, for example, of recognition of the messenger RNA that would dampen the ability of the messenger RNA to be translated, like the RIG-I, which recognize a small double-stranded RNA, and by triggering interferon 1 response, it will recall for basically degradation and elimination of the messenger RNA. So what do we do at Moderna to avoid the immune reactivity of the messenger RNA? Next slide. What we have found that by changing the uridine, and we know that the difference between DNA, which we know that is not immune responsive, versus the RNA is only one nucleoside difference, and it's the uridine. So we looked at the uridine, and we realized that by looking at the cascade of the synthesis, so what we did, basically, we changed the uridine in our synthesis of the messenger RNA with either a methyl pseudouridine or a methoxyuridine. By changing the uridine, we really have the ability to block the immune reactivity of the messenger, and therefore will be actually translated fully by the cells. So if we go to the next slide, we did some studies, obviously, in mice. We wanted to understand if changing the uridine, we would have a different response in terms of activated B cells frequency. And we saw that by the time we actually modify the uridine and substitute the uridine with either the methoxy or important, because at this point it means that in a genetic disorder where we want the RNA actually to be translated well and efficiently for several hours or days, we really need the collaboration from the immune system to avoid the degradation of the messenger RNA in a very fast manner. And therefore, we actually have now a therapy for genetic diseases in terms of replacement, because the RNA itself is not reactive with the immune system and therefore can be translated fully by the cells. Also, as you see in the green bar, that's just a messenger RNA that is highly purified. But the moment that you substitute the uridines with pseudouridines, that's where you completely lose the ability to activate the immune system. Next slide, please. So, in summary, we have created an effective messenger RNA that they do produce antibody, and that's what we use in vaccination. And generally, these the pseudouridines are not obviously contained in the vaccine, but just in the rare genetic disorder messenger RNA. By substituting globally the uridine, we actually have an immune-silent messenger RNA. And the process that we use, which is highly pure messenger RNA, gives us the opportunity even to further dampen the immune response. Next slide, please. So last things that I want to tell you before I give you an example on how well the messenger RNA works in preclinical studies, and then now obviously we are in the clinic also in genetic disorder, I think is how we do engineer the messenger RNA to achieve a cell type specific expression. So we get to the next slide, please. And as you can see, we have every possible feature of the messenger RNA that is needed for the human body to translate a perfect protein. So the messenger RNA has a 5' UTR, is capped, the coding sequence, the 3' UTR, and the poly A tails. Next slide, next. And so we obviously have a fully translated messenger RNA. But we have because we have all this structure, this perfect structure, we can measure translation, initiation fidelity. We can actually look at a protein per unit time. We can actually look at the half-life of the messenger RNA. And then what we have been doing recently, we have been looking at cell specificity, how to, for example, have a perfect translation in certain organs versus versus, for example, blocking completely the translation of the messenger RNA in a different organs. What do we do for that? We just, in the 3' UTR, we encode microRNA recognition sites that they are specific for a specific organ, meaning that if I want my messenger RNA to be translated into the liver only, I can put some microRNA recognition sites in the 3' UTR. So by the time the messenger RNA gets delivered to different cell types because of the lipid nanoparticle or the different organs, in those organs that they contain a particular macroRNA that has the ability to bind to the messenger RNA and put it into the degradation pathway, we actually see no translation of the messenger RNA in that particular organ. So that allows us to do in a clever way a sort of a specific translation in a specific organ and avoid the translation in organ like the spleen, for example, where the messenger RNA might activate an immune response in precursor cells or any type of immune cells containing the spleen, and therefore avoiding the initial immune responsiveness to the protein that the messenger RNA could trigger in that particular organs. And so we have the ability really to modulate in an organ-specific manner the translation of the messenger RNA. So next slide, please. So in summary, I think we have different lipid nanoparticles that they are optimized for obviously each route of administration and desired cell type with different reactivity to the immune system. So for vaccine, we use a basic chemistry for the uridine, for example. For a genetic disorder instead, or oncology, we use instead different chemistry that allow us to translate fully the RNA and to sustain the RNA because it's not dampened by the immune system. We actually, by incorporating the uridines, we also, the pseudouridines, also we reduce the level of a double-stranded RNA that could also trigger a degradation response of the messenger RNA. Once we synthesize the messenger RNA, which is a fully normal messenger RNA as you would find in nature, we actually engineer the coding sequence and UTRs as well in order to promote the perfect output and translation by also using algorithms that they look at the secondary a very specific translation in a specific organ. So we think now that by looking carefully at the messenger RNA, we can actually have it tailored for different therapeutic areas and different route of administration to give us the opportunity to really be therapeutic in once we get into the clinic. You've seen the vaccine being the COVID-19 has been producing an incredible vaccine, very effective against the COVID-19, and now we are in the clinic with a couple of programs, one for propionic acidemia and one for methylmalonic acidemia, where the clinical trial has started this past springs, and we are actually already having good results in terms of safety and tolerability. We go to the next slide. I want to give you quickly an example of a genetic disorder that we are working on, and we just filed an IND and we got the okay to proceed from FDA, which is the glycogen storage disease type 1. which is probably many of you are familiar, given that there are many rare genetic disorders in the kingdom, and in my conversation with the incredible physician that you guys have, we have already discussed the ability to potentially bring in this medicine also to the kingdom, and to be able to treat some of these disorders. So GSD1A has mutations in the G6PAs, and is a life-threatening disease because it creates severe hypoglycemia in these patients and has a long-term liver and kidney damage. And many of these patients eventually start to develop adenomas and hepatocellular carcinoma as a result of the mutation in the G6PAs. The incidence is, I think, is 1 in 100,000 births, which suggests that there are about 2,500 patients in the US and then 2,700 patients in the EU5. At the moment, there's no therapy besides basically diet. The diet consists for these patients in consuming a huge amount of quantity of cornstarch in order to sustain a normal production of glucose and to avoid actually hypoglycemia that eventually will take them to death. The mortality of these patients is high the moment that they start to go into hypoglycemia, and the hypoglycemia is not controlled. And so if they don't consume enough cornstarch during the day, they can actually go in a severe hypoglycemia and die. And so many of these patients undergo a liver transplantation at a certain point because at the moment is the only way to really support, besides the diet, which eventually does, unfortunately, still produce adenoma and hepatocellular carcinoma in the liver of these patients. Go to the next slide. So the correction basically comes from messenger RNA by replacing the glucose cis-phosphatase. It's an important enzyme because it controls, obviously, and not only the glucose in the body, but also can produce lactic acidemia or hyperlipidemia. In fact, many of these patients, once they have a mutation, they actually have a very fatty liver and with a greater accumulation of triglyceride in the liver. So it's an important enzyme, and by replacing this enzyme, we are hoping to restore euglycemia, avoid lactic acidemia, and more importantly, to deploy triglyceride from the liver. Next slide, please. So basically what we do, we encapsulate in lipid nanoparticle the messenger RNA, which obviously contain a pseudouridine in this case. The lipid nanoparticles are specific for genetic disorders, so they're delivered systemically. And once they are taken up via the LDL receptor into the liver, they have really the ability to release the messenger RNA and then to have a functional human G6PAs, the glucose cis-phosphatase, that has the ability to restore, obviously, the function of the liver, in this case, by producing normal level of glucose and by blocking basically all those mechanisms, the University of Lyon. But obviously, the first things we do once we get a messenger RNA that encode for the protein, we test it in vitro. So we see that in HeLa cell as well as in patients' fibroblast, if we reconstitute by giving the G6PA's messenger RNA, we actually see a very nice expression of the protein. We do actually have the localization, which is in the endoplasmic reticulum, and this is exactly where the protein should be translated and homed. And we actually can measure activity, as you see, once we reconstitute the G6PAs, we actually measure quite a bit of activity in the cells. Next slide. In collaboration with Fabienne Rajas at the University of Lyon, she has a model for a liver model for the G6PAs. So it's a liver knockout of the protein. And as you can see, in the center, when we look at liver morphology, you can see that the liver of these mice are really fatty and contain a lot of triglyceride. And the model has the typical fasting blood glucose pattern of a human, as well as there's a lot of accumulations of glycogen in the mouse. So we go to the next slide. So if we give, obviously, in a dose-dependent manner, messenger RNA to the mice, we actually see that we have an increased enzymatic activity that is proportional to the dose. So X amount of messenger RNA will produce X amount of protein with X amount of enzymatic activity. And you can see that enzymatic activity is very much comparable to protein expression as we see in the mouse model. As you can see, right panel versus left panel, where we look at activity versus expression. It's a very relatable. Next is the only GFP messenger RNA. You see that the liver is still very much full of fat, and it's the typical yellow steatotic liver. Once you give messenger RNA in a dose-dependent manner, you actually normalize the liver to looking like a wild type from these mice. These happen within 24 hours, so you can imagine how fast the messenger RNA gets translated into a protein, and how fast these triglycerides are deployed from the liver once they have an active enzyme in the cells. And so this is beautiful because we can actually have also acute intervention, if you will, because we see activity within 24 hours that is pretty dramatic. Not only normalize the triglyceride, but also normalizing the liver weight that gets close to basically wild type. Next slides, please. We look at different metabolites, obviously, as part of reconstituting the glucose 6-phosphatase. So we look at glucose, and we see that we can normalize glucose 6-phosphate in a dose-dependent manner. We also normalize glycogen by reducing the level of glycogen. And as I showed you, and you saw also from just looking at grossly at the liver, we really deployed triglyceride in a dose-dependent manner. And all this data was reproduced many times, not only by the laboratory from Fabien Rajas in Lyon, but also by us in Moderna. If we go to the next slide. Also, we have the ability not only to decrease this biomarker in the organs in the liver, but also systemically. So if we look at the blood glucose and the circulating triglyceride, and we actually see that we do have reduction. We also measure liver enzyme because these patients having this such a high accumulation of triglyceride generally have elevated liver enzyme. And by giving the messenger RNA, we actually normalize the liver enzyme like ALT, AST, or gamma GT, and suggesting that we do reconstitute a normal phenotype in this animal with normal enzymatic liver activity. Next slide, please. And obviously, we wanted to understand what is the duration of effect after a single dose of messenger RNA. We always give a human messenger RNA to mice just because we want to see not only immune reactivity, but we also are interested in understanding how fast it gets degraded, because that will be eventually our therapy. So we always use a human messenger RNA. As you can see from day 1 to day 15, you can see that the level of blood glucose is normalized after day 1, and then suddenly it decreases up to 11 to 15 days. So we have a duration of effect in mouse about 15 days. Given that we give always a human messenger RNA in a mouse environment, we generally know that species-specific protein generally have a longer half-life, so we hope that once we get into human, we can really sustain a 15 days response in human. Therefore, then basically giving a therapy every 15 days for these patients. Next slide, please. More importantly, once now that we established the regimen, we actually looked at the ability of reproducing doses in a chronic manner. So we dose every 12 days, in this case, every 11 days, given that that was when we saw in mouse protein and activity disappearing. And we see basically that even after the 5th dose, we normalize blood glucose. And this is important because even after the 5th dose, we haven't seen a reduced effect of our protein, suggesting that probably it is the therapy keeps working without being neutralized by potential antibodies. This is a liver knockout, so eventually given a foreign protein, you might have a production of antibodies. Instead, we see that we don't actually see antibody production, and we keep maintaining a reduction of a reconstitution of glucose activity in the mouse even after 5 doses. And we normalize the liver weight, as you can see, and more importantly, those are 2 examples, 2 different messenger RNA with different codon optimization, that they still give the same effectiveness in terms of deploying triglyceride as seen on the right panel on top. Next slide. So obviously we have a lead candidate. As you can see, the duration of effect is about, in the GSD1A mouse, between 7 and 14 days. And we see liver weight and morphology improvement. We see improvement in hepatic biomarkers as well as in sero biomarkers. But more importantly, we retain a therapeutic efficacy through repeated dosing. So right now we have filed an IND and we got the okay from FDA to go into the clinic. So stay tuned because we'll have these messenger RNA soon tested in human, and we'll see eventually the potential efficacy. This will be a very interesting program because if we can basically normalize the diet for these patients and restore normal life activity, I think it will be an incredible endeavor. We see in many of the animal studies a normalization of the phenotype in animal, so we hope that will translate well in human. And as I mentioned, the propionic acidemia and methylmalonic acidemia are actually clinical trial ongoing at the moment, so we are basically testing messenger RNA in human for rare genetic disease. I would like to conclude and acknowledge, obviously, all the people at Moderna that they worked on this project. Next slide. As you can see, it's next slide, please. You can see that it is, this is a few years ago. We were about 800 people. Right now, we are reaching almost 3,000 in a year and a half, basically growth as a result of the COVID-19 and also all the programs that are now into the clinic to support messenger RNA as a platform for different therapeutic areas. So it's a great effort that has happened in the past few years. And now we are proud to say that not only we have a very active vaccine and we are producing many others, as you know, but we also are exploring oncology, cardiovascular, and rare genetic disorder because we're starting to see the safety and tolerability of our messenger RNA and lipid nanoparticle, and so more to come, hope in the future. So thank you very much for your attention, and if you have any question, I'll be happy to take any question. Thank you.
Our sincere apologies, Dr. Martini, for being behind 15 minutes. And now we will have some, just a little discussion. It was a great presentation. Thank you very much. And I would like to invite my colleague, Dr. Abdelali Al-Hawdi. He's the director of the Biotechnology Park, and he is actually a geneticist by training. And I'm also a molecular geneticist, which is really, this is our interest in research. Dr. Abdelali.
Great, great. Thank you very much, Dr. Manal. You know, when I hear a presentation like this by my colleague and friend, Paulo, I just want to create biotechnology company today.
I agree with you.
That's really at the heart of our summit, is how the Kingdom of Saudi Arabia can create such successful biotech companies, including local and international partnerships with your company or other partners that are available and sharing the same interest. Clearly, Moderna has been really as one of the best examples on how biotech company can make not just an impact, but a global impact. Of course, clearly, like Paulo mentioned, vaccine development was one of the main reasons, but there are other reasons behind it. Other examples of similar successful global biotech company, for instance, BioNTech, through its partnership with Pfizer, and a number of others. Of course, I gave just these two examples because it is related to the current pandemic and vaccine development, but other biotech companies have been equally successful in other therapeutics area. Now, let me just say, and correct me or add to my current statement, what does it take to have a successful biotechnology ecosystem in any country? I was thinking about perhaps 3 to 4 ingredients of success. One of them is to start with great science. That is really key. 2, have a great management and also leadership team to take on that science to the next level. And 3 is, of course, to have what I would call an enabling environment or ecosystem. That enabling environment, in brief, I will try not to go over the 5 minutes we have. That enabling environment will include permissive, let's see, government regulation system, availability of finance and investment, and also agile procurement system. The last one I would add is strategic partnerships, whether it's local or global. What do you think, Paulo?
Before you start, I would like to add also a question. So because we have to end the session. Actually, we're going back to the Moderna, and because we have, as you know all, that we have high consanguinity rate at the Kingdom, and you did mention that you have a collaboration with Kingdom of Saudi Arabia. We would love to know more about this, and what do you have new in the pipeline regarding rare disorders or orphan diseases? Thank you.
Thank you. Thank you so much for the questions, and thank you, obviously, for having me. This has been incredible. As my friend Abdelali, and we can say definitely friend because we know now each other for a few years, and we have really the best intention in collaborating. We are discussing a collaboration because I think it's important that an institution like yours, with the state-of-the-art facility that you have and the mentality and the vision that you have, it fits very well with Moderna. Moderna started with 3 people in November 2011 in a basement, basically. And by 2015, we were developing, with my arrival, basically a portfolio of about 28 rare disorders that we are now conducting research in animal model, etc. And the expansion of it obviously was allowed by the fact that we had an incredible CEO that had the ability really to raise money and to make us feel comfortable in you can make something explode like Moderna has actually had the opportunity, particularly with the COVID-19, and then with all the other therapeutic area that we are envisioning right now. in terms of consanguinity, it's important that we collaborate because I know that you guys have a very specific rare disorder, and with Abdelali, we have already discussed the potential of taking some of the messenger RNA to the kingdom and work in collaboration with you guys in order to start the first trial, for example, for any of those diseases that are more prevalent in the kingdom. So our collaboration, I think, is important and vital for patients in the kingdom, and I'm looking very much forward to continue to work with you guys in order to create something that is going to have the ability also to explore within the kingdom, so to speak, in terms of having great therapeutics accessible to every patient.
Wonderful. Thank you so much. I know that we are probably with the chairman of the session. We are a little short of time, but we will continue our discussion even after this summit. Thank you very much.
Perfect.
Thank you very much.
Thank you, everyone. Thank you for having me.
Everyone.
Thank you.
Globally to the summit. You can join the session on the platform. We'll be back in 5 minutes. Stay connected. Welcome back, everyone. Now we will be looking at something different, ladies and gentlemen, how the application of medical technology improve people's health. Welcome back, everyone. Diagnosing, now we will be monitoring and at something different. To start this session, it's my pleasure to welcome the application of medical technology, President of the people's health by preventing, Turkey, diagnosing, and co-chairman, Mr. Raymond Berglund. To start this session, it's my pleasure to welcome and head of chairman of the and life sciences at President of the Nahir Corporation. It's a great pleasure to have both of you with us. And co-chairman, Mr. Biotechnology Summit 2021, Managing Director and Head of Health and Life Sciences at
for this great introduction.
It's a great pleasure to have both of you with us at the Riyadh Global
Welcome
Medical
to the
Biotechnology
number
Summit
7
2021.
of the KMARC Biotechnology Global Summit, which will focus on the medtech and diagnostics for this great introduction. As already introduced, my name is Bushra bin Saoud. I am the regional president for Danaher Corporation in the KMARC Biotechnology Global Summit. I will be co-chairing this session with medtech and diagnostics industry, who will be our panel as already introduced. My name is Bushra bin Saoud. I am the regional president. And thanks to our operation in speakers, we will try as much as possible to shed some light on the session with Mr. Raymond Berglund, trends, and then with our panel the Saudi market. During the pandemic, we have realized, and thanks to our esteemed speakers, important the diagnostics and the medtech shed some light on the and how they could impact us personally, professionally, and then we will zoom into the Saudi market. So during the pandemic, we have realized more than ever how important the diagnostics and the medtech industry were, and how they could impact us personally, professionally, in so many ways. So in today's session, we will have the chance to look at the topic. We will have Mr. Perry Frederick, we will have Mr. Eli Shayo from GP Healthcare, we will have Mr. Ayan Shahrani from Ministry of National Guard Affairs, and Mr. Ray. We will have Mr. Perry Frederick and Mr. Eli Shayo from GP Healthcare, Dr. Ahmed Al-Zahrani from Ministry of National Guard Health Affairs, and Mr. Raymond Berglund will also cover for us a session. Welcome to our speakers, and without further ado, I'll call on stage my two colleagues, Mr. Ayan and Mr. Sherif, that I already introduced. So a quick background on Cepheid. This is a global leader in molecular diagnostics, and Cepheid has been in the forefront of the fight against COVID-19. Another quick fact around the two leaders that will be speaking. Ayan has been in the Cepheid company for more than a decade. He has been mostly working in commercial and marketing leadership positions in Europe, in nations, in the Middle East and North Africa region, Turkey and Central Asia. So Ayan and Sherif will walk us through what is the role that the molecular diagnostics had in the fight against the COVID-19 and beyond. Welcome, and the floor is yours.
Perfect. Thank you so much, Bushra. Good morning, good afternoon, good evening, everybody. Thank you so much for having us. My name's Ian Sharp-Paul. As Bushra said, it's a pleasure to be with you here today. Can we just pull up the slides? I'm not able to see the slides.
You are on the first slide. You can get started.
Oh, okay. All right. Thank you. I can't actually see them. Just bear with me 1 second. I can't see them. Oh, there they are. Sorry, bear with me. Sorry, I was on the wrong screen. Thank you so much, Bushra. Okay, so first of all, thank you so much again. Thanks for having me. It's a pleasure. As Bushra said, we're going to talk today about the role molecular diagnostics had in the fight against COVID and what are some of the possible future applications we can look at for molecular diagnostics now that we've drastically increased our capacity and utilisation of PCR during the pandemic. So just to get us started a little bit, just a quick overview of Cepheid and who we are. So as Bushra said, we are a molecular diagnostics company. We have the Gene Expert range of systems, and on that we run a cartridge-based PCR test, range of tests for which we have more than 30 tests available which are CE marked. We have distribution in more than 100 countries. In fact, we have systems in around 180 countries, so pretty much every country in the world. We have more than 5,000 employees, and to date, we have over 30,000 gene expert systems placed all over the globe. One of our big focuses, to go just a little bit deeper into our technology. So our technology is really centered on three main things: speed, accuracy, and ease of use. We also have incredible scalability. So our tests or our systems are designed to run from a single test up to 80 at a time, and can scale up to being able to run more than 2,000 in a single day. What this allows us to do is place systems either in the hospital laboratory, as you would expect, but also near patient bedsides, in intensive care units, at the emergency department, but also in non-healthcare settings, like community testing, health outposts, and mobile testing units, which is particularly important when you think about the current pandemic and the need to get testing closer to patients and make it more accessible. But then let's look at molecular diagnostics more broadly and the fight against coronavirus. So molecular diagnostics have really come to the fore during the pandemic. So those patients. We've seen a record number of tests, in fact, more than 100 tests developed by multiple manufacturers all over the world, and in record time. In just the space of a couple of months, we saw dozens of tests come to the market to start to try to help in the diagnosis, the identification of SARS-CoV-2. And thanks to strong investment by hospitals and others, we've seen a big increase, a huge increase, in fact, in the capacity available of PCR tests and PCR platforms. We've also, during the pandemic, seen a lot of movement in terms of innovation. So one of the things that's been recognised very early on is the need to get results back as quickly as possible, both for the clinical outcome, but also to know so that patients are aware of their status and they can act accordingly. So we've seen a big drive towards reducing the turnaround time, and then also a drive towards moving testing closer to the patients. So that decentralisation has become a big theme that we've all probably come very familiar with. We now see tests and platforms placed not just in the laboratories, but also in airports, in drive-through clinics. We see them at sporting events, workplaces, and so on. So the PCR or molecular technology has now spread well beyond the traditional settings and has been able to be really ubiquitous across the market and across the geographies as we fight against the current pandemic. But when we look a little bit further ahead, there are some other opportunities where we can leverage this increased capacity and increased awareness around PCR. So just one example of that we'll talk about a little bit today is around antimicrobial resistance. So here's a quote from a recent paper from Bengochea and Bamford from last year, where they talk about the fact that the SARS-CoV-2 pandemic is not the only pandemic we're dealing with. That pandemic is overlaid against or over the top of an ongoing pandemic around multi-drug resistant bacteria. As we've seen SARS-CoV-2 being transmitted between patients within hospitals, we'll also see multi-drug resistant bacteria being transmitted, which has a drastic increase in mortality. And because of the limited arsenal of antibiotics left available to fight against these bacteria, it drastically increases the risk to patients and increases the risk of mortality and morbidity. So one particular area of AMR that is of grave concern are carbapenemase producing Enterobacterales. So these are incredibly prevalent, unfortunately, and really represent one of the greatest threats to public health ongoing beyond this pandemic. If we look to the Middle East region, unfortunately, it's not immune from this. We here in the Middle East see endemic CPE strains across. We see OXA48, NDM, and KPC. And because of the interconnectivity of the world today, these strains and these pathogens are very free to move between regions. And we've seen the fast spread of AMR, drug-resistant bacteria, and other pathogens moving between regions and causing significant issues. When we drill down one step further into Saudi Arabia, as an example, we're seeing an increase in prevalence of CPE or CRE. This is leading to outbreaks in some hospitals, and it's a growing concern within the Saudi Arabian healthcare system. These, as I said before, represent significant risk factors, and it's something that we really need to get on top of. And that's where molecular diagnostics come in. So molecular diagnostics provide the opportunity to identify carriers of these diseases or of these pathogens as quickly as possible, so that not only do you know how to treat that patient, but you also know what to do with them in the meantime. Putting them in isolation is costly and often not practical in some settings. So getting a rapid result and identifying both what the pathogen is and what the resistance mechanism is are critical. So you know how to treat them and how to place them within the appropriately within the medical healthcare system. So as I said, we've seen significant growth in the capacity of PCR. We've also seen a lot of increased awareness and availability of molecular diagnostics. We've seen it being distributed to many new settings. And on top of that, we're seeing a number of areas of innovation. We already talked before about the drive to reduce the turnaround time. The faster we can get the results, the better the clinical outcome, the more we know how to deal with that patient, and we can place them appropriately. We've also seen drastic improvements in the simplicity of this technology. That's allowed us to decentralize into those non-healthcare settings, and also within healthcare settings, pushing the test in, not just from the core lab, not just in the core lab, but push it to bedside, to push it in ICU, emergency department, and other areas. One of the other areas that continues to be developed is the divergence, more the broadening of menu offering. We see very targeted, high-quality tests looking at single pathogens like coronavirus, for example, or SARS-CoV-2. But then we also see a lot of new increase in panels, multiplex panels, which allow us to look at multiple pathogens at the same time. We can also call out particular strains or variants of interest. And we can also identify specific drug resistance mechanisms so that we can direct the clinicians and the pharmacists to the right treatment to deal with that patient. So if you think about the impact, it really boils down, most importantly, to better clinical outcomes. An accurate diagnosis earlier on has a better impact or a better outcome for those patients, and also allows us to manage those patients more appropriately in terms of isolation and where they go within the hospital. We're also drastically seeing an increase in access to care. Any patient today, particularly for coronavirus, can really go to a number of settings, not just their hospital, in order to get diagnosed and treated, ultimately treated for coronavirus. Not only does this improve the convenience for the patient, but it also improves the outcomes when a clinician is able to access those results quickly and can act upon them. And because of the broadening, test availability or test menu, clinicians are able to determine the right test for the right patient at the right time. So with that, I'm actually going to hand over to my colleague Sharif, who's going to talk a little bit more about Cepheid and the role we're playing in Saudi Arabia, and then talk about some of the specific challenges we face in the diagnostic world within Saudi Arabia.
Thank you, Ian. Good afternoon, good evening, good morning, everyone. My name is Sharif Haridi. I'm leading Cepheid business in this geography since a decade. Cepheid started with small presence in the Middle East region back in 2011 with limited activities and limited business. Now in 2021, Cepheid is partnered and well-positioned to continue driving several healthcare-associated initiatives, disease prevention programs, and screening policies. We are available in 18 countries in the region with 24 team members. We have more than 1,100 gene expert systems deployed and running, while the biggest presence is in Saudi Arabia with more than 350 systems. PCR used to be known as a complicated, in fact, still known as a complicated testing, requires big auto-analyzers, which are batch-driven, occupying big space, and dedicated space only for the PCR, demanding daily maintenance with bulk storage of short-life reagents. Cepheid technology is the game changer of the PCR testing. We are using simple, durable, easy to use, and easy to maintain gene expert system, along with the heart of our technology, which is the test cartridge. The magical test cartridge, we call. It's the biggest and yet expanding menu in molecular testing, with more than 30 CE-IVD molecular diagnostics tests available. The flexibility and modularity of the Gene Expert system makes it the best in class and the first choice of any size lab in any size hospital and any point of care testing site, as Ian mentioned. The testing power of the Gene Expert system varies from few tests per shift of 8 hours in any small setup to few thousands per shift in any big hospital or public health laboratory. The same system, the same cartridge, the same technology, and most importantly, the same quality of result wherever you conduct the test. We differentiated ourselves early enough, driving towards prevention of several diseases and healthcare associated infections. While others were busy running behind diagnosing, we started to have the initiatives into the regional screening policies and disease prevention programs, which improved the clinical value and consequently the impact on the patients in need. Important to mention that 45% of our business and activities are coming from Saudi Arabia, while the remaining 55% are coming from the rest of the Middle East countries. Next slide, please. We in Cepheid, as well as many others in the diagnostics industry in Saudi, were faced with several challenges, which once resolved or minimized, will enable the diagnostics to play bigger and more important role to improve the outcome and the clinical value of the diagnostics result and to minimize the impact of these challenges to a bigger extent. With Saudi extended geography improving patient outreach and be available close to the patients while providing the same quality of care and outcome will remain a challenge and the strategic objective, while improving the turnaround time through eliminating the need to batch and collect certain number of patient samples to perform the testing. As the patients do not come in batches, we need to serve them as they come. Lack of enough number of highly qualified and experienced technologists demand and dictate simple while accurate and fast technology, which will minimize the need of highly skilled and highly qualified operators. Made it easy to adopt and operate anywhere and at any level of infrastructure and manpower. Adopt and activate, practice, enforce policies which is already in place to drive and promote prevention versus diagnosing of healthcare associated infections, multi-drug resistant organisms, to reduce hospital days stay and infection rates anywhere in the healthcare setup. Disseminated and segregated budget into different function speciality within the healthcare organization versus holistic look at the entire budget and spending. This will drive the awareness towards the total cost of the service versus individualized price assessment, which will always lead to silo actions and silo decision making. Integration of information, communication, digital and remote technologies need serious consideration in order to increase and accelerate not only the adoption of the implementation of the advanced technologies, but also the overall competencies and effectiveness of the country at the healthcare system. Back to you, Ian.
Thank you, Sharif. So just to summarise, so we talked already a lot about molecular diagnostics and the role they played, and they'll continue to play an important role as we continue the fight against coronavirus. We've seen how the deployment of this technology from traditional settings to non-traditional settings and getting closer and closer to patients has highlighted both their value and utility. But as we look forward, both through the end of this pandemic, but also with the risk of other pandemics, we see there are a lot of other opportunities to leverage molecular diagnostics, and not just in the area of infectious disease. We have significant challenges with aging populations around oncology, non-communicable diseases and other areas, and these are more opportunities for us to leverage the benefits and utility and efficiency and accuracy of molecular diagnostics to improve patient outcomes. So with that, I thank you very much for your time, and hand back to Bushra. Thank you.
Thank you, team. Thank you for the very interesting insight, and I hope that it's more about the opportunity that the molecular device brings specifically in Saudi, but independently from the COVID-19 tailwinds. We know that brought obviously a huge spike for us, and we somehow see that every day. But if we decouple the COVID-19 impact versus our, let's say, I would call it base business, how do you see the opportunity in Saudi? And I would like Sharif to give us his views. Sharif, you are on mute. Okay.
Yeah. While we were supporting the activities around COVID, we were never lose the focus towards the other respiratory and also multi-drug resistant infection, which is very important. And going forward after COVID, this will remain. And with the lack of antimicrobials coming in the pipeline of many pharmaceutical pharmacies, I think we have very less to handle in terms of antibiotics. So we were driving the focus towards continue screening and implementing prevention approach towards the antimicrobial resistant or multi-drug resistant organism, mainly the CARBA-R and MRSA, carbapenemase producers, and the methicillin-resistant Staphylococcus aureus. And while everybody was focusing on the COVID, we were again driving the screening and the preventive approach towards those centennial microorganisms.
Thank you, Sharif. And with that, we will move to our next speaker. So I'm going to invite on the floor Mr. Perry Frederick. Perry is a Senior Director, Strategic Research Europe, Middle East and Africa at GE Healthcare. Perry is leading a team of clinical research scientists supporting strategic studies across the EMEA region. Perry and his team play an essential role in delivering evidence for GE Healthcare strategic research needs. Perry will, in this session, will walk us through the oncology care pathway solutions from GE Healthcare. With that, Perry, the floor is yours.
Thank you, Bushra. Can you hear me okay?
We do. Hear you very well.
Okay, perfect. Thank you. Okay, thank you for that introduction. I welcome everyone to this session. I might ask for help in advancing the slides. I'm having a little trouble with my cell phone. So if we could go to the next slide, please. In terms of an agenda, I'll be describing today the global cancer incidence and current challenges that we face in this area. And I'd like to cover how we can beat cancer with a focus on precision care as applied to the patient pathway. And then finally, I'll share care pathway evolution that we see in 3 prevalent cancer types, namely lung, prostate, and breast cancer. Next slide, please. So in terms of incidence, global cancer cases totaled over 18 million back in 2018, with nearly half of those cases coming from Asia, including the Middle East. Moreover, global cases are expected to increase nearly 70% over the period of 2012 to 2030. With this burden, Key reported challenges are early and accurate detection, access to precise diagnosis and treatment, cancer complexities and treatment personalization, as well as managing reoccurrence and risk. Next slide, please. Thank you. As shown in this graph, adapted from a recent Nature article, I'll ask you to focus initially at the left, today's care methods are resulting in high tumor burden at diagnosis, which then unfortunately continues to increase due to therapy resistance, and this negatively impacts quality of life and survival. To improve therapy response and outcomes, we identify 5 potential solutions. These are earlier detection and prevention, including asymptomatic screening, precise diagnosis, targeted treatment, therapy surveillance and adaptation, and finally, quality of life monitoring. And as shown at the far right, applications of these solutions can hopefully lead to improved outcomes. Next slide, please. So these solutions I just mentioned form the basis of what we call precision cancer care as it applies to the patient experience or pathway. Thus, from left to right, screening is improved by selecting the right therapy. Diagnosis is improved by integrating the right data. Treatment is improved by guiding therapy to the disease and intervening at the right place and the right time. And follow-up is improved by monitoring the right signals or signs. By bringing the appropriate technologies and solutions to bear upon the problem we face, we could support our clinicians and deliver better outcomes for their patients. So let's apply this to our key cancer examples I mentioned earlier. Next slide, please. Please back up one. I'm sorry, I didn't see that you had advanced to the, okay, thank you. Sorry for that. Okay, so we begin with lung cancer and consider the rationale for reassessing current care models. So first, lung cancer is the #1 cause of cancer death globally. Second, the survival rate is less than 20%, and this is at stage 4, with more than half of all patients being diagnosed at this late stage. So clearly too late in the game. Third, 40% of patients will wait more than 2 months to receive their diagnosis. This is clearly wasting precious follow-up time. Next slide. So here we've depicted the current care path across the top. And what I'd like to focus on is emerging and future technologies which appear at the center of the slide and below. So emerging innovations applied to this pathway begin with targeted screening, shown to reduce mortality by nearly 25% in males, in concert with AI reporting and decision support. In the diagnosis phase, rapid staging on the order of 10 days will be enabled with precision biopsy, using ultrasound guidance to replace CT or fluoroscopy methods. Treatment will be enhanced with greater precision in CT and PET-based radiation therapies. Moving to the future, screening and diagnostic events will be enhanced by 2 levels of data integration. Patient-level integration, something we call OncoFlow, will combine imaging, EMR, genomics, and pathology data in a single view for caregivers. System-level integration, something we call Cancer Cockpit, will provide capacity, utilization, and patient flow visualization. Treatment planning will be enhanced by prediction response to immunotherapies. And finally, remote patient monitoring technologies will provide earlier warning of deterioration and acute events. Next slide. Considering now prostate cancer in terms of occurrence growth, we find that by 2030, it is predicted to be the most common cancer type in the UK. Also, inaccuracy of PSA screening is translating up to 75% false positives and 15% cancers missed. In addition, diagnostic inaccuracies mean nearly 50% of cancers are mischaracterized as slow growing, leading to errors in treatment planning. Finally, 60% of men are waiting more than 2 weeks for this error-prone diagnosis. So clearly we can do better. Next slide. Once again, emerging developments yielding a more precise screening and diagnosis include faster and more comfortable multi-parametric MR imaging in the first instance, as well as use of gallium PSMA in conjunction with PET-CT and PET-MR imaging devices. Likewise, biopsy accuracy will improve with MR and ultrasound image fusion. Application of these technologies should result in a rapid assessment pathway on the order of 1 week. Similarly, treatment planning and execution will be enhanced with custom brachytherapy using CT on rails, as well as MR-only radiotherapy planning, embolization, as well as increased adoption of theranostics. Finally, monitoring will improve with gallium PSMA and PET-CT in combination with other low-risk surveillance methods in the community. Next slide, please. Oh, sorry, if you could go back one. There's one more thing I wanted to point out. As was the case with the lung example, we also see the ability to introduce patient and system level data integration platforms once again to drive precision and efficiency during the screening and diagnosis steps. In addition, advanced dosimetry and home observation developments will help improve treatment and monitoring phases. Next slide, please. Our final example, breast cancer, starts with a focus on patient experience. As you see here, imagine an individual faced with 300 appointments utilizing up to 9 different technologies in different locations, translating to up to 3,000 kilometers of required travel in some cases. Additional impacts include the pain and anxiety of delayed results, in addition to managing multiple caregiver interfaces. Next slide. An emerging development in this case, which will positively impact screening, involves invasive cancer detection for dense breast using automated breast ultrasound imaging. During the treatment phase, increased adoption of contrast-enhanced spectral mammography, which is an add-on to our mammography products, as a first-line diagnostics for biopsy and surgical planning is envisioned, as well as ultrasound-guided biopsy using fused MR images. Also, reduction in ultrasound-based reporting variability will be enabled with AI tools from partners like Kios and others. Next, radiotherapy treatment planning will be enhanced by linking key imaging modalities with partners like Elekta. And finally, to improve upon the patient experience, we will operationalize same-day feedback and assurance models for rapid results, resulting in reducing related anxieties. Next slide. So this concludes my presentation. In closing, I want to thank you all for your attention today and for all you do to improve lives around the world in moments that matter within the healthcare context. Thank you.
Thank you very much, Perry. And I would have somehow a very quick question for you, which is more around the fact that, you know, listening to you and to everything you presented, there is clearly a fascination around all the technology and the advancement of the technology towards the fight against cancer. So if we were from that somehow ecosystem to land in Saudi, what would be the application that we can make out of it into the very 6 months, 8 months down the road? Meaning, what can we take out of everything you presented and say to the institutions and the academia, those are the top 2 things you cannot miss in the coming 8 months or 12 months?
Okay. Thank you for that question. You know, when speed counts, when speed is what matters, I have a 3-word response: best practice sharing, okay? And one of the principal functions that we serve inside of GE and even among the research group is to connect our key collaborators around the world to share what's working, what's effective, and bring it into the site of interest for quick adoption and quick impact. That is far and away the best advice I can give, because we can talk about a lot of ideas, but it's when we put them in practice that it matters. And when we can connect you with our global collaborators, we will find those solutions.
That's a good one. Thank you very much. Thanks a lot. And again, let's move to our third speaker. This time it's Dr. Ahmed Zahrani, Director of Healthcare Technology Department in Ministry of National Guard and Health Affairs. So Dr. Ahmed works closely with a team of healthcare providers to address the workflow challenges towards digitizing and automating to support the ventilator manufacturing initiatives by the local SMEs in the kingdom. Thank you, Doctor.
Thank you so much for introducing me. Assalamu alaikum wa rahmatullahi wa barakatuh. Let's start the presentation. I would start the presentation for this quote, which say, from Ibn Khaldun, which say, tough time make strong people and strong people create good time. So I'm sure that we already been through a tough time last 2 years. And I hope, inshallah, we are the strong people who are going to really create a better environment for the humanity based on what we have learned from that tough time. Actually, what we're going to talk about today is how, I would share it as, I would call it as, let's say, collaborative successful story between the government and how they respond to such a pandemic that happened last year. And I would try to make it in a storytelling way, so just to get out of the theme of the scientific presentation. So let's go back in the first case, which is already reported in Saudi Arabia, which was in 2nd of March. So that was the first case reported by COVID-19, and that was the start of triggering the pandemic here in Saudi Arabia. If we go back to that date, we can look at there was a lot of issues happening globally, and you can see the situation globally happening. There was a crisis in hospitals, admission, a shortage in some kind of devices, and also a lot of medical supplier. You can see also there was a lot of economic issues as well happening globally that already gonna result as a panicking for the people. Also, there was a lot of issues for example, the mask, the shortage, and the demand increased significantly when you compare it to the supply. So that also impact the economic globally. And finally, and this is what we're gonna focus about, which is the medical devices and the high demand of the ventilator due to a lot of patients admitted in hospitals, and the shortage of the supplier happening globally, not only in Saudi Arabia. So all of these challenges being exposed that time, and it is really taking, just to think about it from mental health perspective, from the care provider, when they thought they are going to face such a disaster for the coming weeks or coming months. So there is no clear expectation what's going to happen. So this is not a risk. This is really uncertainty. So with that said, if we're going to look at the curve of pandemic, and this is from the Ministry of Health, you can see starting from March, most of the reported cases was between April, starting from April as per the paragraph, and you can see it starts going down August or last of July. Due that time, I mean, we are still in 2nd of March, and that was the discussion happening in 2nd of March before we see this graph. So we are not aware that how it's gonna happen and how can we respond on the shortage of medical devices, and especially ventilator, in the Saudi market, and how can we overcome this issue. So the collaborative happening that time between all of the government sector it was planned to find a solution within 4 months. There was no plan that we were not expecting the pandemic gonna finish or the peak gonna go down within only 4 months. But we discussed that how can we agree on a plan within the next 4 months from April until the end of July, just to make sure that our healthcare system is secure and we have enough medical equipment, especially from ventilator perspective, in the Saudi market. And the main challenge at that time, I remember, there was a lot of countries, they lock already the export. So there was unexpected issue that even if you have some kind of ventilator, simple things like spare parts, like small things, or maybe consumables, we will not be able to import it. And that already adding a lot of, let's say, load on that challenge. I mean, during that time, alhamdulillah, from the leadership from the kingdom, there was full support, unlimited support to strengthen the healthcare. So that was reflected or translated through a lot of committees formulated during that time. And one of them was a committee, our national committee formulated to support the shortage of ventilator and find a way how can we really be able to prevent or detect or mitigate and response to that challenge which is gonna come through the pandemic. So I'm listing here, I mean, the authorities or the government sector which already collaborated during that time, and Ministry of National Guard was one of the important pillar of that committees. And I'm gonna talk about now what was the role of Ministry of National Guard, because if you look at it, it will be obvious that all of these entities, they have a really clear scope, but National Guard here is considered as a service provider from this. But the role was, I mean, the mandated role for National Guard is to support by expediting the local development process for the local manufacturers. How can we support the local manufacturer to expedite, to develop their product if they have product? At that time, we don't know if there is any product in the local market. And how can we do a kind of process to evaluate it and see, is it visible that it can be used or not in crisis situation? For sure, we're not gonna use any device which is not been go through the trial and all of the clinical trial and all of the regulations, but that was exercise to see what can we do in the tough time that we are expecting. So if you look at it, we are not gonna go through the normal life cycle of the product development, which is here according to the to the FDA. So this is the normal development for any kind of product. But during that time, also there was emergency use authorization for some medical equipment. And that was maybe the opening door for us to think about if we're gonna utilize some of these equipment in case if we are shortage, how can we gonna use it, when we're gonna use it, all of the processes, how can we put it all together in a systemic way to prevent kind of crisis that's gonna happen, and also to make sure that we are not compromising the patient care. So before I go to the story, so this is just only I would like to highlight why Ministry of National Guard being selected to this initiative or to participate and contribute with all of other facilities that we having across all of the kingdom, more than 6 hospitals and more than 40,000 employees. All of them, they have, I mean, great experience that they could contribute in this kind of crisis. Second, National Guard known as a pioneer in technology advancement adoption. And there is a lot of achievement being highlighted last 5 years. Also, the organizational capacity that we have, I mean, National Guard, we have a hospital, we have university, we have also a research arm like King Abdullah Medical Research Center. So all of these let's say, giving the National Guard advantage that they could really contribute with all of the other government sector to mitigate the risk that we're having. And this is an example of the things that, let's say, the multidisciplinary team heavily involved with the respiratory therapy and biomedical engineering, quality and patient safety in intensive care unit, Some of the pioneer technologies, advancement in adoption in National Guard, like the electronic medical record and integrating devices with systems, also the patient applications and innovative and integrative workflows. So we are not talking about only technologies, even the innovation of the processes and workflow within National Guard. And finally, example of the organizational capacity that in National Guard, there is a biotechnology park, simulation and testing lab, a lot of advanced test equipment, and most importantly, the innovation culture for the staff in National Guard. They have this kind of mindset, which is not considering them as producer. They also consider themselves as, not consumer, sorry, they consider themselves as producer as well. So how we respond to that situation from the Ministry of National Guard? First, internal arrangement. So we have arranged with all multidisciplinary teams. I remember there was more than 7 departments and plus more than 35 participants or members. They participated to these 2 technical or these 2 committees. So we created 2 committees, 1 technical committees and 1 medical committees. Technical committees gonna go through all of the local manufacturing initiatives and evaluating them from technical perspective. If they will pass the technical perspective, then they will go to the medical committee just only to assess and see how its visibility to utilize this product in crisis situation. Also from internal arrangement as well, we put like a path for any kind of device or any kind of local manufacturer's material or device. When it's received by National Guard or when received by the Ministry of National Guard until it's delivered the report to the National Committee with a recommendation. So you can see the stages for any local product that will be developed. First, they will have to fill the product information and we're gonna receive it. We're gonna do first inspection, and if this will be successful, the stage 3, then the product gonna go to the 3rd stage, which is the technical and functional test. It will be heavily tested through a lot of clinical scenarios. And once this will be passed, then it will go to the technical committee to evaluate if this product gonna be utilized or partially utilized somehow in the crisis situation. And then it will be approved or disapproved. Then it will go back to the National Committee to give the recommendation. So we are not talking about now certifying a product, just to make it clear. We are talking about a situation that it could be a crisis, it could be shortage. How can we deal with it? We have to have a plan B, and this is all what we are doing here to see how can we mitigate the issues. From external arrangement, we put this kind of workflow with all of the members. This is we end up with, that since we receive the device from the National Center for Industrial Development, they are the gate that referring all of the local manufacturers to Ministry of National Guard, and it will go through all of the process, go through the committees, and you can see here also that if the device have technical issues, we are communicating also with the vendor to fix it and coaching them, telling them what to do, and then return back the device and see how can we improve it before we do the functional test. These are some of the local manufacturing initiatives, which is already been evaluated and supported by National Guard. These 5 companies, and it was only evaluated through from May and June, just only 2 months. So these companies, the product they propose, it's categorized in 3 different categories. So first product or 3 companies, they propose emergency resuscitator, and they propose it to Advanced Electronic Company. They propose critical care ventilator, and also just how can we utilize it in the crisis, or if the crisis happen. And the last one is the CPAP ventilator. So this is the 3 categories that have been proposed by the local vendors. Just to not, I'm sure there's just only 5 minutes remaining. So these the kind of technical and functional tests. And if you remember when I said that one of the capacity that National Guard have is the cultural innovation. And just to end up with kind of technical and functional tests for a service provider who doesn't really have any experience before, to put kind of testing, that was one of the things or remarkable things that happened that time, because we work together as care provider with some researchers from KMARC, with some people also from the university, all together to put a systemic way, how can we test, or let's say heavily test this kind of devices, even if it is not certified, but we want to make sure that how can really we be in the, ready for any kind of crisis. So it's summarized in five kind of tests as the operation challenges. So there are more than 30 operation challenges being proposed by the biomedical engineering and RT with the technical challenges. Also alarm trigger accuracy, so that was also tested, and stability test, and also stress test that putting the device under some kind of stress situation. As a result, one of the outcome of that committee is it's end up with a performance report or technical and functional test for one of the devices, which is pass the preliminary test. And that report included all of these kind 5 tests and being sent to the national committee to look at it and give the recommendation, how can we move forward. So thankfully that time we didn't really need this kind of initiative, but that is really a proof for us in a local or in Saudi Arabia that how we resilience as a healthcare system and how can we respond to such uncertain risk that we haven't really expected gonna happen. But the lesson learned from that is there is a lot of ongoing collaboration now happening between all of these partners, just only to facilitate and to expedite the ecosystem, or how to make the Saudi market as really a hub for all of these kind of biotechnology or biomedical devices manufacturing, and how can we expedite all of the effort and facilitate all of the local company. That was one of the most important lessons learned, and one of the most important even outcome from that exercise and journey happened in last year. Thank you so much. I'm finishing here.
Thank you, Dr. Zahrani. That was really insightful, and thanks for the storytelling approach, which definitely I'm sure our audience will appreciate. For the sake of keeping us on time, I will keep the question into the panel, and we will move to our next guest speaker. It's my pleasure to introduce Mr. Eli Chaillot, Vice President and CEO Services GE Healthcare EMEA. Before this position, Eli has been leading numerous leadership positions in GE in several regions, such as Europe, Japan, and Middle East. With that, Eli will be sharing with us his views on the data and the future of healthcare in the COVID-19 era. Eli, welcome, and the floor is yours.
Thank you. Thank you for the invite. I'm pleased to be here today. Can you hear me okay? All right, so we will be looking at data and the ecosystem of healthcare after COVID-19. We all know that COVID-19 has been an unprecedented event in modern times. And so many of the changes that were that happened after the COVID era have been unprecedented. And the hard-earned wisdom of 2019 and 20 and early 21 has led to have a hope that the ecosystem of healthcare is going to change in a much more resilient and much more connected, in a much more virtual way, so we can secure better precision healthcare in the future and making sure that we have the best outcome across the healthcare ecosystem. To succeed in the future, healthcare leaders will have to look into how do we make sure this is scalable, how do we make sure this is continuous and resilient, so we make sure that we make sense of the data that the medicine and clinical science is providing throughout the patient story. So today I'd like to share with you 3 topics basically around the data and how we are living this post-pandemic or through pandemic era of healthcare throughout the crisis. The first one is digital, the growth of digital across many industries, and healthcare is one of them, and most precisely the virtualization of care. So if you take telemedicine in the past, telemedicine has been underused. Consumers used to ask for it just for convenience, but there was no real sustainable, I would say, scalable offer on the market. And then as we move forward with the COVID-19 crisis, you've seen a lot of virtual visits happening, for instance, and that was happening just to keep our family safe, to making sure that we don't expose them to more, I would say, virus than what they would have. And so that went and exploded over the last few months. And by the way, in Saudi Arabia, like other countries, it was one of the things that we have observed over the last few months. And then healthcare institutions from their side, also, we have seen more and more of their staff and caregivers working digitally and virtually across the patient journey. You could see like radiologists reading more images remotely from home. You would see more clinicians and more doctors giving their service remotely. And so that exploded over the last few months more and more. And what this happened basically through this virtual care is we have seen more available resources, scarce resources, connected to patients that you did before. And so by doing this, you are using data and you are basically looking at how do you make sure that this data is not misused. And so all these hospitals had to keep the storage of the data in a way that you don't expose the patient in the future. So the efficiency of that data would happen more the more you store it well and the more you connect it well. And what happened with the digital systems is that these hospitals would have to store this data in a way that they connect it together and make much more sense of it and much more insights for clinical decision making in the future. So with this, we have seen the emergence of much more tele-ICU, for instance. Like we have established in India an 800-bed tele-ICU to monitor the most ill patients and enable us to, from one centralized place, monitor these patients. If you look at Saudi Arabia today, way beyond and before the pandemic, Dr. Sulaiman Habib Hospital Group have established 796 tele-ICU beds in the kingdom to monitor the most ill 24/7. And so looking into this, you will be able to free up some beds from hospitals and making sure that patients stay home and being monitored and being secured and safely watched by caregivers. And we know doctors would tell you that if we keep the patient at home in his or her social surrounding, the recovery will be much faster. And so we've seen providers and caregivers exploring the way they treat patients, and we'd see hospitals taking more and more the most ill treated in the hospital, free up beds for really very sick patients, and making sure that home becomes basically the virtual expansion of hospitals at home. So you'll see this happening more and more as we move forward. And so as we have this data, you need to make sure how do we connect it and make sense of it. If you take like Oregon Health and Science University in the United States, they have established what we call a population surveillance between the caregivers, bedside caregivers and virtual caregivers in order to create best practice sharing and give a wing-to-wing of the patient status across a selected care area or a hospital for the entire health system of that district. And so to conclude on the first topic, the pandemic has accelerated trends that started before the pandemic, which is virtual health, which is virtual care. And I think we need to leverage that momentum to continue building on that, making sure that we free up resources in hospitals, extend the hospital to the home of the patient, and connect the data to make sense of it for better insights for clinical outcome. And the second point would be the convergence of clinical medicine and data science. So medicine produces a lot of data. We know that the body of data in healthcare is doubling every 73 days. And so the more data we have, the more signal we have, and the more noise we have also. And we know that 95% of that data is mainly not used today. So how do we make sense of this data? So the data science is a scientific process today to look into curating the data, into eliminating the noise, and making sure we extract that insight from the data. Today, this is being done by doctors in their heads. If you take one doctor career over 40 years, 6 days a week, they would have seen 5,000, 10,000 exams in their life. And they maybe use a decision support system that is not necessarily statewide or worldwide. But when we start taking all the data and all the insights globally for all these exams, put them together from thousands and thousands and millions of doctors, and you would start looking at value-based healthcare that's going to drive outcome and increase recovery and reduce times in hospitals. So that's basically what AI is driving. During the pandemic, we have launched what we call a thoracic care suite, and this is basically an AI suite of apps that we have installed into X-ray machines, and that was enabling us to look at the lungs and thoracic abnormalities, and in order to identify any pneumonia or any tuberculosis driven by COVID-19. That helped a lot to triage patients coming to hospitals during the crisis of COVID-19, basically. And so if you take like another example in the UK, so the National Consortium of Intelligent Medical Imaging, led by Oxford University, they are putting a wide range of sustainable ecosystem for academic, clinical, and industries to come together and create algorithm to find unmet needs through AI. All right, so you would see this, and all these, I would say, matters started before the COVID-19 pandemic, but were very, very slow. And then as the COVID-19 hit, these were accelerated more and more. So basically, we have a lot of data to conclude on this subject. We have a lot of data. We need to make sense of this data. We need to connect the data. We need to make sure that we have less silos within the data in order to connect the wing to wing and to serve better the patient and get better outcome. And in this context, we want to make sure that cybersecurity at the heart of that topic in order to safeguard confidentiality. And the last topic here I would like to cover, which is another lesson learned from the pandemic, is how do we look at capacity creation and how do we look at using efficiently the resources overall. So the pandemic has pushed us to ask ourselves a few questions like, how do I minimize delays and waiting time? How do I maximize the use of resources I have, whether it's human resources or material resources? How do I improve outcome for patients? For every COVID patient, questions were, how do I make sure that the time is short? Time is absolutely critical for a COVID patient. How do I make sure that the resources like X-ray machines, CT scans, ventilators, monitors, various scarce resources during the COVID-19 are basically used by the most ill people? The deaths was on the rise during the pandemic also. So hospitals had to take a helicopter view, a big picture to understand how to allocate resources to the most ill patients hit with COVID-19. So during that pandemic, you needed to have, if you are a group of hospitals, you needed to have a central view of how are things going? What can I prevent from happening? What do I need to divert? What do I need to accelerate? Do I have enough supplies in that space or that other hospital? Do I have the right flow of patients? Do I need to divert ambulances from hospital A to hospital B? And this is what we have done during COVID. We have launched an AI suite of apps that we call Tiles in order to look at the COVID-19 patients and make sure that we predict what will happen in the future. So if you look at, most doctors today look at clinical data as a historical data. So they look at the past, they try to make sense of it, they try to connect the dots and predict or at least diagnose what they have at hand. The command center that we have connected in 300 different institutions around the globe, and we have one already working in Saudi Arabia that we have dedicated with the help of the Ministry of Health to COVID-19 crisis management. We were able to look into which hospital has bed capacity, free. Where do we need supplies? Where do we need to reconvert or divert patients coming with COVID-19? And you were able to predict the future versus only looking at the past. And so this kind of centralizing decision and looking at like we do in NASA today, you have full screens looking at the whole healthcare systems and making sure that you are adapting the resource to the need. So all this data is basically aggregated in one place, and all we do is just creating algorithms that make us to make the right decision to help address needs across the healthcare system. And so during that pandemic, with the Ministry of Health, we were able to look into the 186 hospitals across the kingdom and looking at the help needed, the resources that are becoming rare, and address that, through the Ministry of Health. So the opportunity here is that we can create in healthcare a lot of waste. We can create a lot of data, and biggest waste for me would be that we don't allocate resources where are needed. And so through the pandemic and after the pandemic, we are in the learning mode still, I would say, to maximize the utilization of resources, to making sure that it's serving the purpose and making sure that we are increasing outcome and we are using in a most efficient way the dollars invested into a healthcare system. So in conclusion on this subject overall, the pandemic has accelerated a lot of the changes that started before COVID-19 era. The utilization of the hospital is most likely going to continue to decrease, and only the sickest of the sick would use it. And I would see much more people treated at home with home monitoring and virtual care increasing, which will increase also the recovery. Today, there's no question anymore whether AI is the right technique or technology. The question around AI is whether, how do we accelerate it? How do we use it? What do we do with it? AI is becoming an established truth to be used in healthcare and many other industries. If you look at the pharma companies, the way they worked on developing the antivirus in few months, things that used to take years and years in the past, they also shared data among themselves, among companies that would be their own competitors, just to find a quick solution and to deal with this disease. They had to scale up very quickly production for billions and billions of jabs around the globe and just distribute it. So this tells you that one company cannot solve it. One country cannot solve it. We will have to work together to make it happen. I'd like to pay a big thank you, basically, to all the caregivers and all our engineers of maintenance and all the healthcare ecosystem around the globe for being our heroes in the past 18 months. Historians over the next 15, 20 years, something to marvel at and talk about. And I think the learning has just begun. We will need to capitalize on this and keep moving in order to make our healthcare complicated system today around the globe much more efficient, much more accessible, and much more precise to drive outcome for patients. So that's what I wanted to share with you today.
I think still I would like to have a very quick question to you. Basically, this is obviously, in many industries we talk about AI, and this is relatively, I would say, exciting from many aspects. Now, when we kind of take that and then land in the kingdom, and then somehow become a bit like, what do we have on hand? I'm sure that we might face some challenges in the way either it is requiring high budgets to have whatever it takes to have to read the data, or it requires talents that we might have or we might not have, or there are other challenges like regulatory or something else. So it would be great if you could shed some light about, what are the challenges that we face as we speak now in the kingdom?
Yeah, that's a great question. Rather than speaking about challenges, I would speak about opportunities. When you look at Saudi Arabia today, there are two main opportunities that gives me hope that we're going in the right direction. First of all is the collaboration between the industry and the institutions. There's a huge collaboration there. We've been in the country for 80 years now, working with public and private institutions. We always looked at localizing capabilities. Right? In a country like Saudi Arabia, you have the scale, right? In the region here, Saudi Arabia has a scale that is not met anywhere else. And so how do you make sure that you localize the talent and making sure that you build expertise in the country to serve the patient of Saudi Arabia? And so that collaboration existed already. You can see today in Saudi a lot of what we call PPPs, public-private partnerships, and also MESs, managed equipment services, on the rise. Right, where the public sector and the private sector want to come together to deliver more patient outcome. So that's the first one. I would say that there is a will and there is a budget for it, and there is a really, I would say, a noble desire to improve the customer experience overall, or patient experience in this case. In the region, if not the first, the very first, was in Saudi Arabia. Command Center is going to help you get smarter at resource utilization. And we apply AI to every single tile of the command center in order to look at the past, learn from it, and predict the future. And then maybe tweak some, I would say, figures to make it much smarter and more efficient. And the second thing is, if you look at the adoption of many of the virtual appointments and visits in Saudi Arabia, across the 10 million. This data, by the way, is maybe a year old now. 10 million appointments booked online. So there is a big will by the Minister of Health, His Excellency, the Minister of Health himself, to drive digital care, to drive online bookings, to drive outcome-based healthcare. And so, to summarize, the great partnership between the industry and the institutions and the public-private partnerships is one opportunity that is already happening and we need to accelerate. The second one, the digitization of healthcare within the kingdom, driven by the Saudi Minister of Health and other public institutions, is very promising, and it's also in line with the 2030.
Raymond Berglund, Managing Director and Head of Healthcare and Life Science in Alvarez Marsal, EMEA. Raymond brings with him 2 decades of experience in transformation, turnaround, and financial leadership in medical device and healthcare organizations. Today, Raymond will share with us his views on what is the roadmap to build a medical device industry in KSA. So again, without no more transition, Raymond, the floor is yours.
Usha, thank you very much. And marhaba, assalamu alaikum to everybody in the room there and across the world. And I appreciate it. Thank you for the audience today. I'd like to say that I am a relatively simple guy. I come from the medical device industry. I was the CFO of the respiratory business at GE Healthcare. I also worked in orthopedics as well as wound care. So I've gone everywhere from capital equipment all the way to consumables and what I might call fancy Band-Aids. So what I was thinking about was, KSA and the Vision 2030 of taking about how to invest and actually how to pragmatically grow a medical device industry. So, oh great, the little clicker works. So I'm controlling my own slides. Like I said, I'm simple. When it comes to medical devices, you have to invent it, you have to make it, and you have to sell it in its most simple form. Obviously, there's plenty of regulations. There's plenty of work to be done for patient safety and how do you apply it? What are the reimbursements around it? There's a lot there. But when it comes to making a medical device industry, you need to invent, you need to make, and you need to sell. And when we break that down just a little further, what you have is discovery and development. So understanding and finding a new technology or new application, and then development would be a clinical study. It's going to happen. It's just, you're not going to have an export immediately. You've got to start somewhere. And so I look at it and I say, well, what's the early stages of a medical device or medical technology industry? In that areas, those groups must be rewarded. They must be funded to drive partnerships with private companies, with external companies beyond the borders, and also complete studies, clinical studies. You need to be able to have simple manufacturing in country that is competitive with other contract manufacturers around the world. If you look at Eastern Europe, for instance, it's a great example of where they might not be making all of the largest machines and all the largest capital equipment, but they are making lots of parts and pieces for medical devices. And they have quality levels and inspection levels that reach to that. And that's early stage work. And then, of course, on the selling side, consumables distribution. You can think about some of the most developing countries where just getting out first aid type of advanced wound dressings, simple consumables and replenishing those. That's probably the simplest, earliest stage of medical technology. And then when you think about what's in that growth stage, when do we start seeing this increase? Well, early on, what you're going to have is private investment. Investors that say, hey, I will fund your research and your way to gain intellectual property. And then we will use a contract research organization who does a lot of studies because they're able to kind of take the cost of multiple studies and combine that. So you start to see those organizations be created within the country and doing services outside the country. You also see CDMOs, rather than contract manufacturers, contract development and manufacturing, and satellite sites. We've got Danaher on here, we've got GE on here, we've got a lot of different companies. Obviously, if they have a satellite site in KSA, that means they have trust and belief in the labor force and the work to actually produce product. And then you've got basic medical device sales, things like ventilation, and then the in-servicing and the service depot, completing it in country, making sure the product gets put into place and serviced in country. And then what is that mature stage? We got big corporate investment. Corporations will fund research. I mean, think about the fact that it costs nearly $2 billion right now to make a drug, a couple $100 million to make a big medical device. And the corporations will fund that early on so that they get the return later on with the intellectual property and the advanced machines that they make. You also have headquarter manufacturing locations, and you'll be man centers. And so they know how best to utilize their facilities, and you do that with medical equipment as well. So what's my assessment of Saudi Arabia? We've got a little green, we've got a little yellow, and we've got a little red. And I look at the green because I know we've got some great companies. You think about Gulf Medical, Themar, and a couple others. You've got a lot of companies that do great distribution, right? In-service, installation of the product. And I think we continue to grow that. I think that's important. And I think the key piece of it, though, is that you're in that invent it and make it zone. And I think we've got some good things going in the invention and the early stage IP work. But then we also need to get into the manufacturing space. And I want to talk about those and not take too much time for you. So 2 slides on inventing it. So when I think about KSA and I think about inventing it, you've got to compare yourself to the global intellectual property landscape. And in that top right box there, you can see that Saudi Arabia is certainly not at the top, which is one of the strongest patent offices to utilize. They're 23rd in the world of countries that are importing patents and registering them there. And then KSA's very own patent office, under the Ministry of Commerce and Investment, takes about 30 months for a final decision. I point out that the most innovative countries in the world, China, US, Australia, Korea, and Japan, really range from 22 to 14 months, right? And so if KSA wants to become a intellectual property business, really driving a industry, you need to start moving that down the line. So I call on that Ministry of Commerce and Investment to consider, how do you speed that up? Whereas the rest of the medical device space needs to start to drive inventions. So I look at it, I say, you know what? How do we improve inventing it? I think Saudi Arabia needs to view intellectual property as an export as important as anything in the country. You need to invent and send out into the world. And we know the top exports from Saudi. We know what counts. Intellectual property should count as much. We need to accelerate the Saudi Patent Office patent review and granting velocity, increase academic grants to focus on research from the academies, establish an incentive plan. This is a way for Saudi nationals, Saudi citizens, whether they're in country or out of country, to be driving IP. And if they become rewarded for it, it drives the right behavior. And it's reward for not just necessarily the Saudi Patent Office, but the US Patent Office, the Patent Office in China and Japan and others. Driving Saudi citizens to be part of the IP landscape is very important. And increase the direct incentives for KSA-based businesses that submit and are awarded patents. And right now, we're in the process of getting the Patent Prosecution Highway set up, the PPH, and that accelerators, ensure the SFDA, the Saudi FDA, registrations are rapid, and celebrate our great innovators in Saudi Arabia. I mentioned a few here, but on the right-hand side, I say how many global medical technology patents have been filed, and if you look at it, it's doubled since 2008 into 2018, and it's only continuing to grow. So Saudi needs to be part of that. So if I just keep on moving, make it. This is another key piece, and I'll move through this to say, all right, this is where we're going to get into manufacturing. If you think about what medtech CDMOs are often utilized in the pharma space, but medtech is using CDMOs as well. This is an industry that from 2019 to 2025 will double in size. It's growing at double-digit rates, about 12%. And there's a number of different industries. We talked about respiratory care just a little bit earlier that have multiple areas of growth. Now, the med device market globally is only growing about 5%, but the CDMO market is growing over double that. And so I think that's very important to understand and to become a part of. The CDMO needs a couple of specific things, prerequisites. You got to have quality levels. You got to have regulatory compliance, and you got to have a business case for an OEM from a big manufacturer or a startup, a group that just wants somebody to manufacture their product that they've invented. You need to have a good business case for them. And I think the labor rates of Saudi Arabia actually make a lot of sense. There should be some good manufacturing in Saudi Arabia because exports are going to be where the economy begins to expand and actually differentiate itself. So differentiators, high innovation, high technology, move in that direction. Scalability, can you ramp up and ramp down with market demands? Geographical footprint. There's not much in the CDMO space in the Middle East, and access to Africa, access to the subcontinent, access to Eastern Europe. These are things that Saudi has, and so that geographic coverage might actually get you to that emerging space that very few have. And then a one-stop shop. If you could be a complete CDMO, great. If you could be just a CMO for a component, okay, you got to start somewhere. There's other success factors we can talk about: commercial excellence, portfolio optimization, these types of things. But I won't belabor the point, but you get it. We can get this industry off the ground. Here's some examples, right? We've got companies that show up across the spectrum, and these are just a few. And so you take these into account, and you can make either these partners, or you can try to create a similar setup as these businesses within Saudi. So I'll just move forward. So why do OEMs use those CDMOs across that value chain? Cost reduction. They really want to make sure that they can get the parts cheaper, right? It's an option for startups. Startups might invent something, they have a prototype, but they need a company to build it for them. So it's not all just being a manufacturer for a large multinational corporation. Lots of times it's for startups. And that's why I say make sure you get your IP off the ground, because then you'll really start to get the startups looking for your CMOs and CDMOs. Market access, getting access to the Saudi and GCC markets. Technical insight, a CDMO might have some capabilities that they've invested in. Product speed, getting things to market quickly. Quality controls, a certified site that's been audited, that's been reviewed, and of course the scalability. So in some cases, KSA has these types of things. In some cases, they don't. But it's an area that must be invested in, and somebody must drive this as a business leader and as someone wanting to expand this space. So if I just take us, nope, I had not. So this is the slide I just spoke through. So talked about why OEMs use CDMOs, right? So you've got those businesses. We need those types of businesses in KSA. So very last slide, take action. You got to light the candle at both ends. We've got the commercial groups going. We've got them growing, but we need to get IP and manufacturing into place in KSA. So play the long game. Innovation and manufacturing take a long time. Start in the early stages. Get the academics going. Incentivize IP. IP should be a great export from Saudi Arabia. Build on partnerships. Encourage other CDMOs. Those logos that you saw on the slide. Bring them into KSA. Have them open a site. Encourage that. Get products manufactured in the borders, and then look beyond the borders. Send the products out. Send it around the world. Make KSA products and KSA innovation something to be sought after. Be fast and be easy to work with. It needs to be simple for somebody to get a Saudi visa, which I think was all just blurred out there. But make decisions. Don't let perfection be the enemy of progress. I think we've all done projects in the Middle East. There's a lot of people to go through for decision making, and I challenge everybody to make the decision, move forward. You will continually change direction as you go, and then build on success. You've got some great companies doing great work with medical devices there. Encourage them, build on with them, invest with them, and I think you'll have an opportunity to build a great medical device market. Bushra?
Thank you. At this stage, we are going on break. It is 4:42 Riyadh time, and we are supposed to come back at 4:40 sharp. So we have 18 minutes break. Thank you all.
Great.
And we'll come straight for the panel, and Raymond will be the leader of the panel.
Thanks.
The pandemic has taught the world of the importance of science.
And our mission is to improve the health of our communities,
including a clear message about how the benefits will flow to the community more broadly.
Riyadh Global Summit for Medical Biotechnology aims to push the boundaries towards a targeted and sustainable medical biotechnology enterprise in the Kingdom of Saudi Arabia. Let's hear from the Summit Chairman and the Summit Vice Chairman.
Riyadh Global Medical Biotechnology Summit 2021 present truly a fascinating program as a blend of sessions dedicated to strategy development, business development, as well as other sessions dedicated to technology development, infrastructure development, to serve truly establishment of medical biotechnology ecosystem vibrant in the country, as well as other sessions dedicated to scientific topics such as genomics, genetics, gene editing, stem cells, and others. All of those are being delivered by top leaders in the field from global pharmaceutical company and biotechnology companies from around the globe, as well as leading academic institutions with the aim to forge global partnerships that are of interest and will impact the country and the rest of the world.
محمد.
Welcome back, everyone. Hope you enjoyed your break. So now I will hand over to Ms. Bushra bin Saud, President of Danahir Corporation, Middle East, Africa, and Turkey, to carry on with the panel discussion with her esteemed panelists. To you, Ms. Bushra.
Thank you. Thank you very much. And now we are getting into the famous Q&A session. I'm going to invite Raymond to lead the session on our behalf. And of course, I'll stay around to make sure that we wrap it up properly. Let's get started.
She's going to keep us on time, of course, which we all appreciate. And Dr. Al-Zarani, we see you. I know you're very lonely up on stage there. We'll come join you soon enough.
Sure.
I'll be down there in a few weeks, so maybe we can meet in person. But listen, I appreciate everybody's input. I mean, we heard a lot about response to COVID. We've heard a lot about data. We've heard a lot about diagnostics and how this is changing the sharing of best practices. So we've got a couple of questions that we've come together from some of the chat room sessions. We're trying to answer those as well. But I'm going to start with Perry, actually. And I just wanted to ask you about, because, I came from GE as well. And, being a global company, you've got the experience of seeing other fast growing and developing markets. And so I asked, what can Saudi Arabia learn when it comes to, adopting and utilizing the best medical technologies or creating a medical technology market itself from those other countries that have developed it?
Okay, thanks, Raymond. Well, listen, I think you touched on some of the elements during your own presentation today. I mean, you really need to set up an ecosystem and an infrastructure that is favorable to doing development, building expertise in the area. There's an educational component as well. And, there are opportunities with grants. You touched on this as well, which help build that infrastructure. At least they exist in Europe. And we try to leverage those when we can, because when we can find synergies between what a government's trying to institute on behalf of its people and ecosystem, and have that overlay with our own development interests, we can participate in a grant together, which helps accomplish both goals. So I really think it's about making that decision, building that infrastructure, putting the things in place to enable those types of opportunities for industrials to come in and help you on that journey.
Great. No, thanks. Thank you. Insightful. Dr. Al-Zarani, up on stage there. I'll reach out to you. I said, med tech. You talked about, very close to my heart. In the respiratory space. We were building CPAPs and ventilators. Actually, during swine flu, if anybody remembers that one, a number of years ago. But med tech is huge. Med tech and diagnostics is just an absolutely massive global industry. So, what are the sub-segments that you think, KSA should play in to really start to create more innovation and maybe kind of move a part of the industry forward?
Thank you so much for this question. Really, this is a very important question. And actually, we figure out this kind of misunderstanding from the local companies when they bring the product that I already presented during the pandemic. So if you remember, there are three categories of product that been provided by the local vendors: EMBOBAG and CPAP and critical care ventilator. Actually, when the people who brought EMBOBAG to the discussion and how can we take it and make it as a plan B if there is a crisis happening. So actually that was not the need of the market. So that's the first mistake. So the first mistake happening from the local manufacturing, they have to know what's the market demand. Or maybe you have to create the market demand in order to start putting all of these kind together. So that's one of the things. And even we told them in the beginning, we told them, guys, we appreciate all of the effort, but just shift it a little bit to something else. This is the need. If we're going to go plan B, we'll not really need EMBOBAG. EMBOBAG could damage the patient lung. It cannot maybe also help the patient. So one of the important things is to know to have a clear market in international companies, and it could be sold in the local market with competitive pricing, and also to make it, available in this market and also in the region. As we remember in the first session that with the opening, the plan of Saudi Arabia to be hub in the market and to hub in the region, all of the Middle East region. And I think first that all of these local companies need really guidance, need a clear data to access the market, to know what exactly need in the market in order for them to pursue which kind of strategy they want to build. Only medical materials, they want to build ventilators, maybe they want to build only just kind of spare parts. It doesn't need to start big. You can start small and then grow a little bit. So that's my comment on this.
Yeah, no, absolutely. It's very insightful. I think you're absolutely right. You've got to start somewhere. You can have a vision, absolutely, but you've got to take that first step. And even if it's the spare parts and those types of things, it matters. You start you get into the value chain, right? So that's fantastic. Thank you. I'm going to go to Sharif. Look, when it comes to developing and growing a diagnostics, a medical technology type market, you've got to have a culture around it, and you got to have an innovative culture. And, we all kind of talked about how, the speed that COVID brought us forward with it, taking action and doing things that only took, it used to take months or years, in only a couple of weeks, which is great. And it proved that we can do some fun things. But how do you create an innovative culture in KSA? What are some thoughts and some steps around that so that we can get further into med tech and diagnostics?
Hey, thank you for the question. For me, it's all about digital transformational now. Creating more value with efficient cost in the new health ecosystem. Integration of data from different new technologies is a key challenge, and it would be an opportunity and a driver. Adapting to the digital era requires shift towards flexible and risk-taking mindset. It means letting go of outdated processes and trusting that disruption will yield big results. For example, telemedicine, artificial intelligence-enabled medical devices, blockchain electronic health reports are just a few concrete examples of digital transformation in healthcare, which are completely reshaping how we connect and interact with health professionals, patients, how our data is shared among providers, and how decisions are made about our treatment plans and healthcare outcomes. Innovation is the name of the game here, with the main goal of streamlining physicians, optimizing systems, improving patient outcome, reducing human error, and lowering cost at the end through amazing web and mobile experiences. That's in summary from my side.
Great. No, thank you, Sharif. You mentioned you got to do away with outdated processes. You've got to move forward. You got to break the mold. And so I think you're absolutely right. It's good to understand that, because we're going to need to move faster and more innovatively, right? Ellie, I'm going to put you, sir, on the spot a little bit. Bushra asked you kind of what are the two challenges in the market, but actually we had a question in the chat room. And it was, I thought it was a good question, and I put a quick response myself on there before we hop back in. But someone asked, how do I get into medical devices, right? And so someone who's had a career in medical device and the technology, and now here it is, it's changed so much. You're talking about all this data and how to tie that together. It's evolved a lot since you and I first got into it. So how does somebody get into that? We have a lot of people watching and listening right now, and it'd be a way to get that set up. So how do you get somebody in?
if I understand well, the question is like, how do I get into the business of making medical devices, right?
Yeah, exactly. Like, just how does somebody become part of the innovative culture and creating medical devices?
Good question, and I'll try to be very brief. In my perspective, because this is very subjective, medical devices of 20 years ago and medical devices of today are very, very different. We used to be chasing the data in the past. So we wanted to have medical devices that will give us signals about the health of a given patient. Today we have tons of those. And the next generation of medical devices is medical technologies, basically, which is the data. What do we do with it? So it's kind of an Uberization of the medical devices business, right? So you have the hardware part and you have the software part. In the past, the software was a very tiny piece of that ecosystem. Today, it's the biggest piece of that ecosystem. And we are in a storming phase. The medical device technologies, medical device companies, technologist companies, and even non-healthcare companies, by the way, coming from other backgrounds, are coming into saying, all right, there's a race today of making sense of this data generated by medical devices. So without being too objective or subjective here, I think the next generation of medical device companies is going to start from the end, not from the beginning, which is, what is it that we're trying to achieve? How do I achieve it in a smarter and less costly way? So how can I be most cost effective? How do I allocate the right resources? And shall I start with the hardware or shall I start with the software? Right? That's one angle to answer your question. Right? There are many other angles, but this is how it personally.
Yeah, exactly. It's not always a classic career path. Electrical engineering is a great thing, but software engineering and computer science becomes more and more a part of this. So that's great. That's helpful.
BNP has never had any hotels to manage in the past.
Yeah, exactly. So
there are many examples like this that you can find today.
Mm-hmm. Indeed. And Ian, if I could go to you, sir, for a final point. Just in summary, why is medtech and diagnostics a great market for KSA to focus and support for growth?
That's a good question. I think the curse and privilege of speaking after everybody else kind of meant much of the answers have already been said. I mean, I think we heard throughout the session today of both the challenges and opportunities in this area. We've seen, we're living in the middle of a pandemic. We are not out yet. If that doesn't highlight the need for investment and investment in the area of diagnostics and medtech, then I'm not sure what does. But when you look beyond that, I think we heard from Perry, for example, talk a lot about the challenges, but certainly opportunities for improvement and enhancement of the way we deal with oncology, for example. We have an aging population. Oncology rates are not going anywhere. I think if you look at the healthcare challenges moving forward, we spoke about AMR, but oncology, non-communicable diseases, other diseases associated with an aging population, the challenges are only going to get richer and more difficult, and so we have to stay ahead of it. We owe it to our patients and we owe it to the community, and I think it is an area that unfortunately will continue to grow. I think medtech and diagnostics, the need is only going to grow. It's a future area, and I think somewhere with the mindset and innovation that Saudi is trying to develop or is developing, I think they place themselves well to think differently. I mean, as Ellie just said, the market and the world we live in is very different from the one 10, 20 years ago where it was dominated by laboratories and technicians and things like that. Now, many of the solutions are coming from the digital element and things like that. And I think going back to just to touch on the career path, I think we're seeing value in our company of bringing non-healthcare professionals from all walks of life into our business, because the business and the market is going in who knows what direction. There's a lot of opportunities for the market and for people within that to come and look at it from a different lens. Biochemists like myself are no longer necessarily at the heart of the innovations coming from others outside of the industry. So it's an exciting time. There's a lot of growth, but still the impact that we can have on society is probably the number one reason that we need to invest in this area.
Absolutely.
No. And I think for all the young people out there watching, this is just an area that is so rich in opportunity, whether you're at the tail end, like Ellie suggested, with data integration, the data's there, just putting it together, or disruptive technologies that are either benefiting caregivers, patients, payers, right? We've got a productivity problem and payment problem too. A lot of good ideas are needed. They continue to be needed.
Yeah. Indeed. Great. No, thank you. And don't sell yourself short, Ian. We're still going to need biochemistry engineers. We're going to need people. So
I was never a very good biochemist. So,
I appreciated everybody. Thank you for the input, and I will hand it back to Bushra to go ahead and wrap up this great session.
Thank you. It was really a pleasure to be listening, without having the pressure to question each and everyone. So thank you for the 30 minutes of very interesting insights. Now, I mean, it will be not giving credit to everything that was said if I could say that I summarized, but I could say I picked some highlights from what has been said. I think that would be more, giving credit to all the quality presentations that I have seen. I could say that as a wrap-up, I could start by the molecular diagnostics have come to the forefront of this battle against the COVID-19. That's something that was striking to me as of the beginning of this session with Ian and Sherif's presentation. Then they also mentioned that as we look forward to a time beyond the pandemic, we have a huge opportunity to leverage these innovations in the diagnostics and medtech industry to address critical public health threats. And I think this one is important because it's like opening a huge opportunity for us as industrials, but also for the people to be well treated, even more importantly. I also was quite impressed by the part that is more linked to the technology and quality of data gathered, analytics generated, and how this will be shaping the future of diagnostics. This in general, and more specifically on the examples from Perry when it comes to molecular medicine, when it comes to molecular imaging, all the examples that the leaders from GE Healthcare has brought were really fascinating, and I think there is a lot to learn from that. And finally, I would conclude with what Raymond left with us, which is more around a medical device industry is a long trajectory. And what matters is just to get started somewhere. If I can summarize it in a simple way, it is not to get in a complex mode, but rather go step by step, every day, 20 miles. That's from a book that I loved reading some time ago. But that's important because we go step by step, but it requires discipline. It requires passion, because after a number of steps, then we have the results that we want. And that's how I would like to summarize. Before concluding, I would like to really thank the speakers, all of them, with the perspectives that you brought. They are very different, they are diverse, and that's the beauty of it. I would like to thank the organizing sponsors, the team that was supporting us, because it was a great opportunity to share our views and to be able to bring our piece of the puzzle. I wish to the NGHA a successful summit. And with that, I think we are even ahead of time. We can conclude unless there is any question that anyone would send us from the chat. I'm looking at the chat box and I don't see any question, unless you guys see anything. But with that, we are completing the session. Thank you all. Thank you very much.
Thank you. Thank you.
Thank you for the opportunity.
Bye-bye.
Thank you.
Take care, everyone.
Cheers. Bye.
Thank you all for sharing with us your greatly informative insights on how the application of medical technology develop treatments, prevent diseases, and enhance the human lives. Ladies and gentlemen, now we'll be diving into a very interesting topic, cell biotechnology and gene therapy. To start the session, it's my pleasure to welcome the chairman of the session, Dr. Mariam Fuch, Global Therapeutic Area Lead, Cell and Gene Therapies, Oncology, Regulatory Affairs at Novartis. After 5 minutes, stay connected.
My job is to make college easier because
Yeah? Okay. Welcome back, everyone. To start our next session, it's my pleasure to welcome the chairman of the session, Dr. Miriam Fuchs, Global Therapeutic Area Lead, Cell and Gene Therapies, Oncology, Regulatory Affairs at Novartis. Dr. Miriam, can you hear me?
Yeah, I hear you well. Do you hear me?
Yes, it's a great pleasure to have you here with us today. The mic
Thank you so much. Thank you. I would like to thank the scientific and organizing committee of this excellent Medical Biotech Summit for inviting to the session today to evaluate aspects of the innovative field of stem cell and cell and gene therapies and to share our experience and learnings. I would also like to welcome the esteemed speakers of this session. The vision that cell and gene therapies can deliver unprecedented and transformative clinical outcomes for patients that may enable them to live disease-free for years or undergo a rapid and dynamic evolution with an important promise for patients globally for improved and novel therapeutic treatment options. The session today will allow us to hear some insights from experts deeply involved in this exciting field of advanced therapies. I would like to introduce Dr. Badar Lamari as our first speaker to share lessons from B-cell malignancies with chimeric antigen receptor T cells. Dr. Badar obtained his MBBS from King Khalid University in 2005 and finished his residency in internal medicine from King Abdulaziz Medical City in Riyadh, and obtained the Saudi board in internal medicine in 2010. He then carried on to complete his clinical fellowship in hematology oncology in 2016, as well as a fellowship in bone marrow transplant from the Washington University of Medicine in St. Louis. He also obtained a Master of Science in Clinical Investigation on the Translational Medicine track from the same institution in 2017. Currently, Dr. Badar is a consultant in stem cell transplantation and cellular therapy division at the Department of Oncology, Ministry of National Guard Health Affairs, Riyadh, and he has multiple publications on his work on novel therapeutic targets for graft versus host disease in experimental murine graft versus host disease models. And his current research work focuses on the development of cellular therapy for cancer management. I would like to welcome Dr. Badar. Thank you so much.
Well, thank you so much, Dr. Miriam, for the introduction. I would like to thank the organizing committee for inviting me for the talk today. Today, I'll talk about the chimeric antigen receptor T-cell therapy and B-cell malignancies. And probably there will be some overlap between my talk and the talk of Dr. Miriam. But I will focus mainly in the clinical aspect of this newly innovative therapy. So let's start first by understanding what is the T-cell response to cancer cells, starting from the antigen presentation of the cancer-related peptide, and then followed by the appropriate recognition of the T-cells. And this recognition of T-cells will lead to an appropriate killing and eradication of these cancer cells. However, cancer cells can go through a multi-step journey toward the ability to escape the immune system. And this is called the cancer immunoediting process that end up with escape from the immune system. And this mainly happen through multiple mechanisms, including the effect of immunosuppressive microenvironment, by the effect of other cellular components, and also by the cancer cell itself ability to upregulate certain molecules that enable them to escape the immune system. So how to help the T-cells to tackle this battle against the cancer? There are multiple ways and methodology to deal with this, and the known one is the vaccine effect, and this can enhance the antigen presentation to the T-cells. But most importantly, and the one that's really showed an excellent effect is the enhancing of killing through the introduction of the anti-PD-L1, PD-1 checkpoint receptors. However, also cellular therapy plays a major role here. I'm talking about the enhancement of the T cell recognition of the cancer cells through what we call it the chimeric antigen receptors T cells or the CAR T cells. So what is CAR T cells? CAR T cells basically having the specific receptors that has the ability to hijack the T cells and make them recognizing the tumor antigen directly without the need for HLA dependency. It's completely does not rely on the MHC restrictions. There are multiple generations. The first generation of the CAR construct composed of the short variable chain of the antibodies that can target certain antigens. However, did not go through because the lack of the co-stimulatory molecules that was started from the next generation of the CAR construct. And there is now currently a new generation of the third generation and fourth generation of the CAR construct. But the most successful one that is clinically applicable now is the second generation. The journey started a long time ago, like 3 or 4 decades ago, when the first generation CAR construct was generated. However, this did not have appropriate clinical effect, mainly, as I said, because of the lack of the co-stimulatory molecule that has the ability to enhance the T cell proliferation and enhance the killing effect of the T cell and the persistence of the T cell in vivo. The first, second generation CAR was generated around 2003. 10 years later, the first clinical application of this CAR construct was first reported in 2009 in patient with CLL, followed by a patient with a pediatric patient with ALL. And then in 2017, we have the first report of the pivotal study that led to the first approval of the CAR T cell, which is the CD19 CAR T cell. And since then, multiple approval happened up until recently this year. We have the last recent approval of the CAR T cell for multiple myeloma. So how is this generated? It's a complex process, and first by do the leukapheresis and obtaining the apheresis product from the patient. Then this cell product will be shipped to the manufacturing facility where it will undergo multiple process of manufacturing, starting by T cell isolation, then activation, then transfixion with the CAR construct that's either using the lentiviral or retroviral that carry the CAR construct that can be safely expressed on the T cells, followed by T cell expansion, and then shipped back to the clinical institution where the CAR T cell will be infused after lymphodepleting chemotherapy. One of the first and the best studied receptors is the CD19 antigen, where this showed a successful story and led to multiple approval of clinical products using actually CD19 as a target for the CAR T cells. And why CD19? Because it's kind of lineage restricted antigen that is expressed nicely on multiple B-cell malignancies, including ALL and B-cells non-Hodgkin lymphoma. Talking about what is the outcome pre the CAR T cell era, the ALL patient has a dismal outcome after the first relapse. This cohort of 609 patients showed that the overall 5-year overall survival is almost 7%. with many of the experts thought that a relapse refractory ALL patient is almost an incurable disease. The same story is for the refractory diffuse large B-cell lymphoma. Well, for this cohort showed that the median overall survival for those who are relapse refractory in a rituximab era is having the overall survival only of 6 months. What is the status of B-cell malignancies in Saudi Arabia? According to the Saudi Cancer Registry 2016, non-Hodgkin lymphoma ranked among the second most cancer in adult male and the fifth in female. In 2016, there has been a total of 82 children and 114 adults diagnosed with BALL, diffuse large B-cell lymphoma diagnosed in 474 adults. Recently, my colleague Dr. Musab Damlaj published the data from the experience from the National Guard in Riyadh of the outcome of 85 adolescent and adult ALL patients in CR1. As you can see here in the curve, the event-free survival for those who went into transplant is around 70%, where those who did not went into transplant around 40%. Still, you can see there is a good proportion of patients who relapse even post-allogenic transplant, and usually those patients have a dismal outcome without an appropriate line of therapy for them. Currently, the CAR T-cell approved product is mainly CD19 product, CD19 targeted products. 3 of them are approved for diffuse large B-cell lymphoma, and 1 for ALL and 1 for mantle cell lymphoma. And also more recently, anti-BCMA was approved for relapse refractory multiple myeloma. We'll talk now about the result from the pivotal trials that led to the approval of those products, starting by the ELENA trial for the Tassicell for children and young adults with relapse refractory BALL. Around 92 patients enrolled into this open label phase 2, single arm phase 2 study with patients with relapse refractory B-cell ALL. They underwent lymphodepletion with fludarabine and cyclophosphamide. And the overall survival overall response rate at 3 months was 81%, which is impressive for such a patient population who are relapse refractory even after allogenic stem cell transplant. The overall survival at 12 months was 76%, and event-free survival of 50%. The next study is the ZUMA-1 for axi-cell in refractory large B-cell lymphoma. Again, this is CD19 CAR T-cell with a CD28 as a co-stimulatory molecule. The patient population were patients with a large B-cell lymphoma, including DLBCL, primary mediastinal, and transformed follicular lymphoma. Those usually a patient with a refractory to at least 2 lines of therapy, and also including some patients who relapse post-autologous transplant. 111 patients enrolled in this study with the primary outcome overall response. As you can see here, the overall response rate was 82% and CR rate was 58%. With a median follow-up of 27 months, the median OS was not reached, and the median PFS of 5.9 months. The second study that led to the approval of the Tassicell for relapse refractory diffuse large B-cell lymphoma is the Juliet study. Kind of similar in inclusion criteria for the axi-cell. And you can see here the overall response rate of 52% and CR rate of 40%, with an estimated 12-month rate of relapse-free survival of 65%. And among those who went into CR is impressive 79%. The most recently approved CAR T-cell for diffuse large B-cell lymphoma or large B-cell lymphoma is the Lasocell. which has also the 4,1-pp, which is similar to the Tacicil molecule. However, in this product, they infuse the CAR-T in a 1-to-1 ratio of CD4 versus CD8 CAR-T. The inclusion criteria here is unique by allowing a patient with secondary CNS disease, and also allow patient who had previous exposure to CD19 CAR. As you can see here, the overall response rate of similar to the previous two products. So this is really good data for those patients who had relapse refractory disease, who usually at this stage of their disease, they go into experimental treatment or palliative care. However, this kind of treatment led to durable remission, which is something we haven't seen for a while for such refractory. There's no understandable yet mechanism. However, there are multi-research efforts to understand better this kind of unique toxicity for the CAR-T cell. Luckily, there is multiple effort from multiple cellular therapy society that established guidelines how to manage this kind of toxicity and how to mitigate this kind of toxicity. And as you can see here, this slide summarizes the rate of such toxicity in the three trials, the ZUMA-1, Juliet, and the Transcend trial. In the upper panel, you can see the CRS rate, and the lower panel, you can see the neurological event rate. The grade 1 is almost about 81%, grade 1 to 2 in ZUMA trial, while in Juliet around 40%, which is similar to a study that tested the lysocell. And neurological event, you can see the Axicil might have a higher grade 3 neurological event when compared with the Juliet and Transcend study. As I said, this algorithm summarizes how this risk of toxicity can be mitigated and how to interfere to manage this kind of toxicity by using steroid and monoclonal antibody against IL-6. Long-term toxicity, including B-cell aplasia, which can be seen up to 15% in adults with relapsed refractory B-cell lymphoma and up to 43% in pediatric patients with ALL. Cytopenia, which is also one of the common toxicity that can be seen. Rarely you see sometimes, can see some form of aplasia post CAR-T cell. Infection also as a consequences of this therapy also can be one of the major side effects of this treatment. We'll talk now about the barrier to application of CAR-T cell in the real world and the barrier to clinical application of CAR-T cell therapy. So this CAR-T cell, there have been many patients who relapse post-CAR-T cell showing CD19 negative relapse, and there is multiple mechanism for how the cancer cell can evade the CAR-T cell and develop CD19 negative tumor cells. Also, the on-target and off-target toxicity, this is one of the challenges for this kind of therapy. And also that CAR-T cell persistence, in vivo persistence of the CAR-T cell, also one of the things that cause an obstacle, and the suppressive tumor microenvironment. This slide will kind of summarize the current research effort to develop a better CAR-T therapy, CAR-T cell therapy that can overcome those barriers. For example, exhaustion can be overcome by the incorporation of BD1 inhibitor, and also in vivo persistent can be improved by improving on the co-stimulating molecule and the secretion of the appropriate cytokines that lead to better persistence of those CAR T cell in vivo. Multi-target effect also we've seeing now dual and even triple CAR T therapy that target more than one antigen simultaneously, and also ways to reduce toxicity in form of safety switch and so on. In terms of manufacturing challenges, one of the things that associated with CAR T cell in general is the high cost. It's the costly treatment that can start from $370,000 US dollar for a single infusion for one therapy, and this is only the price tag for the product alone, without the associated cost of pre and post infusion, and also the limited availability of this product. It needs to be manufactured in well-established, centralized manufacturing facility that has the ability to scale up the production of this kind of therapy to meet the clinical need and demand, which is mainly located in the US and in Europe. The other thing is manufacturing time delays. Again, it's due to mainly the centralized production of this CAR cells. Example of manufacturing delay in the ELINA study for relapse refractory ALL, there has been around 18% of patients who had the apheresis product collected did not receive the CAR T cell for multiple reasons, including poor cell growth, or death of the patient while from the disease progression while waiting for the product, or the development of interval adverse event that make the performance status of the patient to be impaired and unable to receive the product. The median time for cell infusion was around 45 days, ranging between 30 to 100, up to 105 days. This slide will show you how complex it is to manufacture such a therapy, and then T-cell expansion followed by the cryopreservation of the product, then to be shipped to the treating center and to be infused to the patient. However, there has been a recent interest in the multiple academic institutions around the world for using a machine that can do the automated manufacturing of the T-cell, where you can manufacture the T-cell in the closed system, one single machine that can do all the process of the manufacturing, including the T-cell separation, activation, transfection, expansion, then harvesting, then to be ready for cryopreservation. And there has been multiple reports from different academic institutions that report their outcome. This one just an example for one of the published abstracts in the ASH, where using CD19 construct in 12 patients, and the CR rate was 67% with overall response rate of 75%. Two patients have died because of death, including progressive disease or from the CRS. And in the right panel, you can see the persistence of the infused CAR T. At KMARC, we have been working collaboratively with the clinical part and the research team in the KMARC to incorporate this kind of modality and adapt this type of therapy for our patient. We form two different committees, working groups from the clinical side and from the laboratory side to work together and collaborate to make this type of therapy available for our patient with the help from the clinical trial unit at the KMARC. Right now we have the IRB approval, and we are in a process to get the FDA approval for this project. This will be my last slide. You can see here from the slide multiple clinical trial is currently active and recruiting patient around the world. Probably the highest rate is located in China. Thank you very much, and I'll be happy to get any questions. Thank you.
Thank you. Are there immediate questions? Thank you so much for this excellent overview and introduction to CAR therapies, and thank you so much for sharing your local activities to access for patients to CAR therapies with the local clinical trial, which I understand is just receiving approval. Our next speaker is Dr. Joseph Fraietta, and he is coming from one of the key centers of pioneering CAR development, the University of Pennsylvania. Dr. Fraietta has completed his training as a postdoctoral fellow in Carl Schulz's lab at the University of Pennsylvania in Philadelphia, where he conducted research on the mechanisms underlying the persistence of CAR T therapy in patients with CLL. In 2016, Dr. Fraietta assumed the directorship of the Product Development Laboratory at the Center of Advanced Cellular Therapeutics in the Perlman Center of Advanced Medicine. And during this time, he developed novel approaches for the treatment of cancer through genetic modification of T cells that contributed to the initiation of multiple clinical studies. He is currently the director of solid tumors immunotherapy labs at Penn, where his research is focused on enhancing the specificity and efficacy of genetically redirected T cells. And he's currently investigating the role of epigenetics in the effectiveness of T cell responses. He has made several important contributions to the field of immunotherapy, and his high-impact findings have been published in high-profile journals like Nature Science, Nature Medicines, and he has been the recipient of numerous awards. I'm very pleased to welcome Dr. Fraietta to share with us his insights on genetically enhanced T cells in designing, engineering, and building cancer immunotherapies.
So thank you very much, Miriam. It's a pleasure and truly an honor to be here with all of you today to give you sort of a snapshot of our initiatives at the University of Pennsylvania to both develop and further improve our cellular immunotherapy platforms based on CAR T cells for the treatment of advanced cancers. And the primary focus today will be on the hematological malignancies, but maybe considering the previous talk and Miriam's later talk, I'll focus on solid tumors a bit more. So since we've heard in the first lovely talk quite a bit about the successes of CAR T-cell therapy, particularly in advanced B-cell malignancies, perhaps I'll jump right into talking about the challenges. That is to say that despite these successes, particularly in the liquid tumor space, we still have several barriers to address. And some of the unmet needs in CAR T-cell therapy are listed here, namely poor T-cell expansion and persistence, as we heard about, T-cell dysfunction, tumor escape, perhaps due to antigen loss or down modulation, and then, of course, getting this therapy to work well in solid tumor indications without toxicity has been a goal of the field and is still an ongoing goal of the field. So, just to focus in on this a little bit more in the vein of improving clinical outcome, the importance of T-cell intrinsic fitness in the setting of CAR T-cell therapy, I think, is best exemplified when you compare the efficacy of the same CD19 CAR construct that we developed in collaboration with our partners at Novartis in two different advanced leukemias. And so one is acute lymphoblastic leukemia and the other is chronic lymphocytic leukemia. And so we and others, as you heard about, have achieved really high complete remission rates of over 80% to 90% in some trials in the setting of pediatric ALL with the anti-CD19 CAR developed at the University of Pennsylvania. But we've only observed a complete response rate of about 25 to 26% in chronic lymphocytic leukemia. And so the potential reasons for disparities in CAR T-cell therapeutic efficacy between these indications are listed up here, and they can be attributed to innate humoral and cellular immune deficiencies, including in these intrinsic T-cell defects that I'm referring to, that are characteristic of certain cancers like chronic lymphocytic leukemia, and that often worsen with disease progression. So as you can imagine, the above factors, either alone or in combination, may influence CAR T-cell potency and effectiveness. And clinical outcome, of course, as I'll elaborate on, is tightly tied to this intrinsic T-cell fitness. So we've conducted several careful investigations of responders and non-responders in CD19 CAR T-cell therapy of advanced leukemia, and I'll talk about chronic lymphocytic leukemia here. And our results indicated that clinical response to CAR T-cell therapy in chronic lymphocytic leukemia really depends on CAR T-cell proliferation and in vivo persistence. But in some cases, we can't expand enough CAR T-cells to meet the dose, particularly for heavily pretreated subjects, so we can't really even get out of the gate in this situation. And then in other instances, even if we get adequate ex vivo expansion and we can manufacture the products, patients who don't respond following infusion really demonstrate limited in vivo proliferation and essentially no persistence of those products. And so, we did a deep dive into this, and our extensive biomarker studies really revealed that even at the time of T-cell manufacturing, CAR T-cells from complete responders, when you look by RNA sequencing, as you can see here, are enriched in transcriptomic programs of early memory T-cell differentiation and self-renewal capacity, sort of what you would expect, right, from many preclinical studies. But if you look at the non-responder patient CAR T-cells, they exhibit signatures associated with T-cell exhaustion. But the good news here is, I think, and as we treat many more patients and do more of these retrospective analyses, we are actively identifying pathways and several mediators within those pathways to target for improving the success of CAR T-cell therapy. We've shown that CD19 CAR T-cells in advanced leukemia can expand dramatically in vivo. They could also eliminate high volume and bulky disease, and they can persist. And so we've recently published some of this, and I really have to update this slide, but with the product that we use, the CD19 CAR T-cell product, we've documented persistence in some of the earliest patients whom we've treated at Penn of over a decade, which is rather remarkable. And long-term B-cell aplasia accompanies these responses, implying that the persisting CAR T-cells really remain functional. And we also have some direct experimental evidence of that as well. And so in chronic lymphocytic leukemia, really the key, I would say, generic correlates of response are that patients who respond to the therapy, they display, again, this dramatic in vivo expansion of CAR T-cells, usually within the first 2 weeks following infusion. And then that's followed by a log normal decay of the product and the proliferation of the product with complete and often persistent eradication of the leukemia cells. And then, the infused CAR T-cells persist long-term, as I mentioned, in the responding patients. And then finally, the persistent CAR T-cells, again, are polyfunctional, which I'm not showing here, but that implies that they're actively perhaps maintaining this long-term B-cell aplasia that we're documenting. A little more on these retrospective studies. So we and others have observed that some CAR T-cells really proliferate differentially in patients, and these can form expanded populations. And so lentiviral or retroviral vectors are the typical transgene delivery methods that we heard about in the first talk, and these viruses randomly integrate into the genome to encode that CAR transgene. Insertional mutagenesis, though, sometimes alters host gene cell activity so that the locations of the integration sites, so where the CAR lands in the expanded clones, really marks genes and pathways that modulate, hopefully favorably modulate, right, CAR T cell growth and also anti-tumor potency. So I find this to be very useful for identifying modulators that could be targeted for the success of CAR T cell therapy. And I would say, you know, a prime example of this is what we published a couple of years ago. And so this was a single case study of a 78-year-old patient who relapsed through multiple conventional treatment regimens. He was infused with CD19 CAR T cells, and this patient had a delayed expansion of his CAR T cells, which was quite unusual. But after this delayed expansion, or following this delayed expansion, he experienced very robust cytokine release syndrome and tumor lysis syndrome. And essentially, by deep sequencing, we couldn't detect any of the leukemia clones. So it was a remarkable delayed response. And upon investigation, we traced this response to a single CAR T cell with a disruption in a tumor suppressor gene, a known tumor suppressor gene known as TEP2. And the cell expanded massively in vivo. I think it underwent, if I remember, something like 30 population doublings and completely wiped out his leukemia. And he's still in remission and doing remarkably well to this day. So from a single cell, and I think that really underscores the power of the CAR T cell approach if we can optimally program CAR T cells. So the big question, as I alluded to, and what, you know, we want to see as a field, I think, is a clear demonstration of efficacy of CAR T cell therapy in much more common and challenging solid tumor sort of indications. And so what factors do we really have to consider that can influence the success of CAR T cell therapy for solid tumors? Well, once CAR T cells infiltrate into the tumor bed, they're faced with major barriers that tumors often employ to blunt immune surveillance. Moreover, you know, when we infuse these therapies systemically, CAR T cell homing to the tumor sites is going to require, you know, the right cytokine chemokine gradients and the right receptor and anti-receptor interactions. And the degree of expansion of these cells is going to be greatly influenced by preconditioning of the patient before adoptive cell therapy. But as I said, even when the cells could get into the tumor, they're faced with a whole host of immunosuppressive barriers that are listed here and operative within the solid tumor microenvironment. So I had a case study here to just illustrate this, and maybe in the interest of time, I'll kind of sum this up quickly. This was a 63-year-old patient with advanced mesothelioma who progressed through multiple biologic and chemotherapeutic treatment regimens and also radiation. And he decided to enroll in one of our solid tumor CAR T cell immunotherapy trials with the product targeting mesothelioma. And so we did see a rather robust, for at least the solid tumor indication, expansion of the CAR T cells in vivo. And what was very interesting, as you could see here, there was even preferential trafficking of the CAR T cells to one of the solid tumor sites that we biopsied. But despite this, and despite a high level of CAR gene marking at the tumor site compared to the peripheral blood, still the CAR T cells were essentially shut down in that solid tumor microenvironment, and we really didn't see local proliferation or elaboration of effective anti-tumor activity. Okay, and another big problem that I wanted to mention that, you know, we have to contend with in the CAR T cell therapy field is adaptive resistance. So CAR T cell based therapeutic approaches for solid tumor indications also need to address how to increase T cell persistence and, again, anti-tumor effector function in this suppressive tumor microenvironment that actually gradually upregulates immunosuppressive barriers due to adaptive resistance following adoptive transfer of the CAR T cells. And so that's illustrated here in our trial of epidermal growth factor variant 3 directed CAR T cells in glioblastoma. And so what was really notable about this clinical study is that following infusion of those EGFRV3 directed CAR T cells, it seemed that from multiple patients and from multiple post-infusion sites, we saw upregulation of a number of immunosuppressive barriers following adoptive transfer of the CAR T cells, such as IDO, PD-L1. We also saw more regulatory T cell localization within these tumors, implying that perhaps the tumor is evolving in response to that selective pressure by the CAR T cells. So this is definitely going to be a problem. So I just want to sum up maybe some of the valuable lessons that we and others have learned from CAR T-cell therapy of solid tumor indications. Despite trafficking of CAR T-cells to the tumor site, some degree of initial proliferation and elicitation of effector activity, clinically meaningful responses are rarely observed in these indications. And then anti-tumor potency is frequently limited by lack of substantial expansion or survival of the CAR T cells once they hit that solid tumor microenvironment. We do, in some cases, see significant decreases in targeted antigen expression, suggesting that there is on-target on-tumor activity, but this is often transient. And this also, I think, highlights that antigen loss and heterogeneity is a significant barrier to successful therapy. And finally, on-target off-tumor toxicity reported in some trials, irrespective of very low antigen levels. So I think this indicates that we can get there with CAR T cells in treating some of these solid tumor indications. In the interest of time, I don't know, Miriam, if I have, I wanted to talk a little bit about universal CAR T cell products, but perhaps I can, leave that to you so that the session doesn't go over. And with that, thank you very much for taking the time to listen to my talk. And I'm certainly happy to take any questions, maybe not now, but during the panel discussion, again, in the interest of time, so that we're, sticking to this tight schedule. Thank you very much for having me. And again, it was an honor to be here and share some of our work with you. Oops.
Thank you so much, Dr. Fraietta. Fascinating talk, and the solid tumor field, of course, is a totally new challenge compared to the treatment of B-cell hematologies, and I hope that the field is moving in the right direction to enable access for patients to CAR therapies in solid tumors. But looking at the hurdles, maybe more research, more evaluation is required. And I'm sure we discuss more about it in the panel discussion. Our next speaker is Dr. Khaled Al-Sayegh. He's an associate director and research scientist at the King Abdullah International Medical Center, Research Center at the National Guard Hospital. Dr. Khaled received his PhD from the Medical College of Virginia in Virginia Commonwealth University. He is specialized in human and molecular genetics, and he did postdoctoral training at the Institute of Advanced Biomedical Engineering and Sciences at the Women Medical University in Tokyo. His research interest focuses on the important aspect of bridging the gap between induced pluripotent stem cells technology and clinical applications. He works on bioengineering robust and clinically safe induced pluripotent stem cells, derived cardiac tissue, as well as on improving hematopoietic differentiation of pluripotent stem cells. Thank you so much. We are very much looking forward to learnings about the Saudi Bank of Induced Pluripotent Stem Cells.
Thank you very much, Dr. Mariam, for the nice introduction. Good evening, everyone. Thank you all for attending. Today, I'll be speaking to you about our effort at KMARC for iPS cell banking. I started with a few slides to present to you a brief background for people who are not in the field of stem cells, but what iPS cells are. Basically, induced pluripotent stem cells, or pluripotent stem cells in general, they have two defining characteristics. They have the capacity to self-renew in the lab indefinitely. You can culture them forever, basically. They will symmetrically divide. And they can also be differentiated in the lab to any cell type found in the human body. So our first encounter with pluripotent stem cells basically is at the human embryonic stem cell stage. And these cells are obtained from day 5 blastocyst stage embryo. So what you're looking at in this slide basically is an in vitro fertilization process. You see day 1, the pronuclei of the mother and the father. As the embryo starts dividing, by day 5, we reach the blastocyst stage. Basically, at this stage, the embryo is made of an outer layer called the trophoectoderm, which will basically become the placenta, and you have the inner cell mass, which is a clump of cells inside that will later solely contribute to the whole human fetus with all of its complexity, all of the systems found in that human fetus. So it's very intuitive to appreciate the potential of these cells. And indeed, in 1998, James Thomson was the first to isolate these cells, to isolate the human embryonic stem cells and to culture them. And after this milestone in stem cell research, many papers have came out where they have differentiated these cells into functional cells like cardiomyocytes that are beating, or insulin-producing cells that can sense glucose levels, dopaminergic neurons, and so on. Sorry. Yeah. So, however, the stem cell culture, human embryonic stem cell research comes with its downside. Basically, these cells are problematic to be used from the ethical aspect, because these cells are, to derive these cells, we need to destruct the human embryo. And for that reason, these cells, this destruction will cause these cells to lose these embryos, and that could be problematic in some cultures and some religions. And therefore, in 2007, a major breakthrough was achieved when iPS cells were derived by overexpressing a set of genes that are known to be highly expressed in human embryonic stem cells. And then later, these cells were indistinguishable in terms of pluripotency from human embryonic stem cells, and they could be differentiated to any cell type found in the human body. And the nice thing is, what's interesting about these cells is that these cells are derived from the patient, him or herself, so you don't have the chance of immune rejection. And the other thing is that these cells does not involve the destruction of human embryos, so you evade the ethical problems associated with the derivation. And lastly, these cells are as pluripotent as human embryonic stem cells, so basically they are as useful. And for that, of course, Shinya Manaka was awarded the Nobel Prize in 2012. Again, the way these cells are derived, basically we overexpress these genes in somatic cells, and this will turn them into cardiomyocyte, insulin-producing cells, and so on. And interestingly, within only 7 years of their first derivation in 2007, in 2014, the first in-human clinical trial was started in Kyoto University by Masaya Takahashi. And in this clinical trial, they used age-related macular degeneration patients that were treated by iPS-derived retinal pigment epithelial cells. You see the staining cell sheets on the right-hand side. These were developed from or derived from iPS cells, and then were later transplanted in the back of the retina after the new vascularization was removed. And this was compared in that clinical trial with the standard anti-VEGF therapy for this type of disease. And these cell sheets have shown their superiority when compared to the standard therapy. And the other clinical trial that is ongoing now in Japan, also in Kyoto University by Jun Takahashi, is to treat Parkinson's disease patients by converting iPS cells to dopaminergic precursors, and then are intracranially injected into the patients. And also another clinical trial that is ongoing. As you can see, this is from Nature News. It's in 2018. This clinical trial is already active in Osaka University, in which they are treating heart failure patients with iPS-derived cardiomyocyte cell sheets that are transplanted on the epicardium to provide this mechanistic support, like we saw in Dr. Larson's presentation yesterday. Another clinical trial also to treat spinal cord injury with iPS cells-derived neural precursors is also now ongoing. All of these clinical trials, we are eagerly waiting for the first reports that will show their results. But what's interesting is that all these clinical trials that I spoke of have all transitioned to allogeneic therapy instead of autologous. And there's a good reason for that. If you saw the first clinical trial that I spoke of, the age-related macular degeneration clinical trial, the cost per patient, if you want to generate autologous cell therapy, is around $1.2 million. And this is to treat a small organ like the eye. But when we go to a larger organ like the liver or the heart, the cost is going to substantially increase. And like one estimation was on liver grafts that were derived from iPS cells, mentioned that these cells could cost up to 10 million cells to be bioengineered. So the patient, because all of this will, we generate the iPS cells from the patient, and these iPS cells are gonna go through rigorous testing to ensure their safety. They are free of any oncogenic mutations. And again, we're going to differentiate the cells to make sure that we can generate functional tissue from them. So all of this, the patient sometimes does not have the luxury to wait for such a long time, and the cost could be, again, prohibitively expensive. So the alternative and why the reason why these clinical trials are moving towards allogeneic is basically to reduce the cost and at the same time reduce the time and wait by having off-the-shelf cell therapy that is optimized for the population. And the way we do that is by HLA hemoleukocyte antigen matching. So the proposed mechanism for that is basically we have what is called a universal donor. Is we go through the population and we identify the HLA distribution in the population. And then if we can find homozygous donors or homozygous for the most common HLA haplotype, these donors can match the large population and large percentage of the patients that are in need. So this prompted us to investigate the feasibility of HLA-based banking in Saudi Arabia. So the first step was to survey the population in Saudi Arabia and see the HLA frequency and distribution in that population. When I first joined Kmark in 2015, Kmark was just beginning a great initiative called the Stem Cell Donor Registry. It's a registry that is targeted towards providing donors for cancer patients and patients with hematological diseases. And what they did is that they collected a big number in that registry and sequenced the HLA in high resolution, as you can see on the right side table. And these are the frequencies shown here. So we accumulated these haplotypes, and we found that if we reprogram 7 of these donors, we can cover more than 10.3% of the Saudi population. So this is the paradigm, the proposed treatment is that we can find this universal donor and then we're going to focus on human leukocyte antigen locus A, B, and DRB1. And as you can see, the top 2 patients, they are compatible, but at the DRB1 locus, the last one is not matching. So at Kmark, we generated our first IPS line, and we were interested in making the cell line as clinically relevant as possible because Kmark is taking the direction of establishing its GMP facilities and cell processing centers. And we want to work in parallel until we develop these therapies and are ready for application later on. So we used simply, instead of skin biopsy, something as invasive as that, we used a 10 ml peripheral blood sample. And from that, we isolated erythroid progenitors. And these cells are free of any genomic rearrangement usually, and they are more abundant in blood than CD34 positive cells. And these cells are pluripotent. As you can see, they have the pluripotency markers. And at the same time, they are karyotypically normal. So at Kmark, we have 2 projects basically that we want to, that's about to start soon, is the generation of iPS-derived biological makers or cynoatrial node cells from these iPS cells. On the other hand, we have another project that aims to generate natural killer cells or CAR-NK cells to target B-cell lymphomas. So in this project, in collaboration with KAUST and King Faisal Hospital, we're going to use single-cell RNA sequencing we have these beating cardiomyocytes here. I don't know if the video can be turned on in this slide by the organizers. And also, we want to do single-cell RNA sequencing from the primary heart tissue, obtained from explanted heart of patients undergoing heart transplantation. The single-cell RNA sequencing is going to allow us to see how these generated IPS products can compare to the real thing. And that will, the end goal of this is to develop biological pacemakers that have pacemaking ability. All right. The other project by Dr. Abir Sufyani is to generate these natural killer cells. So basically, we're going to reprogram the patient or the donor cells, and then we're going to engineer to have the CAR CD19 antigen, and then differentiate it to natural killer cells. And we're going to also employ here single-cell RNA sequencing to assess the comparison between these cells and natural killer cells obtained from the donor. And the ultimate goal of this, again, is to have off-the-shelf therapy to one day be used on Saudi patients. And again, we want to contribute to and collaborate with other institutes in the kingdom who are interested in developing cell therapies derived from these iPS cells. So we're going to distribute this universal cell lines to other collaborators who are interested in developing iPS-based cell therapies. So some collaborators might be interested in developing insulin-producing cells from these. So this collaboration is going to push forward this iPS-based therapy. The other arm of that project is to bank patient-derived iPS cells. So as you know, iPS cells can also be used for drug discovery. If we have a patient or a disease in which the tissue that is affected is inaccessible while the patient is still alive, what we can do is we can generate iPS cells from these patients and differentiate the lineage of interest and examine that tissue for difference and do drug screening. So this is basically the plan is to, because King Abdulaziz Medical City has around 2,500 beds capacity. So we generate iPS cells from these patients, and then these lines could be sold and distributed for pharmaceutical companies and academic universities to do drug screening and understand disease mechanisms. And this can also, the monetization of this could be really helpful. One project led by Dr. Dua at KMARC is to study a disease that is caused by SLC mutation. We're going to generate iPS cells from the patient and from the normal sibling, and then differentiate these to generate midbrain organoids that can later be used for drug screening and understanding disease mechanism. So I'd like to thank all the collaborators and KMARC, Dr. Askar, Dr. Majd Al-Fadl, Dr. Dunya Jodat. Thank you all very much.
Thank you so much, Dr. Khaled, for that really fascinating talk and introducing us to the opportunities iPS cells may bring in the future. Other questions for Dr. Khaled? If not, I would like to introduce our next speaker, Dr. Amin Ayitu. He is a professor and chair for targeted therapeutics at the Imperial College in London. He has completed his PhD at the University of Liège in Belgium, and during his PhD work, he acquired extensive experience in gene delivery technologies using eukaryotic viral vectors. He completed his postdoctoral training at the MD Anderson Cancer Center in Texas, where he gained experience in bacteriophage, guided gene delivery, and phage display technologies in vitro and in vivo. Importantly, he designed a novel hybrid phage vector for targeted gene therapy transfer. In 2008, Dr. Ayitu established his research team at the Imperial College in London, where he became a senior lecturer in 2013 and a professor and chair of targeted therapeutics in 2019. His research team has become a leading authority in phage-guided gene delivery technologies, focusing on optimizing and developing superior phage-derived vectors for nucleic acid delivery to human diseases, including DNA vaccines. His leadership in the field has resulted in various awards for his research, numerous high-profile publications, and more than 6 patent applications, which also have contributed to the creation of 2 startup companies in the US. He is also a scientific co-founder of a spin-off company at the MIT in the US. He has received a royal decoration in 2015 by His Majesty, the King of Morocco. I welcome an expert in the field of development of bacteria, viruses, bacteriophages, into a delivery technology for therapeutic genes and DNA vaccines against cancer and human diseases. The floor is yours.
Thank you very much for this nice introduction. And I would like to thank the organizers for inviting me to this wonderful conference. Can I have my first slide, please? Can I have my first slide, please? Yeah. I don't see it. Could you show the slide, please? I don't see it on the whole screen. Yeah, I think there's a problem with slide. I don't see the slide on the screen. Can you show it on the screen, please? Okay, yeah. It's a little issue. I don't see my slides on the screen. Yeah, we can get started. So today I will present what we have done over the last few years to develop these bacteria viruses as vectors for targeted gene therapy of cancer. Can I have the next slide? Yeah, so as you can see, gene therapy has been around for a long time. It's actually the delivery of therapeutic genes. Gene therapy was initially conceived to treat inherited diseases, but today more than 67% of gene therapy clinical trials are designed to treat cancer. Because of course, cancer is a major cause of mortality in the world. And glioma, which are deadly brain tumors, were actually the first disease to be treated in the clinic by gene therapy, but clinical trials failed because of the vectors used at the time. Actually, to deliver therapeutic genes, you need vectors. Can I have the next slide? And human viruses have been mostly used because they can enter human cells as part of their infection process. So lentiviral vectors, retroviruses have been used, adenoviruses have been used, herpes simplex viruses have been used, and adeno-associated viruses have been used to derive vectors. And as you know, the AAV, adeno-associated virus, has become the most popular virus to derive vectors for clinical trials. And the principle of making vectors is the same. What you need to do, you need to make these viruses replication deficient. It means you need to remove the native genes, like the case here of AAV, adeno-associated virus. You need to take out the rep, which is the gene for replication, and the cap, which is the gene for the capsid, and replace them with the mammalian transgene cassette, which is the CMV promoter, commonly used promoter to express the therapeutic gene and poly-A sequence. That's the principle of making vectors from viruses. Can I have the next slide, please? So, as you know, gene therapy has been around for a long time, but for many decades, the big question, why gene therapy hasn't delivered the expected outcomes in the clinic? Because gene therapy has faced many problems, but mainly systemic delivery. That's why the majority of clinical trials of gene therapy, they are injected actually locally, because there is a problem with systemic administration. Local administration can show proof of principle, but real clinical benefit can only happen with systemic administration. For instance, if 90% of cancer patients, they die because of metastasis, and metastatic cancer requires systemic administrations. You can't treat all these metastases with local injections. I'm showing here the example of this mouse that I treated a long time ago. This is systemic administration. It's an intravenous administration of an adenoviral vector carrying the luciferase gene. This is an imaging, so I injected the virus into the tail vein. This mouse had a tumor, Kaposi sarcoma. The virus could deliver the gene everywhere, but not in the tumors. You can see they get trapped by the liver, by the kidneys. These viruses have brought tropism. That's why there is a problem. I mean, that's why systemic administration, systemic gene therapy has had limited success in the clinic. And one solution is to target them, to target these viruses. But what you need to do first is to ablate the native tropism, which actually which has been a problem. It was not a success. That's one thing. And another problem is the repeated administrations, because to achieve a therapeutic response, you need to repeat the administrations. But these viruses are immunogenic, so they block the repeated administrations, which actually hinders the successful complete response in individuals. Of course, gene therapy has faced other problems like the packaging capacity, the cost, production to GMP standard, scaling up the production. Can I have the next slide, please? So now I'm going to try to show you what we have attempted to do to solve at least some of these problems. So what we did instead of using human viruses, we turned to other viruses, bacterial viruses, named bacteriophages or phages. If I ask you the question, before the discovery of antibiotics, how bacterial infections in humans were treated by these viruses, by bacteriophages? They do not infect human cells, they do not infect human tissues, so we can deliver them systemically, intravenously, without a problem, because they wouldn't get trapped in the liver and other tissues. They were discovered in 1895, and then later they found that this bacteriophage kill bacteria. That's why they used them before the discovery of antibiotics. They used them to treat bacterial infections. But then after the discovery of penicillin, they were discarded in the West, but some countries in East Europe continued to use them. But now in the West, their interest is growing back because of the antibiotic resistance. Can I have the next slide? For instance, in this paper in Nature Medicine in 2019, this was a clinical trial to save this 15-year-old cystic fibrosis patient. She had an infection with Mycobacterium, which is resistant to antibiotics, but then they treated her with a cocktail of bacteriophage, which actually saved her life and cleared the bacterial infection. And in 2006, the US Food and Drug Administration approved the use of bacteriophage as antibacterial food additives to prevent the growth of Listeria, Salmonella, and E. coli. The point I'm trying, next slide, the point I'm trying to make here is that these viruses are safe. If you want to come up with a new vector, new treatment, new gene therapy approach, you need to take into account two crucial considerations. First, safety. They are safe. They have been given to a human. Second, efficacy. That's why we decided to use them. We can deliver them systemically. But the big question, how can you make a bacteria virus enter into a human cells? What we did, next slide, we used the phage display technology, which won the Nobel Prize in Chemistry in 2018. The phage display technology consists on the display of ligands. You modify the capsid of phage. You just express ligand to make these phages bind to specific receptor into cancer, for instance. Then they become targeted. Next slide. So we used the filamentous bacteriophage here. It's just a virus with coat proteins covering the genome. There is the gray protein, the major coat protein, and you have two minor coat proteins on each side. Next. So you use the phage as first we want to, what we did, we used the phage display technology to display this ligand, RGD4C, because it binds to this receptor alpha-5 beta-3 integrin, which is expressed in cancer, but minimally present in the healthy tissues because we want to target cancer. That's for the entry. So the whole, as soon as the virus binds, it gets internalized, and then the genome of bacteriophage gets released, but it doesn't work in human cells. It only works in bacteria. So to express the therapeutic gene, what we did, we inserted within the genome of phage the genome of human virus, AAV, because both are single-stranded. genomes, so they are compatible to insert into each other. But the big challenge was whether we could produce this in bacteria. So we take the hybrid genome, you transform the bacteria, then we were able to package the hybrid genome of phage and AAV by the capsid of bacteriophage in bacteria. Next. Then if you take this, we name it AAV phage, if you add it to a panel of tumor cells, if you don't have the ligand RGD that allows entry, there is no GFP delivery. But if you display the RGD on the capsid, it gets inside the cells, it delivers AAV, expresses GFP. Next. The same thing in animals. If you take this phage, you inject intravenously into mice with tumors, what happens? It gets cleared. If you use a ligand that doesn't target cancer, it gets cleared. But if you use the RGD on the capsid, the RGD will guide the phage to these receptors, integrins, into the tumors, enter into cancer cells, deliver the AAV to express the luciferase gene. That's what we want to achieve with target. You see, there was no delivery to the healthy tissues. And another group used this technology to deliver the tumor necrosis factor alpha, TNF alpha for immunotherapy, cytokine gene therapy, because TNF alpha has strong anti-tumor activity, but it's toxic. It has to be targeted. So when we cloned into our phage, we delivered IV into mice, intravenously into mice with tumors, they were able to recover TNF alpha expression into the tumors, but no TNF alpha was detected in the healthy tissues, the brain, muscle, lung, kidney, and heart, and other normal tissues. And this is what you want to achieve with targeted. In chemotherapy, is the other way. You get 5% in the tumors, the rest is spread all over the healthy tissues. Next. So if you make sections of these tumors, what you see that actually you have delivery. This is GFP delivery to the blood vessels in red and the tumor cells because they express the receptor. If you replace GFP with TNF alpha, then you blew up the blood vessels and the tumor cells. So if you remove the RGD, nothing happens. So you really need target. You really need that ligand to guide the phage to these tumors. Next. What I would like to show you here is suicide gene therapy because it was the first one to be tested actually in the clinic, in clinical gene therapy against glioma. What you do, you deliver the thymidine kinase of the herpes virus, you give ganciclovir, it gets phosphorylated by TK, it destroys tumor cells. So that's what we did. So targeted suicide gene therapy with our virus completely stopped the growth of these tumors in mice as well as in rats. Next. So then we did, in collaboration with a group from the National Cancer Institute in the US, we did a safety study in larger animals. These are pet dogs with huge tumors. These are dying dogs, and the owners, they bring them to the NCI. So the vets, they did like phase 1, dose escalation, but we never reached an MTD, maximally tolerated dose, because phage is safe. We know phage is safe. And then the vets said, because these tumors are huge, we have to repeat the administrations. But are they safe? Then we repeated once a week administrations for months. They were safe. They were not toxic. And then the big question is that because phage is immunogenic, there are IgGs against phage capsid after 2 weeks. But do they block delivery? Next. Then we did a biodistribution because after 8 weeks, 8 injections, we could still see the phage in red in the tumors, but no phage was detected in the healthy tissues. It means these antibodies against the phage, they don't stop the phage. Unlike the human viruses, the first injections will hinder the efficacy of the successive injections. And this selective accumulation of the phage in the tumors of the dogs resulted in selective expression of TNF alpha into the tumors, not in the healthy tissues. So this technology is bringing TNF alpha back to the clinic. Next. Then we saved dogs. I'm showing the example of these dogs with a huge tumor. It was a fibrosarcoma. So we treated with, you have to repeat. We repeated for 2 months, once a week. So after 4 weeks, from 15 centimeter, the tumor shrinked to 8 centimeter. And after a total of 8 weeks, IV injections, the tumor was completely eradicated. Next. So in the next part of this talk, I will show you what we have done for vector development. That's the challenge, the problems that gene therapy has faced because people rush to the clinic. We didn't want to rush to the clinic. We wanted to improve that because phage has evolved to infect bacteria with no optimized strategies to deliver genes to human cells. For instance, in this publication, if you look at human viruses, they get internalized into the endosomes. but they escape from the endosomes to avoid getting into the lysosomes, to avoid degradation by the lysosomes. But bacteriophage doesn't have tools to do that. This EM shows that the bacteriophage is in the early endosomes, in the multivesicular bodies, but the majority of phage ends up in the lysosomes because it cannot escape. That's a major problem. We have to solve that. If you give chloroquine, which disrupts the endosomes, then the phage can escape and increase gene delivery. Next. So in this publication, we used direct devolution. Actually, what we did was how we can transfer the strategies developed by human virus to the phage capsid to make them escape from the endosomes. So if you look at this phage on the P3 minor code protein, the RGD, we displayed the RGD. But then what we did, we wanted to display peptides from human viruses, and that's why we inserted a recombinant major code protein on which we could display endosomal escape peptide, which allowed the phage to escape from the endosomes, as you can see, and increase gene delivery in vitro as well as in vivo. Next. So here I will show you what we have done to treat glioblastoma, because these are deadly brain tumors, the first to be treated in the clinic. The majority of patients, they die within, 99% of patients, they die within 3 years. So we wanted to treat glioblastoma because phage can cross the blood-brain barrier. It's a major challenge in treating brain tumors and brain disorders. Next. What we did here, we instead of using CMV promoter, we used the promoter that transcriptional targeted that is active in glioblastoma. It's actually associated with glioblastoma resistant to chemotherapy. So if you treat the cells with the phage, with this promoter, you give them low dose temozolomide, then you activate the therapeutic gene from this GRP78 promoter. Next. So in this publication, that went viral in the public, actually, we take human tumors from patients, we insert them with about instead of packaging AAV genome by the AAV capsid is to use the phage capsid. We have to do this in bacteria. So you take the phage, the AAV genome, you transform the bacteria, but you need to make it replicate. Then we inserted an origin of replication of phage, then the AAV starts to replicate in bacteria. And then we infect with the helper phage to provide the capsid. Then we manage to produce small particles with the phage capsid and AAV genome only. But the advantage is that we obtained a dramatic increase of gene delivery. Next. And you see, these are small particles actually. And then we use them, we carried out a comprehensive analysis of gene delivery to compare to the large particles. It's interesting when you reduce the size, when you treat like primary tumors in vitro from patients, from glioblastoma patients, the large particles have difficult to deliver, while the small particles could deliver to all these tumor cells and stem cells and tumor stem cell sphere in vitro. Next. Just to show you an application for CRISPR, because we reduced the size of these particles, we increased the packaging capacity, then we could fit in CRISPR actually, and to deliver Cas9 expression to the human lung carcinoma, up to 70% of the cells were positive for Cas9. And then we could knock down the expression of the mutant P53. This mutation is present in 50% of lung cancer. Next. Just I will finish now with immunotherapy. We used it for immunotherapy. Just to give you an example of interleukin 15, it's a cytokine, it's in the clinic. They give this protein to the patients. There is a response, but there are problems of short half-life, and renal clearance. So we decided to do this as gene therapy. So we inject mice with tumors. These are IV injections with increasing doses. You can see that all the doses generated the expression of interleukin-15 in the tumors, but not in the healthy tissues, because we overdosed the mice. We didn't have any safety issues as confirmed by the LDH, which remained its cytotoxicity biomarker, which remained intact in the serum. And this immunotherapy can be a powerful tool, actually. You can see that the tumor regressed as early as day 5 after treatment with a targeted vector compared to the control. Because we can repeat the administrations, we could then, after a few weeks, we could completely eradicate the tumors. 50% the animals. We saved 50% of the animals that were still alive even 1 year later. Do I still have a couple of minutes? I can finish with the vaccine. Just a couple of slides. Can I have next slide, please? Just to finish that, with this slide, the bacteriophage has been used as a vaccine delivery system because of the phage display technology. You can display antigens on the surface because the phage gets uptaken by the antigen-presenting cells, and because the phage can interact with MHC1, MHC2. And in this publication, we showed that the phage can cross the gastrointestinal tract. So phage can be given as oral vaccine. That's why we used it in cancer vaccine. Next, cancer vaccine consists of vaccine that can induce an immune response against tumor-associated antigens or tumor-specific antigens, but the tumor-associated antigens, they are also expressed in other tissues, and the tumor-specific antigens, they get down-regulated by the tumors. That's why we used a novel approach, next, in which we decided to deliver foreign antigens to the tumors that could be viral. The idea is just to inject the animals or patients with any vaccine. It could be COVID vaccine, flu vaccine, malaria vaccine, as long as this vaccine can induce an immune response against the tumor. So then if you take these animals, you establish tumors, they are vaccinated against, for instance, malaria. What you do, you take the phage, you inject IV to deliver the antigen into the tumors, then you induce an immune response against the tumor. If you take the splenocytes from the vaccinated animals, you can see the if you mix them with tumor cells, they don't kill them. But if you mix them with tumor cells and you give the phage with the antigen to tumor cells, you destroy them. Same thing happened in animals. The tumors, they grow in vaccinated animals. But if you give the phage to deliver the antigen to the tumors, then you induce an immune response against the tumors. Next. So conclusion, just since the dog study in 2009, we uncovered limitations of these particles. We designed strategy to improve them. Then we are ready for clinical trials now. We believe we have the vector to enter clinical trials. Next. If you look at other strategies used, the advantage of our strategies is that it's cost effective, it's targetable, we can repeat the administrations. We don't need cold chain transport. The phages are stable at room temperatures. So our phages can actually solve various problems that gene therapy has faced for many decades. Next. So as you mentioned earlier, so this has generated a couple of companies in 2013, the first generation of phage, which is going to clinical trials, and in 2021, another company with a group from the MIT. Next. And I would like to thank my group and all the students who have worked hard and the funders in order to advance this technology toward clinical trials and commercialization. Thank you very much.
Thank you so much for that truly fascinating talk and presenting us insights into something which really could be a game changer in the treatment of patients. In the interest of time, I suggest that I would start with my presentation. So my name is Miriam Fuchs. I'm a global therapeutic area lead at Novartis in regulatory affairs, cell and gene therapies. I'm a PhD by training. I worked on signaling at the Max Planck Institute in Munich, and I did a postdoctoral fellowship at the Dana-Farber Cancer Institute in Boston, again on signaling in cancer biology. And I joined the biotech and pharmaceutical industry approximately 20 years ago. And I spent the last years in a really outstanding team developing tisagenlecleucel for the benefit of patients. Let me just see how I work the slides. This is my disclaimer. Yeah, so our journey for tisagenlecleucel, also known as CTL019 or Tisacel, started in August 2012 when Novartis entered into a licensing and development agreement with the University of Pennsylvania to develop CAR-T therapies. At this time, the first patients were successfully treated with CTL019, including Emily Whitehead, who had been the first pediatric patients ever treated with a CAR-T cell therapy. Shortly after initiation, the collaboration with the University of Pennsylvania, Novartis purchased a cell manufacturing facility in New Jersey to enable manufacturing for global registration studies. During the course of the tisagenlecleucel development, we used the available tools for an early and close interaction with the regulators, like breakthrough designation with the US FDA and prime designation in Europe with the EMA. And these designations are unique opportunities for an increased dialogue with health authorities and are really helpful in defining the development and registration strategy for innovative products like cell and gene therapies. And we were the first company to run truly global pivotal studies with CAR-T therapies. In August 2017, Tisacel received approval from the FDA for an indication to treat pediatric and young adult patients with relapsed refractory B-cell ALL, and it was the first CAR therapy ever receiving regulatory approval by a health authority. In 2018, we were able to expand the use of Tisacel to include adult patients with relapsed refractory diffuse large B-cell lymphoma after 2 or more lines of therapy as a second indication. But equally important, we were able to gain regulatory approvals for both indications in Europe and many other regions, including Saudi Arabia, to enable patients globally to access CAR-T therapies. We recently announced that the ELARA study, which is investigating Tisacel in third and later line relapsed refractory follicular lymphoma patients, met its primary endpoint, and I will share some data on that indication. For me, the promise of CAR-T cell therapies is symbolized by Emily Whitehead, who had celebrated in May this year 9 years of cancer-free remission. I will skip this slide because we had a very eloquent introduction to the mechanism of action of CAR-T therapies. So the logistics of Tisacel is complex. We heard about it already, given the shipment of cryopreserved leukapheresis from the apheresis center to the manufacturing site, which often is outside of the country where the patient is treated, and then the delivery of the cryopreserved final product back to the clinical site. And of course, all steps are requiring a close control of chain of identity, which is absolutely crucial for an autologous product. The manufacturing of Tisacel itself is sophisticated, including transduction with a replication-deficient lentiviral vector to stably introduce the transgene encoding for the CAR. The manufacture itself lasts around 9 to 10 days, followed by a final product quality release testing, leading to an overall time for manufacture and release of around 22 days. At the clinical site, the patient is receiving lymphodepleting chemotherapy to enable expansion of the CAR-T therapy before administration of Tisacel. And CAR-T therapies are indeed living drugs, and they are supposed to be a one-time treatment. Sorry, I'm moving the slide. So, however, not only the manufacturing and the logistics around autologous CAR-T therapies are very different from what we face with small molecules and biologic. The interactions at the treatment center itself are equally complex, and a close collaboration between multiple stakeholders at the clinical centers, as visualized in this slide, is instrumental for the successful management of CAR T therapies. We were very dedicated to bring Tisacel to patients around the world, and we have now established a global reach with a presence in many regions, as visualized on this map, with access to Tisacel in 330 centers around the globe, and an intention to continue to expand the global availability. A global reach also requires a large manufacturing footprint to provide reliable capacity for manufacture of CAR T therapies for clinical trials as well as for commercial setting, and to enable a geographic reach also from a logistics perspective to facilitate the logistics. We have established 7 facilities around continents to manufacture CAR T therapies, either Novartis owned or working with contract manufacturing organizations. The clinical development of tisagenlecleucel was linked to important learnings on our side, and I would like to share some of the unique features related to the development of CAR T therapies, which to some extent can be extrapolated to advanced therapies development in general. From a clinical trial perspective, the impact of manufacturing an autologous product individually for every patient on the study conduct itself cannot be underestimated, including, for example, the timing between patient enrollment and the infusion of the therapy. Given the often progressive nature of the disease investigated, patients may need to be stabilized with bridging therapy while the product is manufactured. The execution of global multicenter studies requires dedicated onboarding and training of the clinical sites for the management of the CAR T therapies, but also for the local logistics. The logistics for a global study, especially if you have only one central manufacturing facility, need to be established, as well as a close control of chain of identity. And often limited manufacturing capacity is faced in early phases of the program, and that may pose a challenge, as well as adjustments to the manufacturing process, which may very well happen in the early phases of development. Also, development programs may be condensed, especially in later lines of indications when no effective standard of care is available. And many of these advanced therapies are actually developed for rare indications. Consequently, there's a unique role of the natural history data of a disease to contextualize the pivotal study results. And indisputably, academia plays an important role in driving the innovation in cell and gene therapy space. Many early cell therapy clinical trials are initiated by academic centers across the world, as these centers are actively engaged in investigating novel approaches and technologies. From a CMC perspective, however, it is critical to consider that these therapies may be requiring iterative development and optimizing for GMP production at commercial scale, especially when initially developed at these academic centers. In respect to autologous products, we have to consider variable patient material as a starting material. We may also face lot-to-lot variability of human and animal-derived materials used in manufacture. There is potentially also limited product knowledge in the early phase of clinical development, and all these elements may pose challenges in the development of a robust commercial process. We need to understand the product critical quality attributes, the critical performance parameters. We need to establish clinically relevant product specifications, develop robust analytical tools for release. All these are critical aspects in developing a suitable commercial manufacturing process and control strategy that ensures a consistent product quality. Also, establishing reliable comparability methodologies is a critical prerequisite in the context of process improvements, which may be introduced during a clinical study, as well as to expand the manufacturing process to new facilities. The initial development of tisagenlecleucel focused on indications in pediatric and young adult patients with relapsed refractory ALL and adult patients with relapsed refractory diffuse large B-cell, both indications with high unmet medical need. And at the time of the study initiation, there were no effective treatment alternatives available. So our pivotal program was based on single-arm studies. And the pivotal data allowed to conclude a positive benefit-risk profile on tisa-cel in the approved indications. However, we know the experience in real-world clinical practice is very relevant, and for CAR T therapies, this experience is documented in registries for CAR T therapies, like, for example, the registry from the Center of International Blood and Marrow Transplant Research, CIBMTR in the US, or the EBMT in Europe. And these organizations entertain registries which were originally developed to follow hematopoietic stem cell transplant patients, and they have been expanded to also follow CAR T patients after infusion. Importantly, as presented in this table, the experience in real-world clinical practice with tisa-cel is consistent with the pivotal trial experience in respect to efficacy, as shown by the overall response rates and the survival data. And the data also show that the safety of tisagenlecleucel is generally well manageable by physicians in the commercial setting. I will take a moment to speak about our experience with tisa-cel in adult patients with relapsed refractory follicular lymphoma, which presents the second most common form of non-Hodgkin's lymphoma after diffuse large B-cell and is the most common form of indolent non-Hodgkin's lymphoma. And follicular lymphoma is characterized by relapsing remitting phases and is considered incurable in advanced stages of the disease. Our pivotal single-arm study called ELARA investigated tisa-cel in patients who were refractory to 2 or more lines of systemic therapy, including a CD20 antibody and an alkylating agent. We have seen in a prior presentation the study schema for the early clinical studies, and it's similar for the follicular lymphoma study. It's a single-arm study with phases of screening, leukapheresis, cryopreservation. Patient is enrolled, potentially receives bridging chemotherapy while tisa-cel is manufactured. Prior to infusion, there is a lymphodepleting stage. Tisagenlecleucel is infused, and there are regular assessments to monitor response and safety. The primary study endpoint was met at the pre-planned interim analysis with a high complete response rate of 66% and an overall response rate of 86%, shown on the left table. And of note, the durability of responses presented in the right graph indicates a clinical benefit for many of the treated patients. And based on available data, we propose to conclude that the benefit-risk profile for tisa-cel is favorable for the treatment of patients with relapsed refractory follicular lymphoma after 2 or more lines of therapy. And we will seek regulatory approval for these indications to enable patient access. Innovation does, of course, not stop with the first cell and gene therapies receiving regulatory approval. The field is now facing rapid cycles of technical and scientific innovation to provide patients with novel treatment options and to achieve better outcomes for more patients in more indications. For the CAR T specifically, the field is innovating to achieve higher response rates, overcome the mechanism of resistance, and improve the persistence of responses, as well as to expand to new indications, including solid tumors, and we have heard about it in the prior talks today. Loss of CD19 antigen, for example, is well documented in pediatric ALL as a cause of relapse. We spoke about it. Other mechanisms of resistance are also present, such as CAR T-cell exhaustion, lack of persistence of CAR T-cells, or resistance of cancer cells to killing by CAR T-cells, even when the CAR T recognizes the antigen on the surface of the cancer cell. And on the left little figure, we are presenting some of the some of the tumor intrinsic mechanisms, and that's of course a field of intense research. Importantly, avenues to improve T-cell fitness may also improve the clinical outcomes, and to this extent, Novartis is developing a novel CAR-T manufacturing platform that will serve as the foundation for the next generation CAR-T therapies in our pipeline. And this novel manufacturing process results in a product in which T cells retain a less differentiated, more stem cell-like phenotype, which is thought to be an important T cell characteristic closely tied to the therapeutic potential of CAR T cells. And thus it is expected to lead to deep and durable responses and improved long-term outcomes. We can expect that some of the improvements in the CAR-T field we are seeing now may be incremental in respect to clinical benefit. However, of course, every improvement is meaningful for patients. And one of the challenges ahead of us is to define how we can best develop these innovations to assure timely patient access to these improved therapies. Going forward, a close collaboration between developers of advanced therapies, regulators, and HTA bodies, the payers, early in the development process will be instrumental in order to gain regulatory approvals and enable timely access for patients to innovation. So early engagement is critical to align on key aspects of the development program, like trial design, endpoints, patient population, but also to recognize long-term uncertainties and address them in the development program and post-authorization. Specifically, the use of real-world data, real-world evidence to contextualize pivotal data pre- and post-approval will be key in these discussions. And especially in the cell and gene therapy field, there is a unique opportunity, but also a need for the use of real-world data to contextualize the clinical trial data. I would like to conclude with a deep appreciation of where the field is now, but also to emphasize the importance of a close collaboration between all stakeholders, academia, biotech, pharma, physicians, regulators, payers, as well as patients, to ensure that the pioneering work in the field and the rapid innovation cycle we are currently seeing can reach and benefit patients in a timely manner. Thank you for your attention. Are there any questions? Otherwise, I would suggest that we move to the panel discussion. Okay, so thank you for the very, very interesting presentations we heard in that session. I really enjoyed it. I would want to pose a few questions, and I would like to start with the first one. We have seen that the recent development of CAR T-cell therapy has revolutionized the field of cellular therapies. How do you see the accessibility for CAR T therapies changing in the future? What is the economic impact of these CAR T-cell therapies and the expected impact over the next few years? And I would like to pose that question to Dr. Bader.
I'm a clinician by nature, and I'm not an economist. CAR T-cell has really revolutionized the way we treated patient and introduced a new idea in not doing an innovative way, but also an effective way of treating those patients. I mean, we are seeing more durable responses, and that can lead eventually to cure for some of those patients that used to be an incurable patient. In terms of economical impact, I mean, the cost of the medication of all this treatment considerably high when you compare it with other type of therapy. For example, and however, I mean, it is working. It's giving good result, but I'm not sure about the how this treatment will be available, readily available for patient, and how patient will have an appropriate access for this kind of therapy. In terms of economic impact, it has been estimated in US, for example, in 2026, the CAR T-cell therapy market will reach up to $1.7 billion for CAR T-cell that treated hematological malignancy. That's a good share of market. However, the expenditure is going to be a burden in the buyer. And for example, recently published data from Europe, for example, showing that in European countries, the expenditure will be increasing over the next few years, up to 2029, showing that cumulative impact on economic impact on those countries. For example, it has been estimated that countries such as Netherlands will expend around 1.4 billion euro, up to 1.6 something euro for a country such as Germany. That's only a direct price for the CAR T-cell product itself, including hematological malignancy, and that's including myeloma products as well. So that's a huge expenditure, and of course that will be equal to a huge economic impact.
Okay, thank you so much for that assessment. My next question is directed to Dr. Fraietta. CAR T-cell therapies are now one of the most promising and sought after cancer treatments. How do you see this area evolve over the coming 5-10 years? Off the shelf, allogeneic CARs, CAR NKs?
Yeah, that's a great question. So accessibility is key, I think, in the autologous space, even though we've demonstrated that we can scale up and scale out, right, and make commercial products. Too many patients, as we've heard today, are still unable to receive their autologous products because of, manufacturing failures, or the fact that, and again, I'm not a clinician, I'm a biologist, but this is what I've seen, or they're not stable enough, right, to receive their CAR T cells. So I think allogeneic products are really the future, because we can increase patient access and have, hopefully on-demand, products for on-demand treatment.
Thank you. Do you think that CAR NK cells, allogeneic CAR NK cells, would have a better chance than allogeneic CAR T cells?
Oh, that's a good one. That's a good one. I mean, I'm a T cell guy. And okay, so if we turn to solid tumors, perhaps, right? But solid tumors, if you just look at endogenous solid tumors, they're not really infiltrated by NK cells. So I think we're going to have to do a little more engineering to figure out how to get them where they need to be. And they're also susceptible to immunosuppression within the tumor microenvironment. You can manufacture them, but I think just like what you're doing at Novartis, we need manufacturing improvements as well for these cells. So for instance, I know if you like soak NK cells and IL-15, which I think everybody does to keep them surviving and to, manufacture them, they tend to be exhausted if you could get them to a solid tumor site. So I think there's going to be a lot of crosstalk with what we're doing in T cells. But yes, obviously, that could solve potentially, a lot of issues with patient access. I think it's going to come down to, do we have to keep re-administering these? How often are we going to have issues with adaptive resistance, like we're seeing with T cells across several indications, across several centers, if we have to hit hard and heavy and keep re-administering them? So, but they're a very attractive modality for sure.
Okay, thank you for that assessment. Stem cell therapy has raised a lot of hopes, yet clinical successes in this field are still limited. We have seen some interesting data today. Dr. Khaled, what are the challenges faced in the development of the stem cell therapies? Do you think they could ever become routine clinical practice?
I mean, that's the hope, right? So the major challenges are basically to have full grasp on cell fate and be able to differentiate these iPS cells or human embryonic stem cells to where we exactly want them to be. We want to generate functional cells from these cells that resemble exactly the primary cells that are found in the adult human body. So basically, is efficient and highly accurate differentiation of these cells. This is one. The second thing is scalability. I mean, with the usage of bioreactors, this have been improved. And then the other thing is the whether it's autologous or allogeneic. That's another issue. There are some countries where the genetic pool is very heterogeneous, and having these stem cell banks might not be the best option for them. But for places like South Korea or Japan, and also as I presented today in Saudi Arabia, it seems like a good direction to take. So we are now assessing the feasibility of this. And these are the major problems. Problems other than that is basically the teratoma formation was at once a big concern, but as we can see now that there are so many ways that have been developed to get rid of any contaminating pluripotent stem cells after we differentiate these cells to the tissue of interest. So that is no longer that much of a concern like it was before with all these developed techniques. So basically the cost and the full grasp and control of the cell fate of these cells is really key.
Okay, thank you so much. Similar to the stem cell therapy, gene therapy as well has still limited success cases. Dr. Aishitu, what are the common scientific and technical challenges the field is facing from your perspective?
Thank you for this very important question. Well, I can say that gene therapy is still suffering the mistakes and problems of the 90s. At the time, gene therapy caused death of patients. The best example is that of the child, J.C. Gilsinger, because at the time, scientists and clinicians rushed to the clinic to inject these vectors into patients without understanding how these vectors work, the immunogenicity, the repeated administrations, the safety, the production to gene standard. That's why gene therapy received a hit in the 90s. But I still understand because we faced that when we come up with these new particles, because instead of rushing to the clinic, we decided to improve them, to get them ready for clinical trials. But the other limitation is how to get funding. People can give you funding for something that can go to the clinic, preclinical studies, safety studies, but it's difficult to get funding for vector development. So now we have these particles, another challenge, how to produce them to GMP standard, because it's very expensive. We need millions. Then if you go through the academic track, the whole process takes time because you have to apply for funding, and that takes time. There is the other track going through spin-outs, which is faster. But I can tell you that will really spend a lot. It takes 1 year, and to get the production to GMP standard, for instance, now it has been approved in the UK and Europe to produce, for instance, phages at GMP standard, but it took many years to get there. So I believe the fast track would be through spin-outs and startup companies to get faster to the clinic. So if I look back, I think I wouldn't go through the academic track. I would go through the industry track to avoid all these years trying to publish these big papers in order to attract funding, all these high-impact journals, which actually I don't know how they measure the impact, actually. And they can say that, like PCR was never sent out for review by Nature, it was not sent out by nature, and you can see the impact of PCR. So that's the whole process of time, how to get there. It's one after the other. Yeah.
Thank you so much for these insights. And the interconnectivity between like vector systems and cell therapies, I expect has also put a lot of attention to vector capabilities and should foster attention going forward. I would like to open the panel to additional questions from the panelists. If there are no questions, I would like to close the session, and I would like to thank the organizers of this excellent Medical Biotech Summit for enabling this interesting session and the speakers for their presentations and valuable insights. We have heard quite a bit about the experience with CAR T-cell therapies, which is one of the early entrants of cell and gene therapies into the clinical practice. And we have also heard about the opportunities of scientific developments and innovative technologies in the field, including pluripotent, induced pluripotent stem cells and phage methodologies. While the administration of an investigational gene therapy to a patient had happened more than 30 years ago, the field is now in a rapid and dynamic phase of scientific revolution and evolution with continuous efforts to pioneer. The development requires close partnerships between many stakeholders globally to bring this innovation to patients, and there are unique needs and opportunities linked to these fields, which are also aligned with the Saudi Vision 2030. And with this, I would like to close the session. Thank you so much.
Thank you.
Thank you, Dr. Mariam, and thank you all for sharing with us your insights and views, focusing on the improved understanding of new advances in cell and gene therapies that offer the potential to transform medicine. Everything has been shared in this session is very promising, so thank you so much. And now, ladies and gentlemen, we're about to begin a 30-minute break. A quick reminder, you can follow the conversation of our sessions on Twitter at RGMBS2021 and also participate in the discussion by using the hashtag RGMBS2021. We'd love to hear from you, so share with us your feedback and views. We'll be back at 7:35 p.m. Riyadh time for the last session of the day, an exciting one, so stay connected. Thank you so much.
The pandemic has taught the world of the importance of science.
And our mission is to improve the health of our communities,
including a clear message about how the benefits will flow to the community more broadly.
Riyadh Global Summit for Medical Biotechnology aims to push the boundaries towards a targeted and sustainable medical biotechnology enterprise in the Kingdom of Saudi Arabia. Let's hear from the Summit Chairman and the Summit Vice Chairman.
Riyadh Global Medical Biotechnology Summit 2021 present truly a fascinating program as a blend of sessions dedicated to strategy development, business development, as well as other sessions dedicated to technology development, infrastructure development, to serve truly establishment of medical biotechnology ecosystem vibrant in the country, as well as other sessions dedicated to scientific topics such as genomics, genetics, gene editing, stem cells, and others. All of those are being delivered by top leaders in the field from global pharmaceutical company and biotechnology companies from around the globe, as well as leading academic institutions with the aim to forge global partnerships that are of interest and will impact the country and the rest of the world.
Welcome back, everyone. We have reached to the final session of the day. In this session, our esteemed panelists will discuss in depth genomics, genetics, gene editing, and bioinformatics. Very interesting topics. To start, it's my pleasure to welcome the chairman of the session, Dr. Paolo Martini, Senior Vice President of Moderna. Welcome, Dr. Paolo. It's great to have you with us at the opening of the last day and even the closing session of the day of the Riyadh Global Medical Biotechnology Summit. The mic to you.
Thank you so much. It's a pleasure, it's an honor for me to be part of this meeting. So thank you again for inviting me and to be part of this incredible endeavor that you guys put together. It's incredible. I'm chairing session 9 together with I would like to call my friend and colleague, Dr. Majid Al-Fadel. We know each other for a long time right now. And I'm so proud to be able to introduce him today in this session about genomic genetics, gene editing, and bioinformatics. He's going to give us a lecture on genetic and rare diseases programs, GARD, an opportunity for an orphan disease biotechnology. So I'll give it up to you, Majid. It's my great pleasure to introduce you. Thank you.
Thank you, Paolo, for this nice introduction, and thank you for the scientific committee and organizing committee for inviting me to present in this important summit. I will talk today about genetic and rare disease program, which we established in our research center in Kmark, and what is the objectives and what is the achievement so far, and what is the future plan. Rare genetic disorders are individually rare but collectively common. We are 10 times higher incidence of this disorder compared to USA and 4 times higher than Europe. Discovery in rare genetic disorder lead to discovery in complex genetic disorder. As you know, we have one body, so any discovery in genetic rare, any discovery in a single gene defect with this lead will open a horizon to discover in complex genetic disorder, and we have a lot of examples in the literature. For example, for obesity, congenital leptin deficiency is a single gene defect, while obesity is a complex genetic disorder. So the scientists now, they are working to find a treatment for obesity through this single gene defect. Another example is mitochondrial disorder. The only, or one of the barrier for establishment of genetic and rare disease program was the high cost regarding the medication and the treatment. Why we are care about genetic and rare disease program in Saudi Arabia, because we have a 3 sites in Kmark. It is distributed all over the kingdom. We have over 500 staff. We have around 50 scientists. And in Kmark, we have running 200 projects. And also we have one of the biggest biobank in the Middle East, which expected to collect 200 samples, and infrastructure is available. From the genetic point of view, we have 5 genetic consultants, 2 assistant consultants, and really well-equipped staff to do such program. On the other hand, one of the rationale, there is a huge expansion of the genetic disorder over the years. So this is from statistic. As you can see, actually this is 2 years ago. 2 years ago it was 4,274, and yesterday I checked statistic. We reached around 6,000 diseases, which is phenotype known and molecularly known. And this indicates that we discover around 1,200 new diseases into years. And as you know, we have around 30,000 genes discovered, and only we link to human around 6,000 so far. So still there is opportunity to discover more and more until we know the capacity of genetic disorders. Again, other rationale that now actually the phenotype become more related to the genotype. So you could see now 2 or 3 phenotype related to a single gene. Before only we have 1 phenotype and 1 genotype. Currently, you can see that the number of gene with 2 phenotype and 3 phenotype and 4 phenotype is increasing. So let's talk about rare genetic disorder in Saudi population. I will start with a newborn screening program, which we established in 2011 in National Guard Health Affairs. And this is part of the National Newborn Screening Program. And as you can see, it is in collaboration with King Salman for Research and Disability and King Faisal Specialty Hospital and Research Center and Ministry of Health. And actually, in this study, 139 hospitals were contributed to this study, and 775,000 newborn were screened. When we screened for 16 genetic disorders, most of them inborn error of metabolism and 2 endocrinopathy. And we found the incidence is 1 in 1,000. As you can see, the disorder, how it is distributed over the region. And we found the commonest is congenital hypothyroidism, followed by congenital adrenal hyperplasia, and propionic acidemia were the 3rd common in Saudi population through this prospective study or a newborn screening study. And we publish this. Also, the other thing indicating the high prevalence of Saudi inborn error of metabolism and genetic disorder in Saudi population. This study which we did is showing that the incidence around 1 in 600. This means that each 1 in 15 individual will carry 1 of genetic disorder. So this is very important to tell you how much these disorders are prevalent in our population. So again, I will emphasize on this comment, inborn error of metabolism or genetic disorder are individually rare, but collectively common. The program will be modeled for precision medicine mainly and personalized genomic. It has a clinical and basic component. It is strengthening the idea of translational research. So I will explain it easily. I saw the patient, or my colleague saw the patient in the clinic. It's an interesting case, non-diagnosed, not resolved. We will take it and adopt it until we find the diagnosis and the novel discovery on this patient. So it's helping the patient, and also it is cost effective. So it is to uncover disease mechanism, to develop a new diagnostic platform, to develop a new therapeutic approach, develop mechanism and strategy to prevent propagation of such disorders. And actually, it is a collaboration between KMARC and university and NGHA hospitals over the kingdom. And also now we're starting collecting cases from outside NGHA hospitals. Actually, this is a central piece of research and development, and it is a mega project in KMARC. All departments participate in this program, not only genomic department. And actually, once we collect the case, we do whole exome or whole genome sequencing, and then we go and do a segregation analysis for other family member, and then functional studies including proteomic, metabolomic, sometime quantitative PCR, Western blot analysis according to the discovered gene or variant. And then sometime we go further to animal model, and finally we are hoping that we reach therapeutic discovery. So as again, it is a strategical program from KMARC, from all the department together. We have diagnosis, then characterization with biomarkers, and prevention and therapeutic. How we did this program, we start with establishment of whole genome sequencing in our center, and it was really a huge challenge, but with collaboration of the team, we were able to do that. And what I mean by whole exome sequencing, as you know, the gene is formed from promoter and tail, intron, and exons and boundaries, and the most mutations are found in the exons. So we screen all the exons of the whole genome. While whole genome sequencing, it will sequence the whole genome of the human. So we published this, and we found the WIS rate in our population is around 50%, which is higher than Western population, which is around 25 to 30%, and this is because of the high rate of consanguinity. And again, we were one of the first group who published the hit rate of whole genome sequencing, and we found the hit rate of whole genome sequencing was higher by 10% than whole exome sequencing. And to establish this, it is really a collaboration between bioinformatics department in KMARC and IT, and also our genetic and genomic department. I should highlight here, you need high capacity of storage in order to do such a project. So when we did first 100 genome, our data center called us that we really reach a lot of storage of our server, and now we are working with others to able to expand the capacity of the storage. And then we did that, and we established our KMARC genome database, and through that database, it helped clinicians to find allele frequency of rare variant among Saudi population, and help researchers to investigate genomic variant role in combination with other clinical features for disease development and mechanism. And this database, it will be opportunity also in the future for artificial intelligence and other application for database, as you know. And you can see here we reach now around 2.56 million variant and around 1,500 individual and families. And also we published this recently as the secondary finding in Saudi population. We found that around 8%, which is higher than any other population. So what does this mean? This means when you do whole exome and whole genome sequencing, you will see other diseases other than what related to the presentation or phenotype. The rate of this in Saudi population is 8%. We, through this program, we discover more than 50 novel genes and diseases. As you can see, this is with international collaboration. We discover 2 genes responsible about hyperammonemia. And through collaboration with Professor Al-Quraya and King Faisal Hospital, we discover 33 novel genes in this. And also this disease, we discover it, which also present with a neuroregression, developmental delay, and lactic acidosis. And you can see vanishing white matter in the brain. This disease, after we discover it, as you can see, we make another publication to detail the phenotype, and this is what we did. Once we discover a novel disease or novel gene, we start collecting cases in order to understand that disease, and actually we published 10 cases from our population and become the international reference for all articles published with this disease. And this is, we call it SCA2 gene defect. Because of the time, I will not go in that, details. But 24 patients has been described so far, and we are the leader of this disease over the world currently. As you can see here, the interesting thing about this disease, the glycine level in the blood is increased and also in the brain. So usually they have an effect in the glycine cleavage, because they have problem with the lipoic acid biosynthesis defect in the sulfur mitochondrial pathway. Again, we were the second group who discovered this disease, which is asparagine synthetase deficiency. And after that, this disease is present with microcephaly, developmental delay, and low asparagine level in blood and CSF. As you can see, the interesting thing about rare diseases, you can see a clear pathway. And this clear pathway would help you to develop the therapeutic approach easily than obesity, cancer, and other kind of diseases like infectious diseases. And this is our publication about this disease. Again, we become reference over the world for this disease. This novel disease also we discover, it's called SLC6A9 defect, which is a GLIT1 encephalopathy. So this is a problem in the transporter of glycine in the brain, and we were the first group who described this disease. They present with high glycine in the blood and in the CSF, but it is different than non-ketotic hyperglycemia, the classical one. Again, from the clinical genetic point of view, we discovered this disease actually 2 years ago, which is present with global developmental delay, intellectual disability, and speech delay. It's called AMC10 gene defect, and we confirm our finding in KMARC with quantitative PCR and Western blot analysis, as you can see in the picture. And we were the first group to publish this disease. Again, RAP1 GDS syndrome, actually also we are the first group who published this syndrome, where they present with dysmorphic feature, intellectual disability, and speech delay. After our publication, this is the quantitative PCR picture. After our publication, the King Faisal also published this as a part of other genes. And also in Europe, they published 6 novel genes from the Syntogene group. Again, we were the second who discovered this disease, which has a problem in the glycose aminoglycan biosynthesis. And this patient present with a global developmental delay, axial hypotonia, and a problem in the glycose aminoglycan biosynthesis. And also with Dr. Baha Rifai, he's here also hearing us, we discovered this disease, which is a neuromuscular abnormality, infantile spasm, intellectual disability, developmental delay, hypotonia, spastic quadriplegia, and delayed myelination. And interestingly, in this syndrome, we use CRISPR editing in order to prove the pathogenicity. Finally, KIF16B, it's also, we discovered this disease will present with seizure and developmental delay and hypotonia. So in conclusion, genetic and rare diseases are common in Saudi population. The incidence is 4 times higher than Europe and 10 times higher than USA. This is an opportunity for biotechnology companies to invest on these diseases. Kingdom of Saudi Arabia is one of suitable countries to run a clinical trial for rare diseases because we have high number of patients. Logistics should be facilitated in order to shine more in this area. Again, whole exome and whole genome sequencing allow for detection of more novel genes linked to human diseases. The rate of consanguinity in our population is really high, which is 60%, and this really need to more of ultra-rare novel autosomal recessive disorder. We need more larger cohort and follow these diseases clinically with international collaboration in order to reach the detailed phenotyping and diagnose more cases in the future and find therapeutic area. Again, collective and collaborative leadership among scientists and clinicians around the world is essential for gaining better understanding of new genes, pathophysiology and how it relates to human disease, and then moving to identify biomarker with them, and finally gene therapy. Again, this is not one-man show. This is done with a huge team of 4. I would like to thank my lab scientists and the clinicians, Dr. Badr Mzeni, Dr. Yusra Al-Yafi'i, Dr. Abira Tuwajri, Dr. Yazid Al-Hidan, Dr. Muhammad Umair, and our technologists, and also the clinical genetic team, our genetic consultant and coordinator and counselors, and metabolic dietitian, and finally our pathology department led by Dr. Ahmed Al-Faris and his colleague. And thank you very much.
Overview and incredible work in really identifying these diseases. Am I allowed for questions for Majid? Can you hear me?
Yes, you can. We can hear you.
Okay. I just wanted to ask you, Majid, you showed this incredible data and in identifying rare genetic disorder and obviously all these different mutations. I'm assuming that there are many founder mutations as well. How homogeneous are these diseases in terms of symptomatology and severity? And the reason I'm asking is you've done an incredible work in characterizing all of these, and it seems to me that the kingdom will be a perfect site for many of the clinical trials, mainly because of the characterization that you have in place for these diseases and the ability really to understand the severity of it. So what are you, how homogeneous are, and what are you doing in terms of collaborating more with the companies that have potentially the ability to bring a new medicine for these type of diseases?
Yeah, thank you very much, Paulo, for this excellent question. I think these diseases are mainly neurogenetic disorder with global developmental delay, hypotonia, and some of them are dysmorphic features. Many of them has a biochemical defect. So, for example, asparagine synthetase deficiency has a problem in asparagine synthesis. As we can see, UGDH has a problem in glycosaminoglycan pathway. So I think focusing on that biochemical markers in order to open the horizon for discovery of therapeutic area is really our next step in the future. The companies, as you know, we are not yet reaching the big company in US and in Europe, but we hope that with this work they will recognize us and come to Kaymark. And this conference actually is one of the way to communicate with the companies and develop our local biotechnology park.
Good. Thank you. Thank you, Majid. Are there any other questions for Dr. Fadel? All right. So should we move to the next speaker, which I think is me? I think, Majid, we did the introduction this morning, so why don't I just go into the, is that okay?
Yes.
Perfect. Sorry, I cannot change the slide because, as you can imagine, the Moderna computer are super protected, as we had a lot of attempt recently of hacking. We are becoming probably a little too famous in the world, and that's it becomes a problem sometimes. So I would like to talk to you today about a little bit. I already gave you an overview on the messenger RNA and why we are making the messenger RNA specific for basically rare genetic disorder. So I'll give you a brief overview on that, and then I would like to introduce you a little bit on the work that we have done on propionic acidemia, which is now currently a clinical trial happening in the United States and and eventually all over the world, and in the UK as well. So if we go to the next slide. Next one, please. So I'll go briefly over the messenger RNA, mainly because one of the things that I want to reiterate is that the messenger RNA that we make for rare diseases, which is use a systemic delivery, is very different for the messenger RNA that generally is used for infectious diseases or for other indication. We want our messenger RNA delivered in a systemic manner so it can reach different organs. Unfortunately, the dosing has to be chronic. As we know, unfortunately, messenger RNA is not very stable in time. So we have to do a chronic dosing. That doesn't take away, though, that a chronic dosing, if it's safe and well tolerated, can be certainly happening in patients. And one of the beauty also of the messenger RNA is that it's dose responsive, so that we can really manage the amount of messenger RNA and the expected response from the protein or the enzymatic activity that we expect, so that we can actually be a little bit more precise in terms of dosing and regimen, and also gives an advantage compared to gene therapy in the fact that we can, for example, combine many different messenger RNA at the same time in a single lipid nanoparticle without the limitation of being in a vector or constrained by a vector. And also, then in case of safety, if you have some safety issue, you can certainly stop the therapy and the messenger RNA will basically degrade very fast, as well as the lipid nanoparticle that we have, they're hydrolyzable, so they will disappear in between 8 hours and 24 hours without leaving any residues inside the body. One of the things, obviously, that we've been working on is the immune activation. We know that messenger RNA generally is recognized as a foreign nucleic acid, and therefore is recognized as a virus. But we actually unraveled why the mechanism or recognition of the messenger RNA is triggering an immune response, and we've been able to block that response. So we go to the next slide. And so we have been working quite a bit on messenger RNA in terms of understanding the structure of the messenger RNA, in understanding the stability and the longevity of the messenger RNA, in understanding the immunogenicity of the messenger RNA, and we actually built an incredible algorithm that they take in consideration all these aspects, even when we change the chemistry of the nucleosides, in rebuilding a secondary structure that seems to increment the affinity to ribosome to have a perfect translation. And we can make the messenger RNA also tissue-specific by using microRNAs as a way to dampen the translation of the messenger RNA in certain tissues. Lipid nanoparticle is the vehicle that we use to deliver the messenger RNA. So the lipid nanoparticle that we use, for example, in rare genetic disorder, once again, is very different for the one that was used in the COVID-19 vaccine, mainly because we wanted to be sure that we didn't mount an immune response to the lipid nanoparticle, and more importantly, that the lipid nanoparticle have the ability then to deliver to the cells. And we'll go a little bit in details on the composition of the lipid nanoparticle. And obviously, we do manufacture our messenger RNA, as we know, as we were the first one to basically come with the concept. So we wanted to develop a manufacturing facility that would support not only clinical trial, but eventually a marketed product in the future. And we have a state-of-the-art facility south of Boston, which is only 20 miles from Boston, but that produces right now part of the vaccine and many of the messenger RNA that are in the clinical trial currently. Next slide, please. So for obviously rare genetic disorder, we use IV injection, while for other obviously therapeutic areas we do either intramuscular, intratumoral, or intracardial, and that's the intracardial injection is actually a collaboration with AstraZeneca, where they're looking on restoring vascularization in ischemic tissues as a result of myocardial infarction, and they have produced incredible data in their phase 2 trial. So we are focusing on IV injection mainly because in rare genetic disorder we know that we can do IV infusion, and more importantly we need to target liver and many other organs in certain instances, but with the majority of the liver being the recipient of our lipid nanoparticle. Go to the next slide. And so the lipid nanoparticle obviously are sort of standard, if you will. The only things that we do have differently to the currently marketed lipid nanoparticle is that we have our own proprietary aminolipids. And our proprietary aminolipids are also ionizable, meaning that they have the ability to basically disrupt endosome because of their charges, and also they're hydrolyzable, that they're very fast degraded once they hit the cytosol of a cells when they are internalized as an endosome. So it's a very, we utilize basically the cells and the mechanism and the cytosol to basically degrade our lipid nanoparticles so we don't have any accumulation in the liver of the animals or now in the patients. We use sterols, cholesterols, generally phospholipids, and we also have PEG lipids. And once again, the PEG lipids are proprietary and they've been changed accordingly, and they're also hydrolyzable. And knowing that we have IgG antibody against PEG, having hydrolyzable PEG that they generally are decorated in the lipid nanoparticle, the moment that the lipid nanoparticle are injected into the bloodstream, and the blood being aqueous by nature, the PEG lipids are shedded completely from the lipid nanoparticle, leaving basically a circulating lipid nanoparticle naked from the PEG, so there's no interference from the IgG eventually recognizing the PEG and allowing the lipid nanoparticle eventually to opsonize with ApoE and being taken up by the LDL receptor and eventually transduced into the cells. So we get to the next slide, please. So once the lipid nanoparticle release the messenger RNA into the cells, obviously we have a few issues to take care of. One is to be sure that we have enough endosomal escape, that we have enough ribosomal engagement, so that the messenger RNA can translate any type of protein that we can think. We can do secreted, transmembrane, intracellular, intramitochondrial. We really can do it all. And I'll give you an example of propionic acidemia, where we actually deliver 2 different messenger RNA for encoding the 2 major subunits for the PA. So we go to the next slide. And this is just to show you how efficient are our lipid nanoparticle in releasing a messenger RNA into the cells. If you see the red dots in the left panel, those are still lipid nanoparticle encapsulating messenger RNA that haven't been able to release messenger RNA. While if you look at the Moderna LNPs, mostly like 99% of the lipid nanoparticle really release the messenger RNA into the cytosol. And so we have a 25 times better endosomal escape and about 2.5 times a better translational engagement, mainly because when we do codon optimization of our messenger RNA, we look at the secondary structure and we try to build the secondary structure of the messenger RNA that have a very high affinity for ribosome, basically guaranteeing that we have a much better translation compared to the endogenous one. So if you think of, for example, of treating an autosomal dominant disorder, we know that our messenger RNA is going to be translated much better than the endogenous one, and therefore, even if you have a disease with a dominant negative, you can certainly overcome the amount of the dominant negative versus the amount, obviously, of the active messenger RNA and eventually protein. Next slide, please. And so we also, as I mentioned initially, we have modified the messenger RNA to avoid the immune system. Messenger RNA generally is recognized as a virus, and so as the ability through the uridine to bind to the toll-like receptor 7 and 8 when they dimerize, as well as to create a second double-stranded RNA once internalizing the cells, and therefore being recognized also by the Rigai complex. And so by changing the uridine, if you go into the next slide, we are capable basically to block completely the immune reactivity of the immune system to the RNA, but just using either pseudouridine or methoxyuridine or methyl pseudouridine. They really have the ability to create a steric hindrance to the toll-like receptors so that the uridine doesn't bind anymore to this complex. It does create a much less double-stranded RNA because the pseudouridine is modified. Therefore, we are able to deliver an immune silent and a very highly efficient translatable messenger RNA into the cells. And the messenger RNA that we make is actually the typical messenger RNA you would find in humans, so as a 5' cap, a 5' UTR, a coding sequence, and 3' UTRs, as well as a poly-A tail. So we synthesize the whole things to be sure that it gets translated perfectly by the ribosome. Next slide. And so we see here how basically utilizing our messenger RNA compared to the legacy process, which uses normal uridine, which is in the gray bar. In the blue and green bar, where we use pseudouridine, we completely dumped the activation of B cells in mice, suggesting that our messenger RNA this time is not recognized as a virus anymore. On top of it, obviously, we have a purification process that has allowed us to purify the messenger RNA to a great level, and also that part seems to contribute to a less immune reactivity to the messenger RNA itself, as seen in the gray bar, in the green bar, sorry. Next slide, please. So the last things, obviously, we have all these features for the messenger RNA. We have 5' UTR, which is guaranteed translation initiation fidelity. We can calculate protein per unit time. We have now incredible mechanism of understanding ribosome and codon sitting on the messenger RNA and timing for it in order to create a perfect wild type protein structure. And we can calculate obviously the half-life. And also we can start to make the messenger RNA cell type specific by introducing in the 3' UTR microRNA recognition sites. For example, if we want the spleen not to translate the messenger RNA, we look at the abundance and the unique microRNA that are in the spleen, and we identify the microRNA recognition site that they're introducing the 3' UTR of the messenger RNA. So the moment that the messenger RNA is transduced into the spleen cells, because of the abundance of a certain microRNA that recognize those particular recognition sites, take the messenger RNA into degradation pathway without the translation of the messenger RNA. So we can make, for example, a liver-specific messenger RNA, avoiding a spleen, kidney, and any other tissues translation because of the microRNA utilization. So as you can see, we always use a very natural processes of making the RNA suitable for human and more importantly, stable in time, and elimination of lipid nanoparticle or endosomal escape by using very simple mechanism of hydrolysis or in these cases, utilization of microRNA for cell specificity. So we don't do anything that is chemically modifying the physiology of the cells. We just use the body to basically make the RNA the way we want it, which I think is an incredible way of thinking about this therapeutic. Next slide, please. So as I said, we actually have engineered LNPs and optimized for the different route of administration. In particular, for rare genetic disorder, they are very much immune silent. And also we have the ability to shed the PEG that potentially could react with the local IgG antibody generated by any type of PEG that we use on a daily basis in creams, toothpaste, etc., and having a perfect delivery into the target organ. We also obviously modify the messenger RNA to avoid the immune system by incorporating modified uridine and eliminating obviously double-stranded RNAs. These allow us to be a little bit more precise in delivery and more importantly in dosing and regimen, because at that point we know exactly that the messenger RNA will not be destroyed or immune compromised, but will be delivered at a certain amount that will give a certain amount of protein. And obviously, we also codon optimize the messenger RNAs and UTRs in order to favor translatability, which allow us to really treat every type of disease in autosomal dominant disorder, because the way our messenger RNA is recognized as much higher affinity compared to the endogenous one. And we can make a cell type specific, as I just mentioned to you, using the advantage of utilizing microRNA. So in the next slide, given that we actually have the ability to create this messenger RNA that they're immune silent and particularly well translatable, I want to give you an example of the propionic acidemia, which is now mRNA 3927, which is currently in the clinic, and we just terminated a first cohort, and we got the approval from FDA to move to cohort 2 because of safety and tolerability. And so we are very happy with that. And we are starting to see great signs of the messenger RNA therapy in rare diseases as well. We go to the next slide. I know that Majid or Dr. Al-Fadel just spoke about propionic acidemia as being one of the high prevalent diseases in the kingdom. That's why I wanted to talk to you about what we are doing in particular. And we also have a methylmalonic acidemia program, which is just started in the UK in the clinic. And I know that these are highly prevalent diseases in the kingdom. That's why I wanted to give you an overview on what we are doing based on the animal studies and how we got to the clinic. So in the case of propionic acidemia, there are two major subunits that are involved in terms of mutation. There's PCCA and PCCB, and there are mutations in both subunits. So in theory, if you use a single messenger RNA, what happens is that you might correct only one defect in one subunit. What we actually thought about it, since these two subunits come together to create an important complex to process propionyl-CoA into methylmalonyl-CoA and eventually to be part of the Krebs cycle, was actually to create a drug that would contain both messenger RNA for the PCCA and PCCB, so that we don't discriminate anymore of the different type of mutations for these patients. So this is an advantage of the messenger RNA because we can combine as many messenger RNA as we want and to tailor the therapy based on the type of mutations that you have. And if you have more subunits mutated, we don't need to discriminate anymore for the single patients with a single mutation, but we can treat every patient with that disease because we are basically reconstituting the whole complex without having to focus exclusively on one mutations or the other. So if you go to the next slide, obviously a little bit of an overview on the propionic acidemia is an organic acidemia. As I mentioned, it's a defect in the propionyl carboxylase alpha and beta, and when the two comes together, they create this hexadodecamer complex that is very stable. But once you have a mutation either in the alpha or in the beta subunits, this complex basically doesn't form in many instances, or if you have only one subunit, there's the other subunit that is missing, and both subunits start to actually disappear. And so that's why we decided to combine two different messenger RNA that would allow us not to deal with potential lack of an endogenous level of the counterpart subunits that was not mutated. So it's a pediatric disease, mainly because these patients have difficulties in passing the 6 years of age due to metabolic decompensation, frequent metabolic compensation. And right now, the only therapy that is approved besides the diet, which is very brutal for these patients, because it has to be deprived of many protein and amino acid. So the only care for the moment is liver transplantation and diet that not necessarily resolve the issues of this disease. And so in the next slide, please. So what we have done, basically, we, in a healthy, obviously, propionic acidemia patient, the hexadodecamer, the two subunits will come together, will create this big complex, and process the propionyl-CoA into methylmalonyl-CoA. When we have a patient that has a mutation in one subunit or the others, basically propionyl-CoA cannot be processed to methylmalonyl-CoA, and it starts to produce metabolites like hydroxypropionate, 2-methyl citrate, and ammonia, which are very toxic for the body. So what we have done, next slide, please. we actually have combined the two messenger RNA, and we wanted to test the ratio of the two messenger RNA subunits in order to understand what was the best input of one subunit versus the other, or if we needed both subunits at the same level. As you can see in the first panel, we are just testing a single subunit in a fibroblast from PCCA patients or PCCB patients. And we see that once we replace with both, we actually get a greater activity rather than the single subunit, which is the lower light blue bar, while the big blue bar is the combination of the two. If we look actually at the ratio of the two subunits that we need to give to cells, either in PCCA patients or in PCCB patients, we see that basically 1-to-1 ratio is the one that works the best. And so these actually gave us the opportunity to then start to look at those dependencies increase in PCCA activity, and we see that obviously we see a dose-dependent increase in activity once we give the two subunits in 1-to-1 ratio in a dose-dependent manner. And that's why we decided to make this combination as a 1-to-1 ratio of a messenger RNA encoding for PCCA and encoding for PCCB as well. If we go to the next slide. What we obviously look always is the localization, and in this case, the mitochondria is the organelle where the PC complex has the activity, and so you can see in the last right panel that's the co-localization of the PCCA subunit and PCCB subunit within the mitochondria, merging with obviously the staining for the mitochondria, and so therefore. We decided that we could move into animal and do some experiments in animal to see the efficacy of the combination of the two messenger RNA. Next slide, please. We did studies, and this actually was an inspiration in my conversation with Dr. Majid Al-Fadel when they started the carbaglue studies in human, suggesting that there was at least an impact in dropping the level of ammonia in these patients. And so we did the same in mice using carbaglue or carglumic acid as a single treatment. And also we wanted to use our messenger RNA to see how comparable in dropping the level of plasma ammonia the messenger RNA was to carglumic acid. As you can see, both the blue bar and the orange bar, where orange is carglumic acid and blue is the messenger RNA combination, those have the ability to drop the level of ammonia similar to the hypomorphic mouse, the heterozygous, which is actually non-affected. And so you can see that the messenger RNA has the ability to drop the level of ammonia equally to carglumic acid. The difference, though, is that with the messenger RNA, not only we drop the level of ammonia, but we were able to see actually dropping level of 2-methyl citrate, C3C2 propionate ratio, and a 3-hydroxypropionate, both in plasma and in liver, which actually is a great features to have because we are not only dropping the level of ammonia, but we are also dropping all the metabolites that seems to have an impact on the disease. So we go to the next slide. We also wanted to do a multi-dose studies to show that the consistency in repeat dosing. And so you can see that we went up to 12 weeks, dosing every 3 weeks. And every time we dose in a dose-dependent manner, we actually are able to drop the level of a plasma biomarker. In this case is on the far left is plasma 2-methyl citrate. In the center is 3-hydroxypropionate. And then on the right is the C3C2 ratio. And you can see that we drop the level and then they come back. And then when we retreat, the level drops again. And more importantly, even after the 5th dose, we still see this drop, suggesting that, in fact, we don't have anti-drug antibody formations in this case, and we can continue dosing without any immunogenicity issues. The things that I think is important to note that once you express protein in the liver, I think the liver start to work towards the tolerization, and there's been proven in many different papers now, and even in some of the gene therapy studies, by targeting the liver, even in a knockout model, you start to tolerize for that particular protein, and this is why we can do multiple dosing without seeing actually anti-drug antibodies dampening the effect on the enzymatic activity of the PC complex, which I think is a great feature of that. Next slide, please. So obviously, not only we look at multi-dose studies, we're obviously looking for a consistent response, particularly in 2-methylcitrate. But what we notice also that the mouse model that we use has the ability to develop in time a little bit of a cardiomyopathy. And we saw actually that with the treatment, after the 5th dose, we started to see a reduction in the heart rate as well, suggesting that probably we did have an impact on cardiomyopathy as well in time. So this was actually great news. And then when we went to FDA to file an IND, they were very pleased with the data, and they were very pleased with our toxicology package with our lipid nanoparticle. And so they gave us the okay to enter a phase 1/2 trial, which is a multiple ascending dose directly into patients, which is unprecedented. And they gave us the opportunity to start the first cohort between 6 years of age and above. And then the second cohort, we can open up to 1 year of age and above. As they understand the safety of the system of this platform, they gave us the opportunity really to treat very young patients from the very beginning. So this is good news. As I mentioned, we just completed the first cohort of these phase 1/2 studies, and we got the okay to proceed with cohort number 2 because of safety and tolerability. And so now we are starting recruiting for cohort 2 for propionic acidemia patients. And we just started also the clinical trial for methylmalonic acidemia, which is actually all the preclinical data are published, and you can see them online at this point. So in the last slides, I think, I just wanted to conclude saying that we have successfully identified a lead candidate, which is a combination of the 2 messenger RNA encoding for PCC and PCCB. The duration of effect is about 21 days in mice, and we have a significant improvement in ammonia level comparable to carglumic acid, in serum biomarker, and more importantly in heart rate. And we know that these patients generally develop cardiomyopathy, and that could be also one of the causes of death. And more importantly, we retain the therapeutic efficacy throughout repeated dosing, suggesting that probably we do tolerize by expressing in the liver the protein, and we don't see today the anti-drug antibodies. So this was great news. So with this, I want to thank all the people at Moderna. This is just a fraction of them. This was a picture taken before COVID, and now if you look at the picture post-COVID, that room is not enough anymore to contain all the people, because the growth for COVID has been absolutely important in order to provide the vaccine to all over the world, but also to concentrate now on this rare genetic disorder that seems to have, a messenger RNA seems to have an impact as well. So keep your finger crossed, and potentially we'll have a therapy for propionic and methylmalonic acidemia soon. Thank you very much for your attention, and I'll be happy to answer any question. Okay, if there are no questions, okay, we can actually go to the next presentation. The next presentation is given by Dr. Hisham Feniri from Northeastern University, and is going to talk to us about magnetically guided rosette nanotubules siRNA delivered for cancer gene silencing. Dr. Feniri, please.
Dr. Martini, thank you for your introduction. If I can have the first slide. I don't know if my clicker is working. Is it on? Good. Okay, good. So on this first slide, what you see is a set of molecules that are entirely synthetic. There is no equivalent to these molecules in nature. And they have the ability to self-assemble spontaneously into superstructures that I will show you in the next slides. So on this slide, you see some of the applications that we have explored with these materials. In the center of the slide are 5 nanotubular structures that result from the spontaneous self-assembly of this synthetic moss. And around those structures, we have a variety of applications, such as drug encapsulation and delivery. We use them for tissue engineering and screening for medical devices and for a compatibilization of implant devices with human tissue. We have also functionalized them with a variety of nanoparticles, such as gold, platinum, palladium, copper, silver. And we have used some of those nanoparticles and nanotube hybrids as antimicrobials, and we have shown that they have very effective in antibiotic-resistant bacteria. We have also used them to deliver microRNAs to reverse inflammation in patients, not patients, but these are actually models of patients suffering from atherosclerosis. And we have also functionalized them with certain peptides to deliver them on the right panel, bottom right panel, to deliver them to human lung adenocarcinoma cells and induce programmed cell death or apoptosis. So these molecules are shown here. They are inspired from the natural molecules, so we're deviating dramatically from the particles that were described by Dr. Martini for delivery of messenger RNA and small interference RNA. These are a different architecture and different structures. They are derived from nature, but they're not natural. So we're combining a guanine and cytosine into a new base called the GC base that we can synthetically modify into a variety of derivatives. And on the next slide, what you will see is this structure has the ability to self-assemble into a super macrocycle that you see here. It's a ring of 6 molecules maintained by 18 hydrogen bonds with nanometric dimensions. And then this structure spontaneously self-organizes into a tubular structure. We can control the dimensions of this system, length, diameter, functionality on the surface, functionality inside the nanotube, and stability of the system, the thermal as well as the physical and chemical stability of the system. So I'm not going to get into the details, but just to show you what they look like, this is an electromicrograph that shows the self-assembled nanotubular structure. In this case, they're extremely long. We haven't controlled the length in this case. We want them actually to grow as long as possible. And this is just a close-up view of those structures. They are 3.5 nanometers in diameter. And so they express a certain number of chemicals on their surface, and we have complete control on the nature of those chemicals on their surface. So on the next slide, you see the 5 families of nanotubes that I mentioned in the second slide. And they are, as you can see here, this is the GC, this is the twin-base GC, this is the tricyclic equivalent of the first one. It's what we call XK1. This is one that is functionalized with a group that points towards the interior or the lumen of the channel, therefore modifying the physical and chemical properties of the nanotube, and therefore the ability to capture molecules that are not compatible with physiological media and can deliver them, as I will show you in the next few slides. And the one on the right, YK1, is the largest one. We made that one actually for a different type of application in molecular electronics. Okay, on this slide, what you see is an encapsulation of a molecule inside these nanotubes. And those plots that you see next to the nanotubes, those are the potassium mean force, which is basically the association, the energy of association of this molecule inside the channel. And you can see as this channel as these peaks become higher, it means that the molecule have more and more difficulties to move along the main axis of the channel. And so therefore, by changing the nature of a nanotube, we can control the height of these peaks and therefore control the rate and the rate of release and the stability of the molecules inside those channels. So we have looked at the drug data bank, and the drug bank against these 4 nanotubes, 7,000 drugs or so, and we have a very beautiful scattering between 750, actually between 500 and 1,000, showing that there is some selectivity for certain molecules. It is more obvious on the next slide. On this slide, you can see, for example, in the case of dexamethasone, which, as you know, is an anti-cancer drug and also an anti-inflammatory. It's used actually in the case of COVID patients. This molecule has different affinities for the 4 nanotubes, and it varies between -7 kilocalories to -16 kilocalories, therefore allowing a fairly nice control over the encapsulation energy and release of the molecule. Not only that, we can actually tailor these molecules with radioactive elements for SPECT imaging, with fluorine 18 or 19 for PET imaging, or with fluorescent probes for FMT imaging. So they can be not only delivery agent, but they can also be traced using existing techniques for imaging. So it is, in a sense, a theranostic tool for delivery, for diagnostic imaging and drug delivery. On the next slide, what you see is the same series of nanotubes. Here on the left is a nanotube functionalized with a lysine amino acid, therefore the surface is very positive, charged positively. On the second nanotube with the red peptides, that's an RGD peptide that we know binds very strongly to integrin receptors. And on the third and fourth nanotube to the right, those nanotubes are a hybrid of the first 2. And therefore that gives us some leverage on controlling the density of functional bioactive molecules on the surface of these nanotubes. And we have used that trick to decide the nanotube that I mentioned to you. And using this methodology, the clicker doesn't advance as quickly as I want. So these nanotubes functionalized with these peptides can actually bind to the overexpressed receptors, integrin receptors on human lung adenocarcinoma cells, and trigger the MAP kinase pathway and lead to programmed cell death of this human lung cancer cells. Now, the next application draws actually from all this knowledge, and we have looked at this nanotube in a different way. We have thought that by functionalizing them with varying degrees of charges, of positive charges, we can control the charge density on their surface, and therefore the affinity for negatively charged molecules such as siRNA and messenger RNA. And we have tried these systems with different functionalities on the surface to deliver siRNA. So the molecules, this is a very schematic description of the process. The molecules self-assemble into rings, the rings self-assemble into tubes. The tubes bind to the siRNA on the surface and form particles. Those particles show a net positive charge. They are recognized on the surface of the cell, endocytosed, and released inside the cell. And then the siRNA expresses its activity inside the cell. So what happens when you mix siRNA with these nanotubes? On the left panel, so this is the schematic. On the left panel, here is a transmission electron micrograph of the nanotubes without siRNA, and you can see that they have that typical rod-shaped structures. But on the right side, with siRNA, you see particles on the micrograph. And in fact, when you zoom in on these structures, what you see is that there are very beautiful hexagonal structures of this nanotube viewed from the top. So each one of these white dot corresponds to a nanotube viewed from the top. And the dark area between the nanotube is where the siRNA is trapped. So these are the structures that are bioactive. And so when we look at how this siRNA and RNT bind to each other, we did a gel shift assay, looked at the amount of nanotube that could bind to an siRNA. And we found actually there is a very nice correlation between the length of the nanotube and the length of the RNA. And the RNA can grow almost indefinitely. However, in these cases, the growth of the nanotube is controlled by the length of the RNA itself. And you can see here that from the zeta potential, we can determine the charge, the net charge of the nanotubes alone, K1 and K3. And here the negatively charged siRNA. It has a negative zeta potential. The complex is positive with K1 and even more positive with K3 because K3 have more charges. So this is what we draw from this. Obviously, this system is also very stable to enzymes. So the siRNA is fully protected from oncogenic enzymes. When you look at this slide, we looked at the cytotoxicity of the cells of this complex, and we found that the RNAs are actually non-toxic up to 100 micromolar, which exceeds by far the amount needed for the therapeutic effect that we are seeking. On the next slide, we're looking at a variety of cells. We have actually screened a battery of 60 different cells, and we have found that the nanotube with siRNA cargo enters the cell within minutes with an efficacy between 98 and 99%. So it's an extremely efficient delivery system for siRNA. And then we looked at the gene knockdown of these structures. And in this case, we have looked at, we compared this to lipofectamine, which is considered to be the gold standard for in vitro systems. You can see that K1 and lipofectamine are essentially the same against these two cells. And here, this is just additional examples for siRNA that signs STAT3 protein, the expression of STAT3 protein. So on the next slide, what you will see is an attempt at silencing KRAS. KRAS gene is expressed in 90% of pancreatic cells. And here what we can see is on the left side is a delivery of all star cell death siRNA. You can see that the effectiveness of the lipo is slightly higher than K1 and K3. And when we do that for the KRAS expression in cancer cells, what we see is actually the efficacy is essentially similar to lipofectamine. The advantage, however, in this case is that the concentrations we use are significantly lower. And secondly, our material is not toxic. In addition to that, we have taken advantage of the open channel of these nanotubes, and we loaded it with gemcitabine, which is known to be one of the effective drugs against pancreatic cancer. And we found actually that the efficacy of the drug is amplified by a factor of 3. So this leads me to the last part of this particular section of my presentation. And that leaves me with 1 minute 30. So I don't have time to get into the detail of this process, but these are magnetically guided RNA delivery system. We have magnetic nanoparticles that we can coat on the surface. On their surface, we can coat nanotube, and using layer by layer deposition methodology, we can actually layer nanotubes and then siRNA, multiple layers. We can guide these nanoparticles using a static field, so we can aggregate them so that they can respond to magnetic field. We can drive them to the site of action, and then we can use a dynamic field to separate the particles from the aggregate, and then those particles can easily enter the tumor. So that's where we are currently. Before I end, I would like just to tell you about this is a slide that describes the magnetic delivery. And before I end, I would like just to tell you about the foundation that I represent as well. It's Masir Foundation is a nonprofit organization in Morocco, and this foundation has been working for a number of years on promoting science, research, innovation in Morocco. But we have collaboration across the world, established in 2007, and has grown into a very respected research foundation in Morocco, focusing on microelectronics, biotechnology, and material science. And we have, in the area of biotechnology, which is relevant to this conference, we focus on medical biotechnology and green biotech. especially developing by diagnostic tools for a variety of disease. So it was actually the foundation that created the first COVID test in Morocco, and it is now used across the country. And I'll also mention that we have tremendous amount of collaborations since 2007, where now this slide goes to 2020, and we are expanding this network, and we are open. This foundation is a nonprofit foundation and is looking for expanding its reach and collaborating in a variety of fields across the world. And with that, I'd like to thank you for your attention, and I'm happy to take any questions you may have.
Thanks very much, Dr. Fernieri. That was very interesting. And I have a couple of questions. Otherwise, I want to see first if anyone has any question. Otherwise, I'll ask my question. I'll start to ask a question then. You show nicely the conjugation of these nanotubes with silencing RNA, and you mentioned also potentially messenger RNA. It looks like from your schematic that the silencing RNA is sort of attached to the nanotubes on the outside. And when you try messenger RNA, do you see the same? Because I would imagine that probably you will get degradation of messenger RNA faster than a silencing RNA.
We have worked with the short messenger RNAs up to about 300, and we're looking into much larger structures for now. Up to 300, they're perfectly stable, just like siRNAs.
Okay. Great. The other question is, once I saw that obviously once you bind to the receptor, you mentioned it gets internalized with a great efficiency. What's happened to the nanotubes at that point? What is the degradation pathway? What is the accumulation if it happens?
So we use a very, very small amount of these nanotubes. So we're talking about nano or sub-nanomolar concentrations. Okay. These molecules, as I showed you in the previous slide, they are derived from natural molecules, so it's peptides, and the bases are mimics of the natural bases. We are assuming that they are degraded in the same metabolic pathways that degrade peptides and nucleic acids. We have looked actually in detail because the question never, well, obviously this question we have thought about it, but it did not rise to the level of investigation yet because all the animal studies we have done didn't show any toxicity.
Okay, great. Thank you very much. I appreciate it. If there aren't any questions, we can move to the next speaker. Yes? Okay. It's my pleasure to introduce Dr. Madhuri Hegde. She's from PerkinElmer, and she is Senior VP and CSO at PerkinElmer Genomics, and she's giving a talk about Beyond the Exome, whole genome sequences for rare diseases diagnostic. Dr. Hegde, please. Welcome. You're on mute.
Hello.
Thank you.
Thank you, Dr. Martini, and I'm glad to be here, and thank you for the opportunity to speak today. So what I'm going to talk about is we're going to switch gears a little bit into diagnostics of rare diseases. And essentially what we are going to discuss is how we are moving towards whole genome sequencing from the exome. And what I want to start with today is to talk a little bit about my own background and a little bit about PerkinElmer and how this lab really is operating on a global basis. So as we talk about PerkinElmer Genomics,
We lost Dr. Madhuri.
So what we're going to talk about today is the whole genome sequencing for rare disease diagnostics. But if we look at a little bit about PerkinElmer for a few minutes, because it's really important to understand where PerkinElmer genomics sits today. My own background is in academia. I am a clinical molecular geneticist, and I have worked in diagnostic testing for almost 25 years. PerkinElmer genomics exists within the ecosystem of PerkinElmer. And the reason I mentioned this today for rare diseases is that we just heard a talk on the RNAi therapeutic side of it, that we are very rapidly moving towards therapeutics. And what we can do now is bring all of the diagnostics, applied genomics, life sciences, analytical tools together to do diagnostic testing and identify these individuals or patients who can benefit from a variety of different therapeutic approaches. So just as a quick background of PerkinElmer genomics, the foundation is in newborn screening. As we all know that newborn screening is done across the globe. About 150 million babies are born every year, out of which 55 million babies on a regular basis actually receive newborn screening in some form or the other. What we did at PerkinElmer Genomics was build on that foundation and expand into genomics with a full service clinical laboratory, and now a near-term expansion is going to be in omics. The idea really being that we want to do a continuum care model going beyond traditional genetic screening and testing. So what do we actually mean by that? What we are looking at here on this slide is the mother, the infant, and the adult family. And what we are now able to do today with the advances in genomics and omics is do sequencing, biochemistry, and now even with NIPT, using a complete omics capabilities, and that has been our goal from day one. So with that said, just key facts about PerkinElmer Genomics. We have 9 global locations in India, China, Malaysia, Sweden, and the US. As I mentioned, I am from academia, so I myself have published extensively, but together we have almost 300 plus peer-reviewed publications. And we continue to expand our capabilities based on the most recent technological advances and the scientific knowledge that we gain from these advances. So with that introduction, let's jump into whole genome sequencing. You know, the title of this talk really gives you that perspective of that we are trying to go beyond the genome or beyond the exome. And what does that mean? So if we just today look at the milestones we have achieved in genomic testing, you know, for those of you who remember doing Sanger sequencing using radioactivity followed by the fluorescent labeling, and then now the most recent advances going towards the next generation sequencing technologies, and we're going to continue making some rapid advances into nanopore technology. In our own laboratory, we do sequencing using the NovaSeq. We have 10 NovaSeqs in our lab, with the idea being that are we going towards whole genome sequencing? So let's spend a couple of minutes looking at how does whole genome sequencing actually work. Before that, let's look at panels and exomes. Essentially, when we are doing panel and exome sequencing, what we are doing is selecting the regions of the genome to amplify, sequence, to identify the possible variants that could be causing the disease. So here on this slide, what you see is in our laboratory, we use a sure select system from Agilent. We have over 300 panels and the whole exome, which we can sequence using this sure select library. Now, what are the drawbacks of exomes and panels and a hybrid and a target selection method? So in my experience, I've tried everything from PCR-based amplification selection to hybridization-based. And there are plenty of publications now which have shown that hybridization-based methodologies are superior, especially in rare disease diagnosis, to the PCR-based amplification methods. But we still have that PCR step, which can create a bias or an allele dropout and thereby introducing errors. So what we are trying to do here is, can we go straight from our DNA to sequencing itself? And that's where whole genome sequencing comes into play. What we are essentially doing in whole genome sequencing is taking the naive DNA, ignoring all these middle steps, shredding that DNA into tiny pieces, and then sequencing it through a massively parallel sequencing or NGS. Now, just one disclosure here that whole genome sequencing does have a step of PCR, but it's minimal. It's called tagmentation, and Illumina has a nice video on their website of how this tagmentation actually works. So how do we bring whole genome sequencing to the clinic? On this slide, it really shows you how it was important for our laboratory, the PerkinElmer Genomics Lab, to be within the PerkinElmer ecosystem, and how we are using this instrumentation in our own lab. The first thing which we did was, the reason I showed you newborn screening is that newborn screening is done on dried blood spot cards. So today, we can do whole genome sequencing on a dried blood spot card. So we have validated whole genome sequencing on dried blood spot card, saliva, blood, and also tissue types. Now the extraction is done on a Chemagic 360. Chemagic 360 is a very robust instrument for DNA extraction, but recently in the COVID times, it has been widely used for RNA extraction as well. Then DNA quantification is done using the Victor Nevo, followed by library preparation on a cyclone liquid handler, again QCing on the lab chip GX, and then followed by sequencing on a NovaSeq. All this exists within our laboratory, but we also attempted to write our own analysis software, which is called ODIN. ODIN really stands for Ordered Data Interpretation Network. And what ODIN really does, it grabs variant data from the globe, from our own labs, and helps in data interpretation. As we all know that there are, though we are more than 99% similar, there is that minor variation which a clinical geneticist has to look at and differentiate between disease causing or common variation in that population. So let's compare the sequencing options here. Panels, exomes, and genomes. The panels and the exomes target a region, so they give you 100x coverage, a higher depth of coverage. Today, the genome is sequenced at 30x or 40x. But because you're not doing target selection, a 40x genome is comparable to a 100x exome or a panel, or at times better than an exome and a panel. Because the genome now allows us to detect copy number variation, at times some pseudogenes, and I'll talk about that, and also repeat regions. Incidental findings need to be addressed across the board from panels, exomes, and genomes. And I'll give you just one simple example, which is the Gaucher disease gene GBA, which is implicated for Gaucher disease and Parkinson's disease as well. Now, if we do a direct comparison of the depth of coverage, as I just mentioned previously, that a whole exome at 100x is directly comparable to a whole genome at 40x, and a 40x genome is at times better than a 100x exome. And the reason for that is the uniformity of the sequencing itself. What you see in the top panel is exome sequencing with the troughs and the peaks, whereas in genome, you get that uniformity of the sequencing itself. The next thing to look at is the coverage of clinically significant regions. And this slide really says it all. that from the ACMG selected genes to the SGMD database genes to the RefSeq genes, whole genome sequencing gives you better and a more uniform coverage. A question that gets asked very often when I give these presentations is, does genome sequencing give a tremendous amount of data and does it make it very difficult to interpret? I think let's just compare directly to the number of variants in an exome versus a genome, exonic and total variants. On the lower two panels circled in the green box, you see the exome to genome comparison there. If you look at just at the exonic variants, the variant number is quite comparable, where you see this number of genes here and the number of variants. The genome definitely gives you more variant, but you can limit what you see because the deep intronic regions can be investigated in a more tiered fashion. Now, GC-rich regions, as I mentioned, the coverage is more uniform. So what you see here on this slide is on the left-hand side, you see where the whole exome coverage drops off and the uniformity of the coverage from whole genome sequencing across the x-axis, which shows you increase in the GC content itself. On the right-hand side is a single exome comparison of an exome, which is extremely GC-rich, 77%. And you can see that the whole genome can cover that exome versus the exome, actually you get a dropout of that exome. You cannot sequence it using whole exome sequencing. So beyond the exome, the genome really has that ability to capture those variants which we cannot capture using exome sequencing. There are lessons learned. We can identify those deep intronic variants and then tag them in the exome and make the exome powerful as well. So basically what you're doing is investigating the entire genomic sequence of the individual to make sure that you have looked at the gene in its full completeness, but not just the exon and the covering or the flanking intron exon boundaries. Now, let's look at some couple of cases here. The first case is an intronic variant case because I've just talked a lot about having that extra sequence which is a deep intronic sequence, which can become very important. And I'm going to show you a very simple example. You would expect this to be a simple molecular confirmatory step post biochemical analysis. So this is a 5-year-old male with a suspected diagnosis of MPS II, commonly known as Hunter syndrome. Biochemical results showed reduced iduronate sulfatase levels, and Sanger sequencing and MLPA could not identify the sequence variant which was associated with this child. The family was really frustrated and approached us for whole genome sequencing to identify the variant. And what we found was a variant in intron 6 at -72 position, which was causative of this particular condition, the Hunter syndrome, in this child. Subsequently, functional analysis was done, and we were able to move this variant of unknown significance to pathogenic in this particular child. So now this really shows you the power of whole genome sequencing. Let's go to the next step, is detection of tandem repeats. What I've shown on this slide is again a very simple example of the PABPN1 gene, which causes OPMD. And what you can see is on the top panel is the whole exome results, and the bottom is whole genome. And the genome can actually resolve ambiguous repeat sizes detected in exome sequencing. We have done extensive validation of repeat regions, and what I wanted to show you is how those reads actually look. So on the top panel, you see the whole exome data, and the bottom, you see the whole genome data. What you can see is that region where the PABPN1 repeat region sits is not well covered by exome, but is completely covered by whole genome sequencing. So we have done extensive validation of the repeat disorders by whole genome sequencing, and we are doing about 40 disorders now. We can report them. what we are doing is we can rule out, but we cannot rule in. And what I mean by that, repeat disorders which are in the smaller range, where the repeats are about 10 to 40, we can easily detect them, even the expanded repeat, by whole genome sequencing. When the repeat size goes extremely high in 2000 and 3000, we cannot detect that today by whole genome sequencing. But if we see heterozygosity or heterozygous repeats in the normal range, we have then essentially ruled out that disorder. An example will be Fragile X in a male or even a female at times with skewed X inactivation, or the smaller ranges of the normal repeat range in myotonic dystrophy, the DMPK1, where if we see homozygosity for the normal repeat, then a southern blot would be needed for confirmatory testing. This has been very, very useful for those cases which are coming to our lab with specific clinical conditions such as ataxia or a myopathy. Now, the next level of whole genome sequencing is the ability to detect SMN1 and SMN2 copy numbers. And we are today routinely reporting SMN1 and SMN2 copy number from whole genome sequencing data. What you see here on this slide is a publication from 2017, which essentially shows you the region of investigation here and the SNPs that are actually investigated or looked at to distinguish between SMN1 and SMN2. As you all know that it is not very easy to do that because it's a single nucleotide change in exon 7 that really separates the two genes and one deep intronic change. And we have been able to extensively validate that and now are routinely reporting the SMN1, SMN2 copy number, which is important for therapeutic purposes, as we know. And the last one is exon level detection. With a genome and exome sequencing, essentially what we do, and most labs do, is take that data, create virtual probes to detect the copy number variation. But because of the uniformity of the data that you get from whole genome sequencing, it makes it more powerful. What do I mean by that? Can I demonstrate that? So let's look at an example, is utilizing the power of whole genome sequencing for single exon detection in the presence of a larger detection on the other allele. So this is a 6-year-old male with intellectual disability, abnormal hands and legs, speech problem, and there was no family history given to us. What you see here on this slide is a really power of whole genome sequencing where you can detect a deletion within a deletion on both alleles. We've been very fortunate that we have been able to extensively partner with advocacy groups. What you see here is Decode Duchenne through the Parent Project Muscular Dystrophy, because we sequence the entire Duchenne gene. We also have a project called the Lantern Project with Genzyme, which has been very successful for muscular dystrophies. But really the key is, how do we separate ourselves and make this whole genome sequencing technology more amenable to the patients? Is ability to work with any type of sample, the extensive breadth of the menu itself from newborn genome or newborn screening to adult testing, and then the global laboratory network, which is essential to understand the genomic variation globally. So I mentioned previously, we have labs globally. You can see on this slide, really, the labs which are present globally, and we congregate all this data into our OD database for data analysis, for improved and tailored variant interpretation. So with that, I'm going to stop and take any questions. Thank you.
Thank you so much, Dr. Ragde. Great presentation, great talk. Let's see if anyone has a question. Otherwise, I obviously have a question for you. It's very interesting. No, I'll start at this point. It's amazing. I'm always in awe with the progress in technology and how it goes so fast. So my question is actually this. It's impressive how now we can sequence whole genome basically from different patients, and I see that you participate in newborn screening. But how fast can we actually do a full sequence? And the reason is that I'm asking is because if we think of some of the liver metabolic disorder where we have basically only a few days before these children start to develop metabolic decompensation, and before we understand, for example, if it's an organic acidemia or a urea cycle disorder, because we see ammonia, but we don't differentiate between the two. So how fast can you produce the data so that we can, in fact, one day, once therapy will be available for infants and newborn, produce this data that allow us then to be very precise with the type of therapy that we give to these patients?
That's a great question. So today you can, from the time you receive the sample, you can produce the data in about 3 days. What we can do is we can, it depends on how quickly the sample comes to the lab, obviously. We've been doing stat sequencing with a turnaround time of about 7 to 10 days. It really is including, from the time the sample comes to the lab, the DNA extraction piece of it too. But on the sequencer, you can generate data in 3 days. Now, you raised a very good question is, for the newborns especially, which are in the, NICU, who need that data so quickly so that could be some more action oriented. And what we are now able to do is take that data and marry it with the biochemical data so that if there is a disorder especially when there is a disorder that is treatable, you really want to get in there very fast before the clinical presentation has advanced too much. And we are able to do that now and combine the data with newborn screening data as well. And that is why, right, blood spot cards also become very important because you don't have to go back to the baby. We can just take the blood spot and sequence it.
Perfect. That's phenomenal. Thank you so much. I think it's incredible that we can do this. I remember 20 years ago, we were still running gels, counting base by base. And now we have 3 days genome sequencing. So it's phenomenal. Congratulations. Thank you.
Thank you.
I don't think there are any questions, so I'll move to the last speaker of this session, and then we'll have an open panel for discussion with all my colleagues. So the last talk is given by Dr. Majed Al-Ghoribi from KMark, and it's talking about empowering infectious disease surveillance using whole genome sequences. Dr. Majed, please.
Thank you, Paolo, for this introduction. Bismillah ar-Rahman ar-Rahim. Assalamu alaikum wa rahmatullahi wa barakatuh. Today, I'll take you away from the whole genome sequencing of human to whole genome sequencing of infectious disease, especially in the pandemic, the COVID pandemic situations, how the infectious disease surveillance program using the power of whole genome sequencing play an important role to understand the evolution and the transmission dynamic of COVID all over the world. If you allow me, I will start my presentation, with this quote by the great microbiologist Carol, who contributed in the Tree of Life. And he said in the 1998 that genome sequencing has come of age and genomic will become central to microbiology's future. As we are witnessing nowadays that the whole genome sequencing, especially with COVID pandemic, play an important role to understand the virus, the movement of the virus, and the evolution of the virus. Infectious disease surveillance is a cornerstone of public health. Without infectious disease surveillance, then we'll be able to identify the emerging and re-emerging pathogens that hit the world. As I said, by having infectious disease surveillance, that will help the community and the public health to understand the threat of infectious disease. The traditional surveillance program was using the public health data and the clinical data and diagnostic data. But recently, I think starting from 2003 to up to 2010, there is a revolution of whole genome sequencing, next generation sequencing technology that's come to the science where has been used for infectious diseases. As I said, COVID is a point, become a turning point of genomic history, where whole genome sequencing of COVID never been done before in any other pathogens in the history. Especially for the global surveillance of SARS-CoV-2, we have witnesses a cooperative approach of all countries where they have implemented whole genome sequencing surveillance to identify the whole genome sequencing of the virus all over the world since the start of the pandemic. There is a lot of initiatives has been contributed to the surveillance of SARS-CoV over the last year and a half. And there is a huge database has been created using all the generated data by every country to see the dynamic transmissions of the virus all over the time since the pandemic started. One of the most famous and important database is the GIS-AID initiatives, where all the scientists and research centers and public health agency have deposited whole genome sequencing of the virus since the starting of the pandemic. Other tools of the bioinformatic and database has captured this data and put it in a platform where this data has been interpreted using bioinformatic tools to identify the variants and the transmission of the virus. Can you imagine that only in 1 year and a half, almost 1 year and a half, almost 3.5 million whole genome sequencing of the virus has been generated by all over the countries, all the globe. This has never been done for any microbial organism or even human genome. This is a very huge data, is enabling us to understand the evolution of the virus in a very short time. As you can see here in the left tables, that the total genome sequencing has been done by each country. As you can see here, United States has generated almost 1 million whole genome sequencing of the virus, coming after that UK. But to compare that and to see the contribution of the whole genome sequencing, we should do a bit of statistics to see the sequencing per 1,000 cases. As you can see here, some of the countries, they have almost sequenced most of the cases that is reported in their country, including Ireland, Iceland, and New Zealand, and Denmark. That really helps this country to improve their infection control precautions to contain the virus, and also to understand and to determine the variant that's circulating in these countries. Also, by having this huge amount of whole genome sequencing data, we were able to do identify the classifications of the variants. As you can see here in the chart, the first classification of the virus is the virus variant of high consequences. This is fortunately that's never been determined. That is really more severe and make the patients having really severe infections that allowed to stay in the hospital for a long time. The second one, which is the most worrying nowadays, is the variant of concern. We have around 4 variants of concern nowadays circulating worldwide. And also, there is many variant of interest that even has been monitored by the Public Health Agency to monitor these variants to make sure that it is not becoming a variant of concern. Also, many countries have identified variants under monitoring. This is still under monitoring, and there is many of them in the database. This is the most famous variant, has been detected over the last year. Alpha, which is belong to the B117 lineages, and this is first detected in UK. And also beta, which is similar to having similar mutations to the alpha, has been detected in South Africa. In KMARC, we have recognized the importance of whole genome sequencing surveillance of infectious disease early on, starting from 2015. We have been working on this to establish a state-of-art whole genome sequencing facility in KMARC to be able to tackle any issue in infectious disease. The vision of this surveillance program is to halt the dissemination and reduce the threat of infectious disease in Saudi Arabia and the regions. And our objective is to conduct a large-scale national-wide surveillance to track routes of disseminations and monitor the trend of emerging, re-emerging pathogens over the time in Saudi Arabia and the region. If you allow me, I will take you through our journey of developing this surveillance program, where we were concerning about building the capacity, transfer the technology and knowledge to our center to be a kind of a leading center of whole genome sequencing in the region. We're starting by building capacity. We were focusing to establish the microbial genomic lab to bring the most advanced sequencing platform in our lab. That will allow us to sequence pathogens that never been sequenced in Saudi Arabia to generate cutting-edge research in the region. All that is coming together to serve as a surveillance program to monitor the dissemination of high-risk pathogen in Saudi Arabia. And also we were focusing here to build the human capitals and train scientists and technologists in our center. Also, we are serving as a reference lab to most of the National Guard hospitals, where we serve this lab when there is a difficult diagnostic of a certain infectious disease in these hospitals. This is including a manual detections of unknown infectious disease, unknown pathogens, using advanced molecular techniques. In 2018, we started this surveillance program by building a strategy of infectious disease in KMARC, where we have the planning phase, where we start by designing the experiment and making networking with all the National Guard hospital in Saudi Arabia to be able to create a comprehensive surveillance program by designing a random screening as well as design screening for a certain pathogen that is become a high priority in the region. Also, at the same time, starting with 2019, we've been able to bring the whole genome sequencing platform in KMARC, where we're able to sequence these pathogens. I will give more details about the whole genome sequencing platform that we have in KMARC. Nowadays, we are in the analysis phase and development phase, where all these whole genome sequencing generated in our lab are being used to develop rapid diagnostic tests, or also to contribute in the vaccine development. We have in our department Illumina platform. Because, as you know, the genome of the microbe is very small comparing to the human. And Illumina, they have different sequencing platforms, starting from MySeq, which is a small throughput sequencing platform. This is kind of suitable for a small lab who is working to sequence minimum around maximum 100 pathogens per month. We're starting by the small scale proof of concept to make sure that we have developed the pipeline from the sequencing up to the bioinformatic analysis, where we can interpret the data and understand the evolution of any pathogens we are sequencing. The only problem with this approach is required a manual approach to do a library preparations, but that can be done, can be avoided by developing automated library preparation. We have this facility in our lab as a small scale when we need it, but also we try to have a high throughput sequencing platform with a collaboration with the MGI. We have in our center an automated automated machines for library preparations, and also an automated machine for nucleic acid extractions. All this, when we go back to Illumina, here we can sequence around from 25 to 30 bacterial pathogens. This is a small scale, and also for viruses we can sequence up to 100 virus. This is depend on the size of the genome of the pathogen. When we compare it to MGI, this machine can sequence around 300 bacteria in 2 days. And that will help us to move from a small to high throughput scale, and also it's important to associate that with an automated library preparation. Just going back to the surveillance program, we have 5 national hospitals in the region, in Riyadh, Dammam, Al-Hassa, and Al-Madina, and Jeddah. All these hospitals are sending a sample on a monthly basis, started from early 2018. And we have designed a selection criteria for this pathogen to be sent in every month. And then after that, we collect all the clinical data associated with this pathogen, just to kind of to have a platform where we can have a selection criteria for whole genome sequencing. Not all the isolates are sending from this hospital are sequenced, but we do a selection criteria for whole genome sequencing, just to have a designed sequencing surveillance. There is 3 core components of the infectious disease surveillance system. The first one is the surveillance site, which is each hospital, and each hospital they has a clinical microbiology lab, and also infection control department. We are coordinating with this department where the clinical microbiology lab, they send us the isolates and all the diagnostic data, and also we communicate with infection control to collect any epidemiological data that associated with each sample. By this, we have created a database for pathogen biobank. All this is taken to be in one platform. The second component of the surveillance system is the AMR NGHA Coordinating Center, which is KMARC, where we collect all these isolates by a team of networking and communication, where they communicate with these hospitals. But also we have a data management and quality control, where all the isolates in a pipeline. Some of them, they just go for the pathogen biobank, and some of them go for further molecular investigations. Then we would confirm that these isolates is meeting the selection criteria for whole genome sequencing. Then we'll take these for whole genome sequencing stage. This is, I'm not going to go in details, but this is the workflow and the pipeline of microbial genomic. We do a manual phenotypic genotypic detections, and also we do a whole genome sequencing ending to the bioinformatic analysis. We have managed to develop an in-house bioinformatic pipeline for antimicrobial resistance bacteria. We have a specific pipeline for each pathogen. For example, we have several pathogens that we collected over the last 3 years. Each pathogen has its own database, own pipeline software that will enable us to generate enough data to understand the evolution of the virus, of the bacteria. In addition, we started in 2019 by sequencing MERS-CoV, which is the virus that hit Saudi Arabia in 2012. And because the number of cases was very low at that time, we managed to collect around 50 whole genome 50 isolates of MERS, and we worked to develop the viral whole genome sequencing pipeline during 2019. By the end of 2019, we managed to sequence 50 isolates, and that will enable us to be ready to sequence any type of viruses that we gain in good experience over that sequencing. Before we go into the SARS-CoV whole genome sequencing, our experience in KMARC, let's start with antimicrobial resistance. This is the main surveillance system that we built. We used the most priority pathogens that has been identified as a high-risk pathogen in clinic: E. coli, Pseudomonas, Klebsiella, and Acinetobacter. We collected more than 20,000 pathogens over the last 3 years. All these isolates being through the pipeline for a selection criteria for whole genome sequencing. This is a number of publications that we have generated over the last 3 years, starting with a message that we have sent to this community to introduce whole genome sequencing in our region. With collaboration with Public Health England, as I did my postdoc in Public Health England in London, I tried to transfer their experience in a national surveillance program to Saudi Arabia. And we put this position article in the corner in the left there as a guideline roadmap for whole genome sequencing surveillance program. Also, we sequenced several pathogens, including E. coli, Klebsiella, that's showing a pan-resistance. Pathogen are resistant to all the antibiotic available. All this experience is cumulative to build all the protocols for an effective surveillance program. On the other side, we were trying to cover a little bit in a parallel to clinical pathogens. We were trying to look for animal and food microbiology, including Salmonella and Brucella. This is just an example of our analysis. We usually end up with a genome analysis of high-risk pathogens that are harboring genomic elements, for example, like this circular plasmid, which is harboring the resistant genes that enabling the bacteria to emerge and become more resistant. This analysis is a downstream analysis. Just we look for the genes and the mobile elements, including the plasmid, the insertion sequencing of these pathogens to understand the evolution of the antimicrobial resistance in the region. For each country, usually there is a type of resistance genes that are circulating all over of the country, and we are trying to take the surveillance program to the national level, and also not only to the national, regional, and hospital, we call it local surveillance, to understand if each of these plasmids are circulating in one city, or circulating in one hospital, or even circulating in one ward as an outbreak in a hospital. This is another example of one of the first KBC genes that are detected for the first time in Saudi Arabia. I think it's been existed in Saudi Arabia for some time, but because there is a lack of whole genome sequencing surveillance in the region, we were able to pick up the first case in Saudi Arabia. Taking all this experience together, we have been invited by the WHO to meet, to visit Geneva for the first meeting of the GLASS whole genome-based MR surveillance committee. This was held early in 2020 before the COVID, but that enabled us to develop a technical note where we can use our experience and other country experience to be together in this technical note to help other countries to implement whole genome sequencing surveillance in their countries. Going back to the COVID, MERS, and SARS viruses, as I said, we have gained a great experience dealing with MERS from human and camels. We have sequenced a good number of isolates comparing to the cases reported in Saudi Arabia. And also, by March 2020, when we have the first patients in National Guard, we were able to have the RNA of the virus in just one day. And when we got the RNA, we've been able to sequence the virus only in 3 days. All over the time since the starting of the pandemic, we've been sequencing in a designed screening. We are not sequencing every case that we have in our hospital, but we've been sequencing from time to time. By the end of 2020, we have observed one of the interesting mutations that has appeared in our sequencing, which is the 501T. This is similar mutations that has been detected in the UK and South Africa variant, and then we focused to look for this variant more further. Then in around February 2020, we sequenced a huge number, 300 isolates from 1 month. We found that 60% of these isolates contain this significant set of mutations that never been reported worldwide. Then we kept our sequencing facility going to see if that is a real variant or variant of interest or variant of concern. We collaborated with the Infection Control Hospital in Riyadh, and we have collected all the clinical data of the patient who been infected by this variant. We found that only around 6 patients from around 150 has been admitted to the ICU. Most of the cases were community-acquired infections, and that was a relief for us because this is not high-risk pathogens, not a variant of concern, but variant of interest that we need to keep watch all over the time to make sure that is not really causing a severe infection. Our experience here helped us to see where we can track these variants all over the time to see the source of the variant. We have done a global phylogenetic analysis. We took a prospective whole genome sequencing from all over the globe to compare our isolates with the other variant reported. As you can see here in the middle, there is a clade in a red color having the same mutations that come into our interest, the 501T. But when we look for that, we found this is a cluster of isolate that has been sequenced and isolated from monk in the United States early in the middle of 2020. But as you can see here is the line where is the present and absence of this variant. In the left side of the slide, you can see the red, there is a clade that is having a significant set of mutations not existed in the other isolate. And in the bottom of the screen, this is a zoom in to that tree. As you can see, these isolates mostly detected in Saudi Arabia started from November 2020 until we have a peak in February 2021. As you can see in the left side, this isolate has, Saudi Arabia has reported the most number of these variants comparing to other countries. We worked closely with the Next Strain. Next Strain is a platform for phylogenetic analysis and clade analysis of each of SARS-CoV-2 viruses. We have built a national platform, Saudi Arabia platform, to compare our variants that we are sequencing all over the pandemic. As you can see here, there is a clade with the alpha variant and the gamma variant and the beta variant. And the clade in the yellow color, it's the Saudi variant that we have detected, which is fortunately is not a high-risk variant. I think our experience over the last 4 years become a kind of a proof of concept to establish a whole genome-based surveillance program where we really saw the important and the major role of whole genome sequencing to identify the evolution of the emerging and re-emerging pathogens, where we need always a real-time data on occurrence of high-risk infectious agents through a standardization method. We need a standardization method that is really meet with the international standardization protocol, just to make sure that when we share our data and compare our data with the global data, then that will be compatible. Also, there is an urgent need to enhance sample and data collection for the national level, and also we need to develop more molecular investigations methods across all the compartments of human, animal, and health. Detections of related infectious agents in the time and the locations through phenotypic and the genotypic. We need to combine all the data that is generated in the phenotypic and the genotypic profiling. Also, we have to think about the possibility to observe trend in clone variant as we have experienced in SARS-CoV, and also the ability to rapidly compare between all the type of data using a common language. I think one of the most important messages that I would like to send tonight is the national capacity building. We need more whole genome-based surveillance system in the country, not only in KMARC, but also we need a collaborations and consortium of whole genome sequencing surveillance where we can all share data and then create a comprehensive pictures of the epidemiology of infectious disease in Saudi Arabia. Without monitoring and surveillance program, we are blind to the infectious disease issues. Developing an effective surveillance program aims of the reference labs in every region and country. Each serve as the hub and national network and contributing to the international network. Each performing essential testing, including both phenotypic and genotypics testing, each following international standards. And also, one of the most important elements for this type of surveillance is sharing data. No real-time surveillance without no real-time data sharing. That means we have to create a common platform where we can deposit all the data for the national surveillance system, just to be able to compare that data and then come up with a clear pictures of the pandemic, of any issue in pathogens. By the end of this presentation, I would like to thank the IDRD team in KMARC who is contributing to establish the AMR surveillance program, and also all the hospital, National Guard Hospital in Saudi Arabia. Thank you so much.
Thank you, Dr. Al-Ghoribi. This is phenomenal. Congratulations on the incredible work. You're truly contributing to the safety and the health of the world by doing this work. And I'm glad that you're publishing and more importantly, you're planning to share and contribute and collaborate with international organizations. This is very important. As we have seen what happened with COVID-19 that created a the meeting today. So I would like to pass it on to my colleague, Dr. Majid, if he has an initial remark for the panel. Otherwise, I can start as well.
Thank you, Pablo. You can start now.
Okay, I just, first of all, I want to thank everyone for participating in this session. It's been incredible. We have seen an incredible presentation today, but more importantly, what I see that there is a vision from the kingdom to really not only contribute with the rest of the world in the safety with obviously this incredible presentation and also the ability to really maybe start to build something with the vision of helping the patients in the kingdom. So this meeting for me was an incredible experience and humbling, as a matter of fact, in understanding the vision that this country has in moving forward, but not only in rare genetic diseases, but infectious diseases, etc. So I just I wanted to congratulate everyone for this, and more importantly, I would like to start maybe a conversation on what is the next step for KMark and eventually for the kingdom to really start to tackle down what are the key issues and the key implementation that need to happen in order to bring potentially more medicine and more ability to run clinical trials within the kingdom and eventually to contribute to the human health.
Thank you, Paolo, for this excellent exposition. I think in KMark and all other institutions in Saudi Arabia and research centers, we didn't have deficiency in science or in brains. I think what is the issue that we need here is to facilitate mainly the logistic. So we need the international companies to present more here and develop their infrastructure here so we could exchange the information, utilize the rich, resources that the country have. And I think this summit is the initial step toward this endeavor. I am sure with the 2030 vision, this kind of collaboration and effective communication between industry partners and KMark and other research centers will be more and more in the near future as reflected by the summit.
Great. I think it's very important as we see now new technology coming to life. It was great to see Dr. Fadouri presentation as well, new possibilities for targeting tissues and different diseases. So there's quite a bit of growth and development in the technology. Also, obviously I presented about messenger RNA. But right now it seems like the next step is really to be able to access all the information that you guys have been collecting with great publications and more importantly with incredible work that has been done even in rare genetic disorder, what you presented in terms of understanding mutations diseases and the characterization of the diseases is an incredible work that not many people can actually do. And you have obviously the ability to contribute incredibly to the patient population by obviously creating these databases of patients that you guys have, and it's just incredible. So I would like to hear from other colleagues if they have any remarks or questions.
I just have a question, Pablo, if you allow me.
Yeah.
I want to ask Dr. Maduri regarding whole genome sequencing. I know Birken-Elmer sequence a lot from Arab population. Are you willing to share the data that you sequence with our research center and other institutions in Saudi Arabia? Because, you know, this data is very important in order to reach the precision medicine and in order to have our reference genome ready. We have a lot of variant of uncertain significant because actually we lack the reference genome. And I think if we have collaboration with an international company like Birken-Elmer and Syntogene and other companies who enter the market in the Middle East, I think we will reach our reference genome easily.
We will be very happy to collaborate and share the data. It is not an issue at all. In fact, we do a lot of sequencing from your region on a daily basis. We are doing a tremendous number of whole genome sequencing cases. I myself report at least 20 to 30 whole genome sequencing cases from your region on a weekly basis. So we do have a tremendous amount of data, and you are absolutely right. This is the only way to resolve the variants of unknown significance. Because otherwise, if we don't collaborate and share that data, these variants sit as variants of unknown significance. There's another initiative I have started, and I would welcome the collaboration there, is that many variants of unknown significance, as Dr. Martini mentioned, is liver diseases or any pediatric disease can be moved to pathogenic if the physician has made the diagnosis and started some sort of a treatment and has received a response. We are collaborating with a lot of pharmaceutical companies to enable them to get into clinical trials into various regions across the globe. So we'll be very happy to collaborate and share data.
Thank you. The other question I have regarding the storage, when we did our whole genome sequencing in Kmark, we faced these challenges of storage. Are you keeping the BAM file and other file, or you just save the VCF file? And tell me about your experience on storage, how you overcome this.
So we store all the FASTQ files. We do not discard any FASTQ files. And every 6 months, I can tell you, we routinely get asked by physicians to reanalyze the data. And we find new genes and new variants that can be moved from genes of unknown significance to a gene causative of disease. So we store all the FASTQ files and routinely move them into our Odin software for reanalysis and reinterpretation.
Thank you, excellent. The other question to Paulo, you know that many now technology is running regarding the therapeutic genomic, including CRISPR, including microRNA, including messenger RNA. Why your company choose messenger RNA? Why you didn't go to CRISPR and these technologies?
So we are not excluding these, right? Right now, when Moderna started, they wanted to focus on messenger RNA, and they put a lot of effort in understanding messenger RNA and to make it become basically the best therapeutic so far in the clinic. I just don't want to brag about it, but as you have seen many of the data recently that they've shown the superiority of the Moderna vaccine compared to other, and mainly due to the fact that the company has invested a lot of money and a lot of people understanding messenger RNA, lipid nanoparticle, how to create the perfect, for example, a spike protein for a vaccine as an antigen, et cetera, et cetera. So when you invest so much in understanding the type of platform and therapeutic that you have, you focus initially on really socializing that and then making it become a real therapeutic. It's not excluded that in the future, in fact, and we are already looking at other technologies that involve nucleic acid, because after all, nucleic acid is our expertise, which will potentially include the use of aptamers, silencing RNA in combination with messenger RNA, or CRISPR technology using messenger RNA as a way to deliver the protein, the Cas9 or whatever. So we are thinking about that, but for the moment we were focusing exclusively on messenger RNA because we wanted to become really the leader in the world in understanding what messenger RNA is, how we can make it suitable for human, and what type of delivery we need in order to target many different diseases. We still have a long way to go. The CNS, for example, is an organ that is still tough to address, right? And lipid nanoparticles seems not to be, for the moment, the way of delivering. So it's always interesting to understand also from Dr. Feniri, for example, a different way of targeting a delivery system, using a delivery system. So for us, messenger RNA right now is the future. It doesn't mean that we won't explore any other nucleic acid, because as we build expertise in that and we are becoming the leader in the world, we want to be sure that we create the perfect medicine, as perfect as it can be for these patients, right? And so you won't be surprised in the future if you see messenger RNA combined with different nucleic acid, because of the complexity of also of the disease that we are going to face, right? Right now we are sort of exploring the low-hanging fruit, infectious diseases, vaccine, monogenic disorder. But in the future, we will need to probably target different diseases that are more complex, and different technologies are needed in order to be able to be a little bit more specific in the therapeutic intervention.
The other question, the prevalence of rare diseases in Saudi Arabia is really high. I mean, it's, as we mentioned in our lecture, it's around 4 times higher than other population. However, we see the clinical trials conducted in United States and in Europe. What do you expect, or why? What is the driver for you to go to Saudi Arabia and do the clinical trial there? And what is the challenges right now you are facing that limiting you from thinking about this solution?
So, and I'm generally very direct and honest on these. The challenges right now is that obviously phase 1 trials has been run in other countries because there's experience in running phase 1 trials in those countries. But this doesn't take away that, as far as I understand it, you guys are actually, in fact, getting ready, and also already got it ready for phase 1 trials. So my collaboration that I've been discussing with Dr. Houdi is, in fact, to come to the kingdom and to start to run the first phase 1 through the kingdom, mainly because I would like to see this happening. You have an incredible characterization of your patients. A clinical trial in your country will be much easier to run because of the understanding that you have on the patient population, the stratification of the patients that you have. Making calls on the patients that are eligible for the clinical trial will be much easier in the kingdom, in fact, because of the characterization that you have on the mutation, the metabolites and everything. What I've seen in the past few years when I came and visit, there was an incredible effort to characterize these patients that for a phase 1 clinical trial is absolutely important and paramount to be sure to select the right patients for the trial. So I would love to come. One of the things that was initially posing us was the ability to actually do a phase 1 trial, which I believe now you are getting ready for it. And that's why we are discussing a potential collaboration, because I think it will be important, in fact, to come to the kingdom because of the homogeneity of the studies that we could run and the response to the drug that we could have because of the data that you have on the patients. So as you developed more the phase 1 trial capabilities at KMark, I think you will attract more not only Moderna but many other companies that will feel at ease to be able to do such work in a country that doesn't have an history for phase 1, but has a great history for phase 2 and 3, right? So, and you know, when you test a new drug, is always tough, right? Because there's always the potential of toxicity, tolerability, etc. Right now, we are starting to have our own initial data from the trial in the US and the UK on safety and tolerability. So as we feel more at ease in proposing our drug to many different countries, I think the kingdom is the next best place to do it. So I'm planning to do that.
Sounds great.
Yeah, and I think we are discussing, in fact, a collaboration, and I'm hoping that within a few months we'll be able to finalize it. And there's quite a bit of work involved in that, and I'm so proud to be able to have initiated that, because I think it is the future.
Yeah, that's great. One of the most important elements that we should develop in the national level, that, as Dr. Majid Al-Fadl mentions, the data and the storage of the data. Not only the storage, but we are really need to have a high-performance computing that allowed to do a high-throughput sequencing analysis, which need a power of computational power that enable to do a sequencing. Fortunately, for bacterial pathogen, bacterial genomic, it's easy. It's a little bit doable. But when it comes to the human genome, and then I think me and Dr. Majid always, we are fighting for to run a job in our high-performance computing. And we have usually waiting for the whole genome sequencing of the human to finish, and then we are waiting to put our jobs. I think one of the excellent example I have experience in England, they have built a cloud high-performance computing, a bioinformatic where people able to submit jobs and use that for a high-throughput analysis. Maybe in Saudi Arabia, I think one of the initiatives that should be planned is to have high-performance computing that serve the academics, research, and clinics.
Yes, I agree totally.
Good. I think we are running a lot out of time, but you guys tell me, otherwise we can close this session and give the time to the Master of Ceremony to close the meeting. And so I would like to thank everyone. Thank you first of all, thank you for inviting me. It has been an incredible experience. And it's been always truly informative to be able to share incredible presentation with my colleague today in the session. And thank you for an incredible meeting. This was amazing. And thank you for the organizer to inviting me as well.
Thank you.
Absolutely. Thank you, Dr. Paolo, and your esteemed panelists for a great session. Ladies and gentlemen, we have reached the end of the Riyadh Global Medical Biotechnology Summit 2021. I would like to express my appreciation to all the participants for taking time out of their busy schedule to attend the summit virtually for 3 days. And thank you all to the speakers who joined us from around the world and shared with us their insightful views and rich knowledge. And it was my pleasure to be the Master of Ceremonies for the Riyadh Global Medical Biotechnology Summit 2021 and work with our amazing speakers locally, regionally, and internationally. And now for the closing remarks, I want to welcome with great pleasure Dr. Abdel Ali Al-Hawdi, Head Strategy and Business Development, Managing Director, Medical Biotechnology Park KSA, and the Vice Chairman of the Riyadh Global Medical Biotechnology Summit 2021. The stage is yours, Doctor.
Thank you. Assalamu alaikum wa rahmatullahi ta'ala wa barakatuh. Good evening, good afternoon, and good morning to you all, wherever you are following us in the Riyadh Global Medical Biotechnology Summit 2021. As just been said, we are now reaching the end of this summit, but it's not the end of it. It is actually just the beginning of a long journey for this great country where medical biotechnology will be a mean to position the country as a gateway for medical biotechnology, actually a global gateway for medical biotechnology. Now, let me tell you how we get to this wonderful, fascinating expression that I just put there. If you look at the program of the summit from day one until today, 9 sessions, if you look at them, it was not actually a random selections of topic somehow related to medical biotechnology. It was actually a clear attempt to take all of you, including, of course, the great people of this country, the region, and around the globe, on what does it take to go from great science, that is really the basis, to an economic impact. So we started with the high-level considerations. What are the elements of a biotechnology setup ecosystem, incubators, biotechnology parks, science and technology parks, you name it. Some of them are sharing facilities, some of them are sharing platforms, some of them are sharing management structures, funding facilities and support. Diving now into really how those companies are successful. What do they need to do? Shall they call on CROs, CDMOs? How do they really develop their product? How do they manufacture? What does it take? Now we're going backward because really we start the right way. The right way for us is that we start by developing a strong foundation for solid science. Without that great science, we cannot move forward. Now it's time to start translating that science. And here I would like to quote my dear colleague and friend, Ron DePino, when he said that great publications will not cure patients, but the translation of those great publications into products, into devices, into drugs, into vaccines, that will cure patients. And that's exactly what we're attempting to do. Whether it's through KMARC or other institutions in the country, that is the way forward. Now, from that consideration, we give you highlights through a number of distinguished global guests from biotechnology companies, pharmaceutical companies, academic elite institutions from around the globe, but the science, genetics, genomics, gene editing, cellular therapy, stem cells, gene therapy, vaccine research, vaccine development, all of it. So in reality now, if you put all those nice sessions together, you will feel that is a complete story, complete journey that we are all attempting to go through. A few minutes before I came to the stage, my colleague just told me that we had about 175,000 viewers of this summit from over 150 countries. That is quite impressive while we are at the beginning of this journey. And hopefully, next time when we meet, we will tell you more about where are we, what we have been able to achieve, and hopefully what we can do together with local partnership and our global partners. Without any further delay, I would like to call my great colleague, Dr. Ahmed Laskar, the Executive Director of King Abdullah International Medical Research Center and the Chairman of the Riyadh Global Summit for Medical Biotechnology 2021. دكتور أحمد.
Thank you, Dr. Abdul Ali, Hawdi, and what a wonderful summit. I'm really thrilled and impressed, not only with the attendees and the intensity of attendance and the viewing, but with the program and the whole content of these last 3 days. It's been wonderful 3 days that have been rich with science, exchange of expertise, collaboration, signing MOUs, and most importantly, the will and the enthusiasm that we have seen and witnessed and felt throughout these 3 days. Although we have most of the attendees are online and at a distance, however, we see the enthusiasm and the intensity again to learn more about this biotechnology. As you said, Dr. Abdul Ali, we are not only restricted to science, but we ought to move forward again to translate this science and those publications into tangible products that will impact not only the health of the individuals and the communities, but also the global economy. And we see this, fueled with the in the Kingdom of Saudi Arabia with a vision, the Vision 2030, that wanted to have alternative to the oil production, and that would be science and innovation. I would like to thank His Royal Highness Crown Prince Mohammed bin Salman for his patronage and for leading this vision, which this ought to be part of it. And also I would like to thank the Minister, His Royal Highness, the Minister of the National Guard, Abdullah bin Bandar bin Abdul Aziz, again for his attendance of the opening ceremony and for his follow-up of the agenda and the program for this summit. I would like to thank the attendees, participants, and the speakers, and all of you guys, and those organizers. There are so many people that ought to be thanked, again, for putting this all together. I wouldn't say goodbye, but we ought to see you again in another meeting and another conference, and hopefully, most importantly, to translate what's been discussed and what's been signed into reality. So we need to see it on ground, and we need to work hard, again, to translate all of these discussions into tangible products and with our collaborators. So without further ado, again, thank you very much, everybody, for your attendance and for staying late tonight here in Saudi Arabia. However, we shall see you in another conference and another meeting very soon. Thank you.
الآن وقت الصورة الجماعية اللي أجلناها إلى آخر الحفل. لو تتفضلوا جميعكم على المسرح.